Faculty members, researchers, and dear participants. We now move forward with the academic proceedings of the day 2 of the 7th Annual National Conference of Midnapore City College, MCCCON 2026. At this moment, it's my privilege to felicitate our esteemed guests for the day. May I first invite Dr. Bimal Prashanna Mohanty, former Assistant Director General, Inland Fisheries, Fisheries Science Division, ICAR, New Delhi, to kindly come to the stage. We are deeply honored by your presence, sir. A huge round of applause, everybody. Again, a huge round of applause, everybody, for this auspicious moment. May I request our research scholars to felicitate him as a mark of our respect and appreciation. A warm round of applause, please.
Good morning, everyone. May I now have the honor to felicitate Professor Rosmita Mohanty, Professor and Dean, Faculty of Science and Technology, Ramadevi Women's College, Bhubaneswar, off stage. May I now request our scholar to felicitate ma'am. Join me in big round of applause, please. Thank you, ma'am.
With this, we now proceed to technical session 3 on the theme Animal Biotechnology and Sustainable Development. This session will explore critical intersections between biotechnological innovation, animal health, ecological balance, and sustainable practices. This session will chaired by Professor Dr. Subhasachhi Pal, Associate Professor, Department of Pure and Applied Sciences, Midnapore City College. Sir, we are grateful to you for kindly consenting to chair this important session. I request the chair and our esteemed speakers to kindly take their places as we begin technical session 3.
Honorary Director Sir, Principal Sir, Vice Principal Sir, present dignitaries, my fellow faculty members, and dear students. Today, it's a privilege to be present in the third session of our conference, and our first speaker is Dr. Bimal Prashanna Mohanty. Dr. Mohanty is a senior academic leader and scientist with over 3 decades of contributions to veterinary biochemistry, aquaculture, and institutional development in India. He currently serves as Professor of Veterinary Biochemistry at IVSH, SOA Deemed University, Bhubaneswar, and is the president of the Society for Veterinary Biochemistry and Biotechnology of India. He has previously held key national leadership roles, including Assistant Director General at ICAR headquarters, New Delhi, where he guided national programs on inland fisheries, aquatic health, and sustainable aquaculture systems. Dr. Mohanty earned his BPSc and AH from OUAT, MVSc from IVRI, and PhD from GNU. Followed advanced research training in proteomics and at the University of Aberdeen, United Kingdom. His scientific work spans proteomics, nutrigenomics, fish stress physiology, aquaculture nutrition, and functional foods, with a strong emphasis on translational research for sustainability and nutrition security. He has led major national projects, organized high-impact scientific conventions, and contributed extensively to policy formulation in fisheries and livestock sectors. A prolific writer and editor, Dr. Mohanty has served as editor-in-chief of the Journal of the Inland Fisheries Society of India, member of the Editorial Advisory Board of CABI, United Kingdom, and reviewer for several reputed journals. He is widely recognized for his expertise in scientific communication, manuscript development, and strategic institutional planning. His current work focuses on omics-driven functional food research, molecular biosensors, and capacity building for India's agricultural and veterinary sciences. He is a fellow of the coveted National Academy of Agriculture Sciences, New Delhi. I will request Dr. Mohanty to start his presentation.
Good morning, ladies and gentlemen. সবাইকে আমার নমস্কার, ভালোবাসা। It's a great feeling to be here. All the dignitaries and the dais, the principal, vice principal, director. It's a great feeling to be here amongst you, especially when you see several young faculty, young students are there. Primarily, you see the age comes down. I am a superannuated person. If you take average age of this room, it may be, I think it may be 27, 30. So I am also in that 30 age group now. So it provides us energy really when we interact with the young students and young faculties are there. There is exchange of knowledge. It is the best thing, I think. Like the gurus, they give discourses, religious discourses. So for scientists and researchers, senior people, young people, middle group people, when you discuss science, like we are sitting in the principal's room, and the esteemed principal was narrating, and we could link to many things. Where you started, where you studied, who was the guide, and all the what are our research interests, all these things. And science is truly, this seminar is synthetic biology, trends in synthetic biology and sustainable development. And really it is, science is synthetic. It is multidisciplinary research. Present day science is multidisciplinary in nature. So when you do research, I do biochemistry, I do immunology, I do proteomics, I do, you have to have bioinformatics, then you know a bit of statistics, several things you know. You to find a solution to any problem, you have to be a master of several things, or at least you have a team of scientists working in different areas and putting together our heads and trying to sort out a problem. That is the nature of present day science, and it is real synthetic. So for this talk, again in the beginning I will try to thank the organizers for bringing me here. Last time also I was supposed to come, but because of some other assignments or some health issues in Delhi, I could not come physically. Online I delivered a talk. So this time I promised madam that yes, definitely will come. So I was in USA last month, but before coming also in Bhubaneswar there are some little bit of issues. I was not sure whether I will be able, but I wanted that no, I must be there physically. Because it is beneficial to me also, we interact with people with, we refresh our knowledge, people ask questions, so we get ideas. So this is what science is all about, and sharing science. And I think another important thing is national responsibility. In the sense, I have grown to this age, government has paid my salary, I have got scholarships, fellowships, whatever. We have gone abroad and government has spent the money. So this is all taxpayers' money. So we owe a lot to the country and countrymen. So we must transfer, we must inspire people, young minds must be ignited. We all belong to the land of Dr. APJ Abdul Kalam, Swami Vivekananda. So we all carry their philosophy. So we are standing on that legacy. So if the country is to progress, 2047 we want to be a developed country. If all these things to happen, so our country, the maximum population of our country is now youth. So young people should be, young minds should be ignited. They should do the research. We should do a hand-holding for some of them. We should lead some of them, and that is a national responsibility. So with this background, it's my great pleasure to be here and share some of the work we have done and how we should move. So the outline of my talk is, I will be talking basically tackling the menace of chronic arsenic toxicity in India and elsewhere, the way forward. You know, arsenic is a big issue, and we will be coming to discussing that. Before that, I have organized my talk to this: nutrition security, sustainable development goals, One Health, then coming to chronic arsenic toxicity menace, then synthetic biology relating to this seminar, emerging trends relevant to arsenic mitigation, more of synthetic biology, then what is the way forward. One slide I have not put it. It is Hippocrates, the father of modern medicine. He gave a statement, let food be thy medicine. Let your food be your medicine. So food and nutrition is so vital for our health. Because you have realized when the COVID pandemic was there was no vaccine. All the people didn't die because we have immunity. Our immune system is there, it protected us. Nonetheless, a lot of people died, we developed vaccine. But till that time, our immunity helped us. And what is that immunity? For that immunity, you need good diet. Antibodies, they are proteins. So we must have a high protein diet, and our immune system must be strong, so it gives us a protection. And when so that means nature has provided all these things to us. And beyond that, when it is beyond certain limits, it is necessary that we need medication, vaccination, and other things. But first line, proper nutrition, that is food and nutrition, that is what is very important. In that context, food security, I want to talk about bit of food security, then coming to the SDG goals and what is One Health, and because all these things are related to the nutrition, and then coming to arsenic problem and synthetic biology and all other aspects, linking it. The situation that exists when all people at all times have physical, social and economic access to sufficient, safe and nutritious food. So food security, it has 4 dimensions. What is that? Food availability is important, economic and physical access to food is there, should be there, food utilization and its stability over time. All these things are important. And in simple terms, you can say food security is a measure of the food availability and its accessibility. If for longer time chronic undernourishment, you are not getting proper nutrition, it is hunger, then there may be malnutrition. Malnutrition could be undernutrition, it could be overnutrition. Overnutrition means obesity is a big problem also present days. So if you look at the hunger, it may be protein hunger, calorie hunger. So protein calorie malnutrition occurs in one go many times. Then most important one is hidden hunger, that is micronutrient deficiency. We are really not, body is hungry but we are not feeling from outside, and it is a great problem. Hidden hunger is the most important one, because even though these minerals and vitamins, they are required in micro quantities, unless they are there, all metabolic activities are disturbed. So hidden hunger is most important. Now you must be seeing that people are getting micronutrient supplementation. Then if you look at the globe, what are our research priorities? They are all listed in the Sustainable Development Goals. Again we come to this. Number 1 is no poverty, second one is zero hunger, good quality education, number 4. So all these things, life below water, life on the land, and last one, goal 17, we should have partnership for the goals. Together we should be able to achieve all these things if we work in concert. Agriculture, fishery, animal husbandry, these are the sectors where we get the proteins. Proteins are, I was talking about protein hunger. It is so important because most of the most important roles and immunity, many things depends upon the proteins. So fish, shrimp, milk, egg, poultry, meat, they are the ones the sector is providing. And the challenges is production enhancement, food quality, biofortification, environmental pollution, food safety, food loss, that is post-service management, marketing channels, food hygiene and food fraud. So these are the different aspects. And coming to ecosystem. Ecosystem is vital. Ecosystem health, we should have healthy environment, then healthy animals. Then only we can ensure the healthy people. So it is a triad. And the present day approach is One Health. For One Health, we must ensure that there is environment is healthy, animals are healthy. Then only we can have healthy people. In this context, I come to this problem of arsenic in food chain, cause, effect, and mitigation. And we will be discussing our work on arsenicosis before going to synthetic biology. I think I am audible to the last line. Am I audible? The science I present here, all this work I did in Calcutta itself. I was in CIFRI, Barak, Central Inland Fisheries Research Institute, Barakpur. Whatever science I did, it was in Calcutta itself, and in Bengal itself. I am happy that I am presenting it here. We had a mega project with the same title, Arsenic in Food Chain, Cause, Effect and Mitigation. It was a World Bank funded project. 7 institutes are involved. CIFRI Barakpur was there for the fishery component. BCKV, then Uttar Banga Krishi Vishwavidyalaya, UBKV was there. IBRA Regional Center, West Bengal University Annual Fishery Science was there. And then one medical team, DNGM Research Foundation, DN Goa Majumdar Research Foundation, a famous gastroenterologist and arsenic task force member and all this. So we 7.5 years or more than that we worked in this project. Earlier we had the idea that through drinking water only we are getting the arsenic, which is a human carcinogen and a highly toxic substance. But these studies showed that through entire food chain, using that arsenic contaminated water, if you are growing rice or paddy or whatsoever other vegetables, through that also arsenic is accumulating. We are eating that food, we are getting arsenic. The animal feed or poultry feed or whatsoever we are making, they are getting also that arsenic is coming through that. Breastfed kids, they are also getting that contamination. So it is really a big issue. Then how to tackle it? So in this context, I am talking about identification of potential biomarkers of hepatotoxicity by plasma proteome analysis of arsenic exposed calves. Arsenic is a major toxic environment contaminant and it is a known human carcinogen. Prolonged intake of arsenic contaminated water and food the entire food chain I told, leads to chronic arsenic toxicity. So it is a major public health problem in many parts of the world. Arsenic, it is a multi-system disorder associated with cutaneous manifestation including pigmentary changes, hyperkeratosis, skin lesions, and increased incidence of lung, bladder, and skin cancer. Associated with also diabetes, predisposing to diabetes and cardiovascular problems. The permissible limit in drinking water for arsenic is 0.01 ppm for the world. However, for the national standard for India and Bangladesh, 50 microgram, that is 0.05 ppm. Because getting so low, it is difficult. We have to survive, we have to have water. That's why we are giving our level, international level and national standard, there is a difference you see. That's why it is a devastating disease in many parts of the Bengal, and after that in many parts of the country, in the Ganga, Brahmaputra, Meghna basin, arsenic is in the soft soil. Because of rampant lift irrigation, we have disturbed it for cropping, and it is there. And how can we get, because it is geogenic, it is already in the soil, how to get rid of it? So we have to find out ways. Maybe early diagnosis, maybe remediation, phytoremediation, bioremediation, microbial remediation, whatsoever. In the beginning, when we had the 2007-8, we entered into this field. We wanted to see what is happening, and I was in the fishery institute. We wanted to see the fishery component, because it is the lower vertebrates in which we can study and have ideas what could happen to the higher vertebrates like humans and animals. So we exposed Labio rohu, fingerlings to arsenic, and because it is a dominant species, and of course then the genome information was not available, but now the entire genome information of this species is available. So a lot of work can further be done. Those days it was not available. So when we exposed at specific concentration, at higher concentration, we got these lesions, skin lesions. I am showing you keratosis in humans, and we got similar lesions. Like in humans, it dark and white patches, you are getting skin lesions. And for the first time, we also reported cataract development. And in Taiwan and other countries, many countries, it has been reported in humans that in arsenic contaminated zones, the people they develop reports of early onset of cataract. At the age of 35, 36, they start developing. So it is working as a predisposing factor. So we could see that in fish also it is there. Of course, in the fish available in the market, we never see such things, or we may not find these things, because concentration dependent, and it is a chance factor. We published this in pathophysiology changes in rohu, fingerlings following arsenic exposure. That was the preliminary time we did. Following that, we wanted to see what further happening to the immune system. So in fish, this arsenic toxicity, we reported that it leads to generalized immune suppression through downregulation of interferon gamma, IL-4, IL-10, IL-12, these different cytokines. We studied TH1 cytokines, TH2 cytokines, T regulatory, and all these things. And so we reported, we could find that it is leading to a state of generalized immune suppression in case of fish. Similarly, HSP heat shock proteins, HSP gene expressions we studied in this Labio, and all these studied, it indicated that there is a state of arsenic is leading to a state of generalized immune suppression. So this was 2015, it was published Fish Cell Fish Immunology. So what is important, that diagnosis at early stage, if we are doing, then we can go for therapeutic approaches for this. So the biomarkers for early diagnosis is important, because when the signatures really come at later stages that it is too late. Maybe cancer and cancer development, other things may be there. So early diagnosis is very vital. So before proceeding, I think Madam had with Professor Santosh Kaur, you had some collaboration and yeah. So you was yeah. I had your name. So Professor Kaur is my PhD supervisor. He was in JNU. Yeah. So proteomic biomarkers for arsenicosis. What is a biomarker? Because I am going this bit slow and telling this thing because we have a lot of students and it is important to inform them, stimulate them, inspire them. They should know the things. That is what is important. Biomarker is a molecular or cellular or biochemical change in the host that can be accurately and repeatably measured and can aid in identifying and monitoring physiological and pathogenic responses to pharmacological interventions. Suppose you are treating for a disease, treating for cancer or for treating for anything, monitoring through those, using those molecules, you should be able to find out what are the changes happening. Biomarkers help not only in disease diagnosis, that means diagnostic biomarkers, but they are also helping in identifying potential treatments. What should be your approach? And tracking disease progression, regression and outcome after an intervention. Biomarkers for early detection and therapeutic interventions could lead to cure and better disease management. Early biomarker signal can be at the level of molecular level, subcellular or organelle level, cellular, tissue level, organ level or systemic, organism, population, community, ecosystem. So it depends at what different level it can be studied and which is appropriate for the situation or for the case. Now coming to a particular study, having told this cytokine profile analysis and all such things, we thought of what is important is going for a diagnostic marker. So in the same exposed fish, we did a plasma proteomics. We took the blood, exposed the fish, took the blood plasma, went for a proteomic investigation of that. And what we got? For the students, if you see, this is a 2D gel, this is a gel-based proteomic study. 2D gel, first dimension, proteins are separated on basis of charge. That is PI. Second dimension, proteins are separated on basis of molecular weight. And you see the side line, it is a single HD space gel. You find that maybe 20, 25, 30 proteins. But in this case, there will be hundreds of proteins will be there. So 2D gel is a powerful tool. Protein separation and resolution is very high. You can go for image analysis and there is specific, I won't go into details of this. So you will find that it is a powerful tool by which protein resolution is very, very high. If you are seeing by SDS page or one dimension gel, if you are getting 25 to 30 proteins, when you go for a 2D gel platform, you may get 900, 1000 proteins. Because your job is you are hunting for proteins which are responsible, which could be good biomarkers. So what we got? There are proteoforms. Proteoforms means there are multiple forms of the same protein also. Multiple forms. Because of post-translational modifications, other things, their charge, molecular weight is different. Okay? So out of those proteoforms, like you see, this is ApoA1, this is ApoA1, ApoA1, apolipoprotein, ApoA1, see molecular weight and PI, such a variable ones. And out of the several ApoA1s, I have showed you only the biomarkers. Out of these ApoA1s, not all are your diagnostic marker. We have to find out which particular one could be a good diagnostic marker. That is again a Herculean job. So in this study, we found out that apolipoprotein A1 alpha 2 macroglobulin, it is a short form A2ML, transferrin, and another protein WAP65. They were identified as potential biomarkers of hepatotoxicity and chronic liver disease. Why? Because it is causing these proteins, I am telling, these markers, our proteomic study generated, these proteins are present in the, we found it in blood plasma, because this is a plasma proteomics. Okay? So it is in the blood plasma, and these are livers, but these proteins are liver specific. They are synthesized in the liver, indicating that arsenicosis is causing liver damage, and these proteins are escaping to the plasma. Following this, we tried to, we did lot of studies on curcumin, that turmeric extract of that, which has nutraceutical or pharmaceutical against it against several diseases. It has been reported. What we did is finding that apoprotein gene expression in arsenic exposed to carb and the modulatory effect of curcumin. We have taken the controls, then we have taken different doses of this curcumin, dose 1, dose 2, dose 3. Similarly, C1 plus D1, C2 plus D2, like arsenic and different combinations we tried to see, and we found that APO A12B against specific proteoforms in arsenic exposed fish. So we found that some of them could be worked out further to find out a accurate biomarker, but again we are not able to tell whether this biomarker is for, it is arsenic specific, because there are other hepatotoxic substances. They could be leading to these biomarkers also, producing these biomarkers or enhancement of these biomarkers. So it is difficult at this stage to say whether they are arsenic specific or not. So in this direction further, we continue the study with liver transcriptomics. By next generation sequencing, we went for with curcumin. Experimental design is fish acclimatized in the laboratory for say 15 days. After that, this is the control. Then this is 15 ppm arsenic for 12 days exposure. Here it is fed curcumin supplemented feed for 7 days prior to exposure. When we are exposing to arsenic, before 7 days, we are feeding the fish curcumin through the feed we have formulated. So 7 days, so that it should be bioavailable to them. Under that condition, if they are getting exposed to arsenic, what is the genomic changes happening to that? So we did a liver transcriptomics for that. We collected after this exposure and this feeding, liver tissues collected from euthanized fish, and we analyzed the transcript profile. So there are different differential gene expressions, and how to, again, we found that several genes in such studies you will find that lot of upregulated genes, downregulated genes, and under whether it is in the control what is happening, in the arsenic exposure what is happening, when you have given arsenic and fed curcumin through the diet, what is happening, what are the changes. So these they can provide the information really what are the changes happening, and maybe we may get some biomarkers. Gene ontology. So what are the different activities, and so what we got is differentially expressed. I am focusing here only on apolipoproteins. There are several others. We found that earlier apolipoprotein A1, ApoA1 we are telling in the plasma proteomic investigation. Here we found ApoA1, 2, ApoA4, Apo41A, 1B, Apo42, then Apo14 KDA. So all these we are getting several APO variants. Now it is leading to a situation we have to find out actually. In one way they are looking many, but these are the shortlisted ones we have found out. We have to work in detail, so maybe some of them could be good biomarker, and which could be arsenic specific. We communicated this to a good journal, but they asked that we must have other hepatotoxic substance and as a control side by side to show that it is arsenic specific or it is in general. So that part we are doing in zebrafish, that part is. So in this context I would like to mention curcumin as this is another work we did. In the same exposed fish, lens protein profile we are studying. We studied the curcumin has protective effect on the eye lens against arsenic toxicity. What we found that we collected the lens from the same exposed fish, alpha, beta, gamma, different crystallines are there. Crystalline profile we studied. We found that there is suppression of the gene expression in case of arsenic exposure. When curcumin is fed through the diet, in those fish we analyzed, we found that there is one antioxidant substance, it is increasing, as well as this, it is leading to alleviation, feeding curcumin leading to alleviation of the gene expression. So it is coming to basal level. So it indicates that curcumin has a protective effect on the eye lens. So it was published in somewhere I think Biological Trace Element Research 2020. So having done these things, it is a responsibility also as scientists we keep on investigating things till we reach something, but people need, the public need to be helped in the sense. People, we are fish eating people, Eastern India. Rice and fish is the staple diet. Can we tell them that there are fishes who accumulate less amount of arsenic by nature, whether we understand their genetics or not, region or not. So in Basirhat area, in many ponds, we analyze these things, and this is a published work. So we reported that this is the order in which in different fish species available in that contaminated zone, arsenic concentration in fish. So this is the order. We published this in biology. We gave advisory that this is the way, like Labiorota is accumulating less, Labiocalbasu is accumulating less, Syrinus mrigala. So this is we analyzed their flesh and we found that. So people can go with this type of species which are accumulating less arsenic, even though it is in that locality. And investigating and understanding is another thing. But they can go for this so that they can get less toxicity out of the issue. So this is a arsenic bioaccumulation and identification of low arsenic accumulating food fishes for aquaculture in arsenic contaminated ponds and associated ecosystems. We published this work, and even though this is not deep investigation, we analyzed by ICPMS and found out these profiles. And this was a paper which was after it was published, Current Science again published it in a abstract form from their side, indicating that it is really helpful for the people, and so its significance should be known to people. So having discussed several things, I try to correlate the synthetic biology and its how the theme of the seminar, how it the trends in relevant to arsenic mitigation particularly. So what is synthetic biology? The rational design and construction of biological components, systems and organisms that do not exist in nature, or the redesign of existing biological systems for useful purposes. So it stands on systems biology, genetic engineering and molecular biology, but it adds a layer of abstraction and modularity akin to the engineering disciplines. So it is synthetic biology where bioinformatic tools, docking, several things you are doing and understanding a molecule. Maybe you are synthesizing a chimeric molecule, and which is helpful for a particular purpose. So this is what is synthetic biology. What are the applications, broad applications of synthetic biology across sectors, not only for arsenic? Healthcare and medicine, like vaccines, vaccine development, mRNA platforms for the COVID-19 and beyond, we have seen it. Diagnostics like synthetic biosensors for rapid disease detection. Therapeutic It is engineered probiotics, gene therapies, and synthetic antibodies. In agriculture and food systems, synthetic symbiosis, enhancing nitrogen fixation in non-leguminous crops. Biofortification, engineering crops with improved nutritional profiles. Alternative proteins, it could be cultured meat, it could be microbial proteins, plant-based analogs. Environmental sustainability, like for bioremediation, engineered microbes to degrade pollutants and plastics, which could be arsenic and many other fluoride and many other substances. Carbon capture, synthetic pathways for carbon dioxide fixation and conversion. Again, biosensors for monitoring environmental toxins and ecosystem health. In industrial biotechnology, for biofuels, microbial production of ethanol, butanol or biodiesel. Then coming to bioplastics, PHA and PLA from engineered microbes. Textiles and materials, biofabricated leather, silk and construction materials. In different areas, that synthetic biology has a broad application. So coming to the sustainable development goals, where it synthetic biology, what could be its contribution? Sustainable development goal 2, SDG 2, zero hunger. I am showing you that big chart. So it is engineered crops and microbial fertilizers, they can help. When you talk of good, say, SDG 3, good health and well-being, vaccines, diagnostics and therapeutics, we have seen during COVID mRNA vaccines. SDG 6, clean water, biofuels, energy efficient bioprocesses. In industry innovation, SDG 9, biofoundries, sustainable manufacturing. Then in climate action, so SDG 13, carbon capture, climate resilient crops. SDGs 14 and 15, life below water and life on land. Ecosystem monitoring and pollution control. These are the many areas where the synthetic biology can contribute. Then key trends in synthetic biology, especially for arsenic mitigation. See, under engineered microbial systems bioremediation, arsenic oxidation, trivalent arsenic is the most toxic one, converting it to pentavalent arsenic. Arsenic sequestration by metallocyanins, phytochelatins or engineered binding proteins, biomineralization into less toxic forms. This could all be possible through synthetic biology. Like in biosensors for arsenic detection. Synthetic biology enabled to development of whole cell biosensors using arsenic responsive promoters. So lot of biosensors are there. Still more suitable ones are being tried, but sensors are available. Cell-free biosensors for field deployable detection, paper-based synthetic gene circuits for low-cost diagnosis. CRISPR-based environmental monitoring, detection of arsenic-induced DNA damage, monitoring gene expression changes in sentinel organisms, and developing programmable biosensors. In sustainable aquaculture, engineering fish gut microbiomes for improved detoxification, so reduce the toxicity. Developing arsenic-resistant algal and microbial feed additives, creating biological filters for aquaculture systems. These are all possibilities, and to tackle this issue, maybe these are some of the approaches. Sustainable bioremediation approaches. Phytoremediation using arsenic accumulating plants. Like I was telling, low arsenic accumulating fish species, so that that is good for our health, we can eat them. Similarly, to remove phytoremediation using arsenic accumulating plants, so we can throw them, it can filter out. Microbial bioremediation using native or engineered strains. Mycoremediation using fungi capable of arsenic uptake. Nanobiotechnology for arsenic absorption and removal. So these approaches are environmentally friendly, cost effective and scalable. Arsenic induced transcriptomic dysregulation in calves. We have studied these things, leading to these leads to oxidative stress, altered expression of detoxification genes, disruption of metabolic pathways. So these are the several issues arsenic is causing in the calves. And I have already mentioned about our study about curcumin, antioxidant properties, anti-inflammatory, metal chelating, modulation of gene expression like we have seen in lens and in the liver. We have shown that it is helping. Other studies demonstrate that dietary curcumin restores arsenic-induced gene expression alterations, normalizes oxidative stress markers, enhances detoxification pathways, protect tissue integrity and improve survival and growth performance in calves. So it has a promise as a phytotherapeutic, and in cancer and many other disease people have studied the impact of curcumin also. Only thing that we have to find the best way. It is not highly soluble, then whether we should go for curcumin, we go for nano-curcumin, which form so that it is available, what is the cost effectiveness. So all these things are important. Our research identified candidate biomarkers for arsenic exposure and curcumin mediated recovery. However, again, when you take biomarkers, it is whether arsenic specific, that question remains. Arsenic specific or not, because against other like hepatic, viral hepatitis, there may be other chemicals. So they may be leading to such biomarker also, it has to be confirmed. Otherwise, stress proteins, antioxidant enzymes like SOD, CAT, GPX, detoxification genes, inflammatory markers, metabolic regulators. So these biomarkers can be integrated into synthetic biology based biosensor platforms for environmental monitoring. These are all possibilities. Arsenic bioremediation strategies, advances and opportunities. Microbial bioremediation. Engineered or naturally occurring microbes can be that can oxidize arsenic to less toxic arsenate, reduce arsenate for volatilization, sequester arsenic intracellularly. Then phytoremediation like plants like Pteris vittata, which hyperaccumulate arsenic. For soil cleanup and constructed wetlands, phytostabilization, for such things it can be used. Then synthetic biology enhanced bioremediation, it could be metal binding protein expression, this transporter efficiency, stress tolerance, biofilm formation for arsenic capture. These are ways which can be tried and people are working on it. Then policy, governance and ethical considerations. Synthetic biology in India, it is still in its early stages with governance frameworks evolving to address biosafety, biosecurity and ethical concerns, because these are big issues, ethics and clearances, and it has whether we should go for or not, like that several policy governance issues are there. Key governance challenges: regulatory oversight for engineered organisms, environmental release policies, public acceptance and risk communication, intellectual property and access issues. We cannot straight away release something, it is so much of regulatory issues are there. So how it could be possible? So some are suggested that our country requires we should have a national synthetic biology mission, clear guidelines for environmental applications, public-private partnership, capacity building in university and research institutes, so that in a systematic way we can have it, avail it. Then a roadmap for India, integrating synthetic biology, aquaculture, sustainable development, strengthening the research and innovation, for that establishing national centers for synthetic biology and environmental biotechnology, promoting interdisciplinary research like aquaculture, toxicology and genomics, supporting field trials for biosensors and bioremediation. These are not easy tasks, they have to be systematically carried out. Community level interventions like deploying low cost biosensors for rural water monitoring. Like in arsenic contaminated zones, you must have seen that several places the wells and tube wells, they have been marked red, not to be used. It is so that monitoring is so important. Integrate arsenic safe aquaculture practices. Industry and startup ecosystem. Encourage biotech startup developing biosensors and engineered microbes. promoting sustainable aquaculture feed additives like curcumin based nutraceuticals, for example. Then support green technologies for water purification. And coming to international collaborations, collaborate with global synthetic biology hubs, participate in international arsenic mitigation programs, sharing best practices in South Asian countries where these issues are more burning. So having discussed all these things, so what is the way forward? Some of the suggestions. We know that chronic arsenic toxicity remains a formidable challenge for India and many other countries. However, the convergence of synthetic biology, environmental biotechnology, sustainable development frameworks and nutraceutical interventions offers unprecedented opportunities. They have to be tapped. It is easy said and done. We say that, but combining these things efficiently, handling these things with the policy framework, everything at place and implementing. Some of our studies I have mentioned that curcumin mediated restoration, demonstrating potential of natural compounds in mitigating toxicant induced molecular damage, robust biomolecular sensors, model for integrating omics based insights with synthetic biology tools. So if we are able to leverage advances in biosensing, bioremediation, sustainable aquaculture, maybe we can scale up community friendly and environmentally possible solutions to tackle the arsenicosis, which is a major environmental challenge. So synthetic biology, arsenic is a big problem. We are working on it, means globally work is going on, and with this particular state and other adjoining states. Earlier it was a problem, now it is reported in Northeast it is a problem, Bihar it is a problem, UP, entire Ganga, Brahmaputra, Meghna basin. So more and more areas it has been detected. How to tackle this menace, that is a challenge. However, synthetic biology can be integrated into such studies and maybe that can help us. So the work I presented from my side, it was Sifri Barakpur where I carried out most of the work. It is DBT PDA program, World Bank funded NIP, NAS National Agricultural Science Fund, ICAR outreach activity, FSSI, all they are funded by this, and these are some of the people at different point of times we work together. Soma Bhattacharya, my DBT PDF, doing most of the proteomics work at early phase. He is in Vidyasagar University. Arvind is working in ICR. Sudeshna Banerjee, she is in Hyderabad in Sigma. Satabdi Ganguly in Sifri Barakpur. Tandrima Mitra is in KIT University. Ravi Prakash, I think Zoology Department, Calcutta University. He did his MSc work under me, technical staff. Dr. B.K. Das, Director Sifri. Pranay Parida, Rohan, these people are from Sifri Barakpur, scientist and it is director. And my partner, Professor Susmita Mohanty. My wife and my partner, whatever proteomics study I do, she does the transcriptomic study, because many projects we work together, because doing all work is difficult. So whatever proteomics part I do, at least validation we get it there, so it helps. So it is a long way. Still we found that we are working on it to establish a clear-cut biomarker for arsenicosis. Thanks for your attention and thanks for inviting me.
Thank you very much, Professor Mohanty, for such a nice and illuminating talk. Now we know more about the problem of problem with arsenic and how to deal with it. So now this is the time for questions. So students, if you have any questions, please raise your hand. Yeah. So yeah. Yes.
Yeah. We are all students. Yeah.
Yeah. Please, Professor N.K. sir.
Thank you, sir, for your lucid but highly informative and illuminative lecture. I've got two simple questions. You showed that you people found 10 or 12 species of fish along with some gastropod and crustacea accumulating arsenic in muscle.
Yeah.
Is the accumulation level same or similar in all these fish and other invertebrate species? First question. And second thing is that do you think that we should avoid taking some of those species in order to avoid biological magnification so that we do not suffer from biological accumulation of arsenic in our body through the food chain?
Okay. So for your first question, what I showed is a chart. From this Basirhat 1, 2, 3, all those areas, we collected fish from the arsenic accumulated ponds. And we analyzed the accumulated arsenic level in their tissues, plus whatever fish we are eating. And because overnight there is no solution for these issues, we wanted to advise the farmers that you cultivate these species more, so they are accumulating less. So we analyzed in their tissues, and we have given the gradation top to bottom.
No, do all these species accumulate similar levels of
No.
Which species accumulates most?
I have shown that Labio rohita, because they, on the top. So actually, some of the species we are basically carp eating, Katla, Rohu, Mrigal. So amongst that, Labio rohita and Mrigala is still accumulating less. So towards the bottom which are there, they are accumulating less. So we are advising that, these are the ones preferred in these arsenic contaminated zones, so that they are accumulating less. It is another thing whether you understand the genetics behind it or reason behind it, keep it aside, but people can eat and go for culturing those species, so arsenic uptake for human is less. So they are not accumulating to the same quantity, no. So those who are accumulating less, we are advising for them.
No, should we avoid taking those species that are accumulating most?
We cannot
avoid biomagnification.
So we have a society, socio-economic status is very important. People who can afford, at least they are getting protein. This is the problem. People are eating fish. We eat fish and rice. Because by eating fish, we are getting omega-3. We are getting like hills and other things. We are getting omega-3. Small fish, we are getting microelements, vitamins and minerals. Any fish you eat, you are getting protein from it. So we cannot say don't eat. But if arsenic accumulation is there, actually it is a toxic substance.
At least local people should avoid it.
Whether if they can afford for alternatives, they can go for it. Whether they can afford for alternatives, whether it is available. Several things, because culinary preparation, other things, many things decide.
Thank you, sir.
For second question, what was the second one?
Whether people should avoid taking those species that are accumulating maximum amount of arsenic.
Again, who is going to advise? We have given a generalized study in one area.
But you belong to ICAR and fishery.
That's why we are still for diagnostic. These are it is a complex issue. Several issues are there. Unless we provide people alternative, suppose people remaining in those areas, if there is no safe drinking water, they have to anyway drink water to survive. How many days you survive, 6 months or 5 years, it is another thing. But you need water. If you are not providing safe water.
In South 24 Parganas, arsenic problem was very severe a few years back.
It is still there. It is still there. And
thank you, sir. And I just mentioned one coincidence. You found that curcumin is suppressing cataract development and all these things. And coincidentally, we have today amongst us Professor Tania Das. She happens to be an expert on anti-tumor or tumor suppressive activity of curcumin.
It's good. Curcumin. In
fact, so next question from Tania Das.
Excellent talk, actually, so much data. That is my question. Query was in relation to his one, and now I understand that what you have done. So it is giving a signal to the general people that what type of fish they can eat to get lesser, exposure to arsenic. But if you think about, negating taking fish or whatever, maybe if you now start studying on vegetables, most of the vegetables also, they will contain arsenic, and also where you will stop. So we cannot, stop taking fish or vegetable, etc. But on the basis of this knowledge, we can now study why these fishes are not accumulating arsenic. So if we can understand that,
Yeah.
I understand that, then one day the antidote will be
Yes.
coming. So
Yes.
that is very nice part of
Yeah,
your study.
yeah.
Thank
That
you.
is the reason.
Yeah. So anyone has any more question? So I have a small question. So you showed that the arsenic is accumulating differently for the different fishes in the controlled environment. In the lab, you tested that within the controlled environment. So, but in river, the arsenic concentration may be different. So,
Okay, I got it.
yeah. So
Now,
depending on the
we did not do it in control environment. What we did, arsenic contaminated zones, Posirhat,
Okay.
those areas, arsenic contaminated zones. In those areas, we made several visits, collected fish from that locality.
Okay.
Because we wanted to give a easy solution, at least farmers should get a solution. That we accumulate whatever fishes available with different ponds over seasons. We collected those fish, analyzed the arsenic level in them. So from that field-based study, we suggest that okay, this is the level of accumulation. If there are good species which are accumulating less amount of arsenic, so they can be preferred.
So,
And
so
farmers can go
it's
for
based
first.
on the actual data from the actual
Yeah.
the fishes in the
Yes,
river. So
not
that's
laboratory.
great.
Yeah.
Yeah, that's good. Yeah. Thank you very,
Okay,
very
before I
much.
end,
sir.
I will say that I heard about madam because Professor Kaur sometimes he visited my lab at Barakpur also. He told that he is going to meet you and all this. So I said I am not interested. He was asking whether you have interacted. I said no. But I heard that madam is coming because they have done good work on curcumin, nano-curcumin and in cancer and other diseases. So it's a great pleasure meeting you and you are here with me. Okay. Thank you.
Thank you very much, sir.
My pleasure. Okay. Nowadays he is in Bhubaneswar. So
I met Abdul last week.
okay.
Yeah,
Okay.
global immunology conference.
Of course I was not there that time, I couldn't attend, but we keep on calling and meeting. That's a pleasure.
May I now have the honor to felicitate Professor Tania Das, ICMR Emeritus Professor, Division of Molecular Medicine, Bose Institute. Ma'am, we are truly privileged to have you with us. Please come to the stage. I would like to request our research scholar to felicitate ma'am. A big round of applause, please. Thank you, ma'am.
I also request our research scholars to felicitate Dr. Professor Gauri Shankar Shah, Emeritus Professor, Division of Molecular Medicine, Bose Institute, at offstage.
Now this session will be chaired by Dr. Shobhusha Chipal, Associate Professor, Department of Pure and Applied Sciences, Midnapore City College. But before that, I think DN sir had some requests, sir, please.
For our young generation, very few informations about the Tania. You are very much well known to our great teacher and fellow faculties of Professor Nirmal Sarkar, Professor Devajyoti Das. You all know And daughter of the Professor Devajyoti Das is Tania Das. This is very much important information of this. And obviously, Professor Nirmal Ghosh is the co-colleagues of Presidency College, both of you. And last not the least, may I request Tania, before your talk, please, before your talk, she is not a hardcore scientist. Very nice also, not in science within the world, four wall, but apart from the society or the outside the four wall, she is very nice singer, and particularly for the Rabindra Sangeet. Before that, to boosting our students and also the faculties to sing a song, please. Hey, Amitor, hey, bondhu, আমি বলছি। Yes, sir.
গানটা এটা দিয়ে করে নি।
And she has made several recordings. She has many cassette tapes and CDs to her credit.
না চেয়ারটা দিতে। ও ওনাকে এটা নিয়ে নাও প্লিজ। এরম কিন্তু কথা ছিল না। কেউ কথা রাখেনি, এই কথাটা আমি আজ এসে আরো বেশি রিয়ালাইজ করলাম। আর কেউ চান্স নেই, আর আমার তো 66 বছর হয়ে গেছে, কেউ কথা রাখবেও না। এবার হচ্ছে যে আমার সবসময় অলওয়েজ আই ফিল দ্যাট হোয়েন আই কাম টু দিস সিটি কলেজ হিয়ার আই হ্যাভ এ ভেরি মিক্সড ফিলিং। ইট ইজ নাওএডেজ ইট হ্যাজ বিকাম লাইক হোম কামিং। আমার মনে হয় নিজের মানুষদের কাছেই আসছি, কেন আমি দায়িত্ব নিয়ে বলছি যে এটা শুনতে একটা সিটি কলেজ, আমাদের বেশিরভাগ ইউনিভার্সিটিও এতটা বোধহয় উন্নত নয়। আমি এটা মনে করি সারা পৃথিবীতে সর্বক্ষণ আমরা ঘুরে বেড়াই বিভিন্ন ইউনিভার্সিটিতে, এইখানে যে ধরনের ফেসিলিটি, যে ধরনের রিসার্চের এনভায়রনমেন্ট এবং এনকারেজমেন্ট, যেটা সায়েন্সের রিসার্চের জন্য বিশেষত, অন্য সাবজেক্টগুলোতে আমি জানি না, বলতে পারবো না, কিন্তু সেটার জন্য এনকারেজমেন্ট ফ্রম দ্য পিরামিডের টিপ থেকে সেটা ভীষণভাবে জরুরি। সেটা কিন্তু আমার এখানে এসে মনে হয়েছে, আমি এখানের অন্যান্য কমিটিতেও যে রয়েছি, অনেক ইভ্যালুয়েশন অনেক কিছুতে ডাকে, সবসময় ডাকেন এরা, আসি এবং মিট করছি স্টুডেন্টদেরও ফ্যাকাল্টিদেরও, একটা এনকারেজমেন্টের আবহাওয়া এখানে রয়েছে। আমি সত্যি বলছি, যারা এখানে পড়ছো তারা সত্যি খুব লাকি যে এরম একটা জায়গায় তোমরা পড়তে পারছো, এরকম মেন্টরশিপে পড়তে পারছো। তো এরকম একটা বড় ঘটনা কিন্তু কারুর একার, নিশ্চয়ই একজন দুজন থাকেন যারা মাথায় থাকেন, কিন্তু সকলের প্রচেষ্টা ছাড়া একটা ভালো জিনিস কখনো ভালো হয়ে উঠতে পারে না। তাহলে সবসময় আমরা একসঙ্গে চলবো, একসঙ্গে বলবো, সব কাজ একসঙ্গে করব, তাহলেই কিন্তু আমার মনে হয় সেখানে প্রকৃত ঈশ্বর বা মঙ্গলময় তার অধিষ্ঠান হয়। সেই চারটে লাইন দিলীপ আমার, হি ইজ এ ভেরি ক্লোজ ফ্রেন্ড অফ মাইন্ড বিকজ হি ইজ এ ভেরি ক্লোজ টু মাই ড্যাড অলসো। আই এম ভেরি প্রাউড নট ফর বিং মাই সেলফ বাট বিকজ আই এম মাই ড্যাডস ডটার। I am very much proud of that. So for that 4 lines I will sing. This type of request I have never heard in my whole scientific career, that before seminar someone has been requested to sing. So the song, 2 lines from, actually 1 line from a stotra of Rabindranath Tagore himself, and then 2 lines from the Rabindra Sangeets, that if we go together then God will be there always, everything will be good. So that is the meaning of these total thing. Sangachadvang sangvadadvang sangvo manasi janatam anunduluke মঙ্গলালোকে বিরাজো সত্য সুন্দর মহিমা তব উদ্ভাসিত মহাগগন মাঝে বিশ্বজগৎ মণিভূষণ বেষ্টিত চরণে Sangachadvang Sangvadadvang Sangvomana Sijanatam Anunduluke Mongulaluke Biraju Shottu Sundaru.
Beautiful song, madam. Now I have the privilege to say a little bit about Professor Tanya Das. Professor Tanya Das is an ICMR emeritus scientist, Division of Molecular Medicine of Post Institute, Calcutta. She has completed her MSc in Biochemistry and PhD from University of Calcutta. She has completed her postdoctoral work from Cleveland Clinic, USA. Her research area interest is to develop targeted therapy of cancer. She has more than 140 research publications in different reputed international and national journals with age factor of 39, citation of more than 6,000 and I-10 index of 76. Really very good.
Thank you.
She has supervised 33 research scholars.
35 now.
Okay, 35 research scholars for PhD work. Professor Tanya Das received different awards from different national and international bodies, like Fellow of National Academy of Science, India, Fellow of West Bengal Academy of Science and Technology, Fellow of Physiological Society of India, National Young Woman Bioscientist Award, DBT, Raj Krishna Datta Memorial Award, Indian Science Congress Association, P.B. Ramarao Oration Award of Society of Biological Chemists, India, P.B. Sen Memorial Award, Physiological Society of India, Sananda Nihar Samman from Anandabajar Patrika Group of Publication, Rupa Chakraborty Memorial Award from Bethune College, Bongo Nari Samman, Ma Durga Samman, Shruti Natra Samman, Ritu Samman, etc. She is Secretary, Society of Biological Chemists, India, Calcutta chapter, Vice President, Indian Society of Angiogenesis, member, Indian Immunology Society, Indian Science Congress, Indian Association of Cancer Research, Society of Biological Chemists, Indian Society for Translation Research, and DNA Society of India. She has 3 patents related to her research work. I request Professor Tanya to start her presentation.
Thank you for your kind introduction, and I'm really indebted to Dr. Pradeep Ghosh, the founder director of this very good college, as I already told you guys, and my very close friend Dilip, and also to Dr. Sravani Pradhan for inviting me today, giving the chance of talking, sharing my thoughts about cancer with my students. So whenever I give lectures or discuss my about my work with the students, I always feel very much encouraged, because কেননা তোমরাই এখন সামনের প্রজন্ম যারা এই কাজগুলোকে এগিয়ে নিয়ে যাবে। আমাদের তো এখন আস্তে আস্তে বয়স হয়ে যাচ্ছে, তোমাদের সামনে কিছু বলতে আমার স্পেশালি ভীষণ ভালো লাগে। তো I get chance of discussing with my work with you guys when this type of situation arises. So I am really very much indebted to the organizers and the founder director, and of course Dilip, who is my very close friend. So the today's topic is cancer stem cells, a little bit new term, I understand. The major hurdles at the forefront of cancer therapies. I know that winter is going away now slowly, and spring is approaching. So your mind, and at the time is 12:15. So everyone is feeling hungry and not at all interested to know what are cancer stem cell and what are the hurdles. Very boring, I understand. It is boring to me also. So please stop me whenever you have any question or you feel bored, I will stop, no problem. We will just discuss. I will not give any seminar, so I promise. So to make our life a little bit interesting, we want to know what is cancer. Because you understand that though it is an old disease, you will see here how old it is, but till today it is the most fatal one, you all know that. So actually cancer is a very old disease that even existed even in prehistoric era. See here the first evidence of brain tumor found in dinosaur. You know, so many, 72 million years ago, these dinosaur, Tyrannosaurus rex, they used to reign in this world. And so first evidence of human cancer was found in mummies, Egyptian mummies. And you will be very interested to know that Lord Buddha had treated the malignant tumor, cancer, of Prince Ajatashatru. Everything is actually documented. So nothing, I'm just not the... I have not made this story, all are documented, and this is mention of cancer, we find it in Rig Veda even. So we understand that it is a very old disease, and also breast cancer, about which I will be talking today a little bit. So that is also an ancient disease. We also get the mention of breast cancer in the papyrus. You know the papyrus, the যেখানের মধ্যে তখনকার দিনে ইজিপশিয়ানরা লিখতো। এবার ওয়ার্ল্ডস ওল্ডেস্ট কেস অফ ব্রেস্ট ক্যান্সার যেটা ছিল মেটাস্ট্যাটিক। ইউ নো হোয়াট ইজ মেটাস্ট্যাসিস? ফ্রম দ্য প্রাইমারি place it goes to secondary place. So breast cancer metastasized to brain, skeletal bone, and here also in the bones. That was found in Egypt very recently. And as I told you, Rig Veda 1 verses 162 and 163, we get the mention of breast cancer itself, not only total cancer. And there were treatments too. Compounds of barley, pig's ear, very interesting, etc. were suggested as treatment for cancer. And also nowadays we talk about, just we have heard the use of curcumin. He was telling that madam works on curcumin. So this is a natural product. Yes, I also work on curcumin, but today I am not going to discuss that. And that time also, so many years ago, they used to use plant product castor oil for breast cancer treatment. And so now we understand the concept of cancer was there. So even 72 million years ago, cancer was there. When no these junk foods were there, no pollution was there, but cancer was there. So all our concept about cancer, you understand very well, those were all not right. Am I right? আমরা যা ভাবি ক্যান্সারকে যে সভ্যতার সংকট, সেটা কিন্তু পুরো ঠিক নয়। তাহলে সভ্যতার কি ছিল? 72 মিলিয়ন বছর এত বছর আগে ডাইনোসরদের ক্যান্সার ছিল, এবং ইজিপ্টে যখন এই ইজিপশিয়ানদের যে মামিতে পাওয়া যাচ্ছে, তখন এই ধরনের ফাস্টফুড কোথায় ছিল? পলিউশন, এনভায়রনমেন্টাল পলিউশন কোথায় ছিল? গাড়ি ঘোড়া তো অলমোস্ট ছিলই না। কিন্তু সেখানে ব্রেস্ট, আমরা এখন আবার নতুন কনসেপ্ট হচ্ছে সেজন্য। সো হোয়াট ইজ দ্য নিউ কনসেপ্ট? New concept or scientific concept is, I am talking about breast cancer only. So breast cancer is a multiple signal modulation. You know, always everything what is going on inside our body, there is due to one cell is sending some signal to the other cell. That way they decide what type of work they have to do, and then they also get signal from outside. So pollutants can be signal, nutrients can be signal. So there is a normal signaling procedure in our body, তাই তো? নর্মাল একটা সিগন্যালিং কিছু চলছে বডির মধ্যে, তাই জন্য সেলেরা পরস্পর ঠিকঠাক কাজ করতে পারছে। আমি রাইট? বুঝতে না পারলেই বলবে কিন্তু, তাই তো? কিন্তু সেই সিগন্যালটা যখন, when there is multiple signaling disorder, multiple, mind it, cancer is not a single disease, it is due to multiple signaling disorder. The signals that keep our body in homeostasis, everything is normal, that gets changed. So when there is multiple signal disorder, that disease is actually, that involves unabated cell growth. Oh my God, movie is not doing. Okay, so I cannot show the movie. Movie is not working here. Oh, already it is played? Oh, okay, so you have, yeah, that's good. So you can see that here the cancer cells are, these are the, because they are multiplying, so you can see they are this way, you can see the movie, and they are also migrating, so they are occupying this space, and because of their higher number also, you can see that they are actually filling up this space. So it is uncontrolled growth. That is one property of cancer. This is the new I think this is also, this concept is also now almost 100 years old. And then they have, can you show this one once more? Yeah. See, these are the endothelial cells. Why I am showing? Because when there is unabated growth, so much cell division, they need food. But they don't have any ma, baba, no mommy, daddy. So through blood vessels, the cells get food. Okay. So for making the blood vessels, new blood vessels, actually they require endothelial cell network formation. So I have seen the endothelial cell network formation. Then smooth muscles come, many other cells come and then form the blood vessel. So when the blood vessel is formed, then blood flows through that and then take the nutrients to the cancer cells so that they can divide quickly. They have more calorie, so they can divide quickly. So this is another property of cancer cells, that they should have higher frequency of blood vessel formation. So for that the marker is endothelial cell network formation. So I have shown that. And can we go here? See, this is how the cells are migrating, breast cancer cells are migrating. It is our laboratory picture. So see here, the breast cancer cell, it is migrating. So this way they, from one primary site, they go to 2-3 secondary sites, and that is called metastasis. Now for migrating, they have to make this type of, oder to hat pa kichu nei, this type of processes. See, this is a quick movie. Yes. Kora? Hocche na? Okay. You can see the processes. So these are the processes they make, and then they migrate, as we have shown here. Then they have also drug resistance. See here, the drug has entered in the normal cell side by side, but here drug has not entered inside the cells, and so the cells are not dying. So these are the new concepts of what cancer is, and now there are modern different types of modalities of treating cancers, okay. But despite day by day increasing diagnosis and treatment procedures of breast cancer, surgery, radiation therapy, hormone therapy, chemotherapy, number one, then nowadays immunotherapy. I will talk about immunotherapy a little bit. So we all know, and also because sometimes they give too much side effects, we also work on phytochemical therapy. Curcumin is one of those. Okay, so many, in our laboratory also, my students worked on this type of different phytochemicals, curcumin also, and we have many publication. Actually, I was very happy. You can see my face. Actually, we thought that we have reached at the happy end of the whole story, like the Cinderella. So see how happy I am. Okay, you like this face, I understand, but that was not the reality. If I can think that it is a lion, can it be a lion? No. Like here you can see I am sitting with a tiger. But do you think that I am really sitting with a tiger? No, yes, I am sitting with tiger, but see the reality, there is a chain in such a way that the tiger cannot move. This is in Bangkok, cannot move its head. So you can safely sit behind. So always whatever we feel or think, those are not the reality. So then what is the reality? Actually, change in the death rates of different diseases, though those come down, like different types of diseases, see here, but cancer, you can see that it is not coming down. The graph is increasing and increasing, and breast cancer, see, it is a little bit old data, but till today it has not been changed that much, that though heart disease, cerebrovascular disease, pneumonia, influenza, all the infectious diseases, they are all coming down. See the graph, all are lower than the 1950 versus 2012, 2020 or 2020 also there is a survey, but almost same. And then breast cancer, you can see there is almost no change. No change. 1950 versus 2012, and I guarantee you 2020 up to 20 it has been done, not much change. 1.5 million cases of breast cancer getting diagnosed each year. Among those, 0.5 million deaths in women, with 7% cases under 40 years of age. Previously, we used to know that with menopause, the incidence of breast cancer increases, but nowadays it has come down. Even below the age of 40 years, breast cancer can happen. And also very recently, a girl, Tracy Turner, her age is 8 years only. So she had breast cancer. You can understand how grave this disease is. So though it is very old disease, so many modalities of treatment, but till today this is the, we are roaming in a circular way, this way we are just, just roaming, but not that much, what should I say, that not that much, positive things we are getting. So we ask the question, why all efforts are failing? What are the major hurdles at the forefront of the cancer therapies? That was our question. Actually, over the last 100 years, many studies have performed to determine the origin of cancer, where from cancer is coming. First, we thought that the scientists thought it is genetic. You all know. Yes, some cancers are genetic. And then the concept of epigenetic mutation, epigenetics have, that concepts have come. Okay, genetics and then epigenetic. ডিএনএ থেকে হয়ে গেল, তার পরবর্তীকালে যে চেঞ্জগুলো হতে থাকলো সেগুলো মিথিলেশন হয়, অনেক রকমের চেঞ্জ হয়। এপিজেনেটিক এই ধরনের চেঞ্জের জন্য ক্যান্সার হয় বলা হলো। প্রথমে বলা হলো শুধু জেনেটিক, এরকমভাবে চলছে। এখন ইন দ্য লাস্ট 25 or 30 years a new possible epistemological concept হয়েছে। A very paradigm shift has happened in the field of cancer research. এখন আমরা বলছি ক্যান্সার maybe it is a stem cell disease. So the new term cancer stem cell has come up. So stem cell, you all are from biological science background, I understand. If not, yes. So you know that all the cells of our body, not only all the cells, living cells, they of mammals or organisms, they come from the stem cell. For human being, you will understand easily that when the ovum and sperm fertilize, then first the stem cells are formed, and then finally from the stem cells all the cells of our body, they are formed. And in the cancer stem cell area, cancer stem cell theory says that these cells have, cancer stem cells have, number one, stem cell properties. I will come to that properties. Number two, they have cancer property. Okay, so they are termed cancer stem cells, and now we think that cancer never happens from cancer cells. খুব নতুন কথা বলছি, ক্যান্সার সেলের গুলোর থেকে জন্য ক্যান্সার তৈরি হয় না। Then your question will come to your mind automatically, then why we are trying to kill cancer cells? Yes, there lies the problem. We always try to kill cancer cells, but cancer never happens from cancer cells. So we are trying to kill those cells which are not the root of cancer, but we never try to kill the cells which are the actually origin of cancer. They are, because all the cells come from stem cells, cancer cells also come from cancer stem cells. So the origin is cancer stem cell. If we kill cancer cells, cancer stem cells are there, sitting there. I will show you, it is interesting. So usually in a tumor mass, cancer stem cells sit in the core of the tumor. তাহলে বাইরে থেকে যখন ট্রিটমেন্ট করা হচ্ছে বেশিরভাগ ড্রাগ রিচই করে না ওদেরকে। Okay, and then these cells, these are the differentiated cancer cells which are the major cell type of a tumor. More than 95% are cancer cells, not stem cells. But these 2 to 5% of cancer stem cells, they are non-differentiated cells. ওরা differentiate করেনি, তারা cancer stem cell. Differentiate করলেই ওরা cancer cell হয়ে যাবে। Okay, so they are non-differentiating, and they can reproduce themselves as by division. One cancer stem cell to
divides to 2 cancer stem cells, like all the cells of our body. But unlike all the cells, they can also make cancer cells. So, one cancer stem cell can make 2 cancer cells also, okay, unlike other cells of our body, only stem cell can do that. So, that is another property of cancer stem cell. Another thing is they are highly chemo and radio resistant. So, if they are completely chemo, radio, all therapy resistant, I will show you how they are immunotherapy also resistant. So, then they will sit there in that tissue, and then when all the treatments are done, all the cancer cells died because cancer stem cells, cancer cells are sensitive to chemotherapy, radiotherapy, immunotherapy all. So, they die. Doctors think that cancer has regressed. তারা বলবে ঠিক আছে এবার ক্যান্সার কমে গেছে, কেননা যে টেকনোলজি দিয়ে ধরা হয় সেলগুলোকে, এরা যেহেতু সংখ্যায় ভীষণ কম, ভেরি লেস, লিটল ইন নাম্বার, তাহলে সেই টেকনোলজিতে কিন্তু এই 4টে 6টা পড়ে থাকলে দেখা যাবে না, বোঝা যাবে না, কিন্তু তারাই কিন্তু বাবা মা ক্যান্সার সেলদের। When they see that no treatment is going on, they come back and then reproduce all the cancer cells because they are stem cells. Okay, so that is the problem. So previously it was thought that all cells of a tumor, why I am telling all cells, because a tumor mass is not a symmetrical cells, different types of cells are there, like breast cancer, lural cells are different types of cells are there. So each and every cell of a tumor tissue can produce cancer. That was the old concept, but nowadays we think that only cancer stem cells can very quickly produce tumor. Cancer cells can also do, because cancer cell and cancer stem cell, they are in plasticity, they are interconvertible also. I will not go into that complicated detail, but they can produce cancer, but they are much more slow. First they produce cancer stem cell, then they produce cancer, but cancer stem cells can directly produce all the cancer cells. So, characteristics of cancer stem cell, so now we understand that the cancer stem cells are the culprits, but always all our treatment modalities, those target কাকে টার্গেট করে, ক্যান্সার সেলদের, তাহলে ক্যান্সার সেলদের টার্গেট করছে, ক্যান্সার স্টেম সেল তো বসে রয়েছে। So to kill, so attack some enemy, you have to know what are the properties of an enemy. একজন তোমার এনিমি কিনা তোমাকে তাকে বুঝতে হবে তার প্রপার্টিটা কি, সে কোন দেশের এনিমি, কার কাছে বন্দুক আছে কি নেই, না কি গরিলা কি গেরিলা অ্যাটাক করছে, না কি করছে, সাবমেরিন নিয়ে যাচ্ছে, কি করছে জানতে হবে তো। তাহলে আমরা উই ট্রাই টু ফাইন্ড আউট দ্য প্রপার্টিজ অফ দ্য ক্যান্সার স্টেম সেল। সি এইটা হচ্ছে ক্যান্সার সেলগুলো, সি দেয়ার নেচার, হাউ দে লুক, দ্য ক্যান্সার সেল, ব্রেস্ট ক্যান্সার সেলস ইন আওয়ার ল্যাবরেটরি MCF7, and when they are put in the serum free medium, the cancer cells die because they need serum as food. Okay, serum free medium e dile shudhu jara beche thake tara cancer stem cell, because they are highly resistant, je kono condition e beche thakte pare, বুঝতে পেরেছো? তাহলে সেরাম ফ্রি মিডিয়ামে রেখে এবং তারা এরকম ধরনের স্ফিয়ার তৈরি করে, এরকম গোল গোল, তাদেরকে আমরা ব্রেস্ট ক্যান্সার থেকে বলে ম্যামোস্ফিয়ার বলছি, ঠিক আছে? এদের নানা রকমের মার্কার আছে, তোমরা সবাই এখন রিসার্চ করছো, আমি ফ্যাকাল্টিদের কথা বলছি না, ছোটদের কথা বলছি, তোমরা জানো যে ডিফারেন্ট মার্কার থাকে সেলের বাইরে, এদেরও দেখো স্ফিয়ারের বাইরে যদি স্ফিয়ার তৈরি করে, এটা ক্যান্সার স্টেম সেলের নেচার, তারা এই ধরনের CD4 এটা মানে সরে গেছে, CD44 বেশি হবে, দিজ আর ক্যান্সার সেলস, দেয়ার CD44 ইজ লেসার দেন CD24, এটা এই 24টা আবার ক্যান্সার সেলে বেশি, স্টেম সেলে কম, এরা মার্কারের তফাৎ আছে। There are differences in cell surface markers so far as cancer cells and stem cells are concerned. Okay, and then these are the different CK19. This is one marker which is expressed in the cancer stem cells, but not that much in non-stem cancer cells, just cancer cells. So, and then the drug resistance pumps, as I already told you, these are very high in the cancer stem cells, mammospheres, but not in the normal cancer cells. These are the drug resistance pump. Antioxidant that factors are also higher in the cancer stem cells. Why we are looking for the markers? Because if you want to study something in laboratory, you have to know what are the markers. Otherwise, from the cells, they all look same. How do you know what are the cancer stem cells and what are the cancer cells? And see here, when I injected in the nude mice means they have no immunity. quote unquote no immunity, not actually zero, but they have very, very less immunity. So when we injected cancer cells in a nude mouse, see here after up to 14th day there is no tumor, but when we directly injected the cancer stem cells here, see here within 14th day there is a big tumor. So that thereby proving our theory that the cancer stem cells very quickly form the tumor. And they have also again intercellular markers, Nanox, SOX2, OX4, MRP1, you do not have to look at those. And then see here, when just look at the differences, these are the cancer cells. This is again the endothelial cell network formation study experiment. See here, these are the non-stem cancer cells. The time by which they are not actually cancer stem supernatant we put on the top of the endothelial cell culture. So the time when we took reading, see how nice endothelial cell network has been formed when we put cancer stem cell supernatant on the top of the endothelial cells. At the same time there is no network formation. This is the in vivo study actually, so showing that when we put the control actually the cancer cell media, you can see how many blood vessels are there in chick embryo, and see here when we put the medium of the cancer stem cell supernatant, there are so many blood vessels are formed. So they are getting more food, and so they can take more food from the environment also, and they can form for actually for migration they need to go from one place to another place and again invade there. So see here the different here the invadopodia formations, and here also these are the invadopodias, the processes through which they can invade somewhere. Even they can go to bone. We get metastasis in bone also. Have you ever thought bone is a so hard actually tissue, but we can get cancer there also. So they can go inside the bone also. And so this is the experiment to show that yes, they are showing higher invasive property than the actually the here you can see lesser invadopodia formation, here cancer stem cells more invadopodia formation, here also the processes you can see more than the cancer cells themselves. And then they have the higher migration property also. We are here comparing cancer stem cells with cancer cells. Higher migration property, here you see that they are forming lesser metastatic nodules here when the injection was given with cancer cells. And here you can see so many nodule formation when you directly inject nude mice with the cancer stem cells. So all the properties are matching with the properties of cancer stem cells, and when we give drugs to these mice with cancer, you see the cancer formed by the cancer stem cells, they are not actually curing. They are not getting cured, but if the cancer formed from the cancer cells after a certain time period, that those cancer can be cured, because I have told you already that cancer cells can be killed by different types of drugs. So that is actually going on here. And so we I started with the hurdles of today's therapy, cancer therapies. What are those hurdles? So for that I have chosen two types of therapy, major therapies of cancer. One is chemotherapy, another is immunotherapy. You all know about all these. So first I will talk about a little bit about chemotherapy, nothing in details. So we have found in our laboratory that when we took different types of cancer cells, and whenever we put lethal doses of chemotherapy, so that it is just below the toxic level of chemotherapy, you cannot give patient the toxic level. So up to that dose you can see here the graphs are coming down. All the graphs are coming down, whatever may be the drug, whatever may be the cancer cells, lung, colon, breast, many cancer, actually we many battery of cancer cells we take. Everywhere it is coming down, but never and never it touches the x-axis. What does it mean? That means it is never zero, but we are giving just the lethal dose. So it should touch x-axis, it should become zero. Always there are dead cells, but always there are drug surviving cells. Actually what is happening, when we put conventional chemotherapy, the cancer cells are already dead, but the cancer stem cells are sitting there. So this graph is never touching zero because we are counting cells. So cells are there, very little, but cells are there. But they are cancer stem cells. They have stem cell property. So when the drug is withdrawn, again they produce all the cancer cells and the tumor relapses. Okay, so we have to find out something. I will not show any, I will not talk about any drug today, any medicine today. Today I am only explaining to you why the therapies are failing. So something should be, first of all, we have to understand what are cancer stem cells, actually how they are actually, escape all these treatments. Unless you understand how the thief is escaping after, theft, then how the police will catch the thief. They have to know whether they are through pipe or through window, where from they are escaping. We also have same simple things. We have to know what are the mechanism they are using to escape all these drugs. Either it is chemo or immunotherapy. So chemotherapy, another very, we became very sad to know that first of all, chemotherapy cannot kill cancer stem cells, I have proved. And then now the problem is that first it cannot kill, number two, it increases actually all the drugs, they increase cancer stem cell number. Can you imagine? Chemotherapy is failing to kill cancer stem cell. This is one part. But on the top of that, chemotherapies are increasing cancer stem cell number. So this is really frustrating. This is in the cell line also. We found many types of cancer cells we took, different types of cancer. Always we found that with chemotherapy, the number of cancer stem cells are increasing. So this is in the cell line, or we have also worked in the patient directly. So you can see that with the grade 3-4 cycle of chemotherapy, you can see from 1.09, it has become 6.78. It is the graph is increasing, graph of cancer stem cell. Okay, because their number is very little, so 1 to 6, it is high increase. Very high increase, because their numbers are finally very little. They don't need many cancer stem cells. So this is very grave one. And again, okay, I will not show all these things in detail. And we found that higher cancer stem cell content in the tumor surgical margin cells. What are those? যখন কোনো ব্রেস্ট ক্যান্সার হয় তখন যতটুকু জায়গায় টিউমার বোঝা গেছে তার বাইরেরও কিছুটা কেটে নেওয়া হয়, বাদ করা হয়, যাতে ওখানে যদি কোনো সেল টেল থাকে ক্যান্সারের লুকিয়ে থাকে তারাও যাতে বাদ হয়। তো সেই যে মার্জিনের সেল, সেখানে আমরা দেখছি ক্যান্সার স্টেম সেল নাম্বার অ্যাকচুয়ালি বেড়ে যাচ্ছে যখন আমরা কোনো কেমোথেরাপি করছি। নিউ অ্যাডজুভেন্ট থেরাপি মানে কেমোথেরাপি প্রথমে করে তারপরে সার্জারি করা, তার নাম্বার বেড়ে যাচ্ছে সবসময়, ক্যান্সার স্টেম সেল নাম্বার বেড়ে যাচ্ছে সেখানে। এটা তো সাংঘাতিক খারাপ ব্যাপার একটা। তা কী হচ্ছে অ্যাকচুয়ালি cancer stem cell যখন after formation of the tumor, because they have cancer stem cells are higher migratory, I showed you that they are migrating quickly than the cancer cells, so they go here and there in this normal region, and after even surgery the tumor is no more here, because I told you that neoadjuvant therapy means first chemotherapy then surgery. So you can ask me though, after surgery why cancer stem cells are still there, because they go to the normal place and here and there and hide there. And so when all the therapies are taken out after surgery, they come back for local relapse and make all the tumor again, or can migrate to other side to form metastatic cancer. So then we again ask, scientists, like all the lawyers and advocates, we also always ask questions. So always asking question is the number one job for us. For that government is paying so much money. What are the causes behind increase? Again we have to know why their numbers are increasing, otherwise how we will control that? আমি যদি না জানি যে ওর কেন নাম্বার বাড়ছে, নাম্বার বেড়েছে হাহুতাশ করে বসে রইলাম, তাহলে কি করে তাদের নাম্বারটা আমি কমাবো? Always you have to ask question and then try to find out the answer. So that was our question, why chemotherapeutic drugs are increasing the number. So here is the answer, that as I told you already, that cancer, like stem cells, cancer stem cells under normal condition, these are the markers, forget about that. There are two types of division. One is only symmetrical division, like all our cells. দুটোই ক্যান্সার সেল তৈরি করলো, আর একটা কি হচ্ছে দেখো, একটা ছোট একটা বড়, তাহলে একটা ক্যান্সার স্টেম সেল হবে আর একটা থেকে ডিফারেন্সিয়েট করে ক্যান্সার সেল হবে। দিস ইজ দ্য নর্মাল প্রসেস অফ ডিভিশন ফর ক্যান্সার স্টেম সেলস। But when we are applying doxorubicin, one of the chemotherapeutic drugs, see here we find under normal condition both symmetrical division and asymmetric division. These are very interesting actually. But when we applied chemotherapy in the laboratory, এগুলো আমাদের cell culture, patient না, দেখো এখানে only we are getting symmetrical division. তার মানে ও বুঝে গেছে যে যদি আমি এখন ক্যান্সার সেল তৈরি করি তাহলে তো কেমোথেরাপি মেরে ফেলবে, ক্যান্সার সেল তৈরি করা বন্ধ করে দিয়েছে, শুধুমাত্র সিমেট্রিক ডিভিশন করে ক্যান্সার স্টেম সেল নাম্বার বাড়াচ্ছে অ্যান্ড উই আর গেটিং হায়ার নাম্বার। এটা একটা প্রসিডিওর। আর একটা প্রসিডিওর তো আরও চমকপ্রদ, যেগুলো ক্যান্সার সেলস they understand now that if we remain as cancer cell then we will die because chemotherapy is given. So they are getting converted. I will again not talk about the very complicated mechanism because on these maybe 3-4 papers in grade 1 journal my students have actually published, but this is the gist of that cancer stem cells, when we are giving chemotherapy, again now you know the cytokine because my previous speaker was talking about cytokines. So cancer stem cells produce some cytokines and those cytokines go to the near vicinities cancer cells, and now cancer cells can sense that oh we cannot stay as cancer cells, then we will die. Now they are getting converted to cancer stem cells. আমরা আগে জানতাম স্টেম সেল থেকে তলার দিকটায় নাবে, এখন দেখা যাচ্ছে ভেরি নিউ কনসেপ্ট যে তলার এমনি সেলগুলো ব্যাক করে আবার স্টেম সেল হয়ে যাচ্ছে। দিস ইজ ভেরি নিউ কনসেপ্ট অফ ক্যান্সার স্টেম সেলস অ্যাকচুয়ালি। সো ফার্স্ট অফ অল দে proliferate differently, only symmetric division to make more cancer stem cells. Number 2, cancer cells are getting converted to cancer stem cells, thereby finally increasing the numbers. So এখানে সেটাই দেখানো হয়েছে যে ডিফারেন্সিয়েটেড ক্যান্সার সেলরা এমনি সময় ক্যান্সার এই যে ক্যান্সার স্টেম সেল থেকে ডিউরিং কেমোথেরাপি এই একটা IL-6 সাইটোকাইন তারা যখন এখানে যাচ্ছে they undergo de-differentiation. Differentiation is the process by which cancer stem cells become cancer cells. কিন্তু যখন কেমোথেরাপি করা হয় ক্যান্সার সেলস তারা ডিফারেন্সিয়েট করে ক্যান্সার স্টেম সেল তৈরি করছে। তো অ্যাজ এ রেজাল্ট আমরা টোটাল নাম্বার বেশি পাচ্ছি। তাহলে ফ্রম আপ টু দিস আই হ্যাভ শোন দ্যাট হাউ কেমোথেরাপি হোয়াই অ্যান্ড হাউ কেমোথেরাপি ইজ ফেলিং। সো ক্লিয়ার টু ইউ অল? এরম আসতে বললে তো আমি ভীষণ দুঃখ পেয়ে যাচ্ছি, আমি এত চেঁচাচ্ছি দেখো। তুই তো জানিসই আমি কথা আরম্ভ করলে থামি না, নো ফুল স্টপ নাথিং, আর বলে কি হবে, কিছু কোনো উপায় নেই। আর তোমাদের আজকে গেল খাওয়া দাওয়া সব বন্ধ। তাহলে এবার চট করে চলে আসি ইমিউনোথেরাপিতে। কেন? নট বিকজ ইউ আর হাংরি, বিকজ আই এম হাংরি। সো নাও উই উইল টক এ লিটল বিট অ্যাবাউট ইমিউনোথেরাপি, অ্যানাদার ভেরি কমপ্লিকেটেড ওয়ান। সো ক্যান্সার ইমিউনোথেরাপি অ্যাকচুয়ালি কী করতে চায় তোমাদের কারোর আইডিয়া আছে? ক্যান্সার ইমিউনোথেরাপি কিছু করতে চায় না, ওরা বাইরে থেকে ক্যান্সার যার হয়েছে তার ইমিউন সেলগুলো অনেক সময় বাইরে নিয়ে আসে, বা অন্য ভ্যাকসিনও আছে, সেইগুলো দিয়ে যেটা করতে চায় সেটা হচ্ছে বডির মধ্যে যে ইমিউন সিস্টেম। All of you understand Bengali? Sometimes I am talking in Bengali. You all understand? Okay, if even one person cannot understand, then I will not, of course, speak in Bengali. So, cancer, এই যে ইমিউন সিস্টেমগুলো অ্যাকচুয়ালি ক্যান্সারের ওই প্রপার্টিটা আমি আজকে আগে দেখাইনি, যে ক্যান্সার হলে কিন্তু আমাদের ইমিউন সিস্টেম সম্পূর্ণ ধ্বস্ত হয়ে যায়। তাই জন্য ইমিউন সিস্টেম কেন থাকে? নট অনলি টু ফাইট ব্যাক মাইক্রো অর্গানিজমস বাট অলসো fight against anything abnormal in us inside our body. And cancer কি করে হয়? আমাদের বডির সেলই তো চেঞ্জ হয়ে ক্যান্সার হচ্ছে। Am I right? Nothing coming from outside. No cell is coming from outside. So, but because immune systems are actually getting killed by cancer cells, so there is immune cells are not available. এটা আমি থিওরিটা বলছি। Immune cells are not available for fighting back against cancer. তো ইমিউনোথেরাপি কী করে? ইমিউনোথেরাপি ট্রাইস টু অ্যাকচুয়ালি এগেন এনার্জাইজ দ্য ক্যান্সার ইমিউন সেলস। বাইরে থেকে ইমিউন সেল পাঠিয়ে অথবা বডির ভেতরের ইমিউন সেলকে উদ্বুদ্ধ করে তার সংখ্যা বাড়িয়ে, তাহলে কী হবে? তারা ফাইট ব্যাক করবে ক্যান্সারের সাথে। দ্যাট ইজ দ্য বেসিক কনসেপ্ট অফ ইমিউনোথেরাপি, যে বডিরই ইমিউন সিস্টেমকে বুস্ট আপ করা। ওকে, ক্লিয়ার? তো এইটা হচ্ছে থিওরি। কিন্তু দেখো, with a high hope immunotherapy started, if we can boost up our own immune system, so no toxicity will be there, unlike chemotherapy or radiotherapy, and so very easily cancer cells will be killed. So, but see the scenario, the targeted therapy and standard therapy এইভাবে কমে যায়, মানে সার্ভাইভাল কত দেখাচ্ছে এখানে, সার্ভাইভাল যে সার্ভাইভালটা 3 বছরে গিয়ে অলমোস্ট 3 থেকে 5 বছরে বেশিরভাগ এই সব থেরাপিতে মানুষ কিন্তু মরেই যায়, তাই তো। এখানে দেখো when immunotherapy applied, see কতটুকু বাড়লো, কতটুকু বাড়লো, কয়েক মিলিয়ন মানে মানে ডলার খরচা হয় একটা ইমিউনোথেরাপির পেছনে, আমাদের দেশে কয়েক কোটি টাকা একটা ইমিউনোথেরাপিতে লাগে। কিন্তু ধরো 100 জনকে ইমিউনোথেরাপি দেওয়া হলো, প্রত্যেকে এত কোটি টাকা করে খরচা করলে আমাদের দরিদ্র দেশে ছাড়বে কিন্তু তার মধ্যে ম্যাক্সিমাম 25% ইভেন আফটার ইমিউনোথেরাপি। তো কি হলো, শেষ অবধি সংসার মানে পুরো ভেসে গেল। কিন্তু অ্যাকচুয়ালি এফেক্টিভ মাত্র আজকে টিল টুডে, এটা গ্লোবাল, আমি ওইগুলো শক্ত জিনিস নিয়ে আসিনি, গ্লোবাল পিকচার হচ্ছে 20 to 25% কিন্তু পেশেন্ট সারবে, আদার্স উইল ডাই। আর সব থেকে দুঃখজনক, একজন ক্যান্সার পেশেন্ট যতদিন বেঁচে থাকতো, ধরো কোনো থেরাপি করা হয়নি, সে তো একটা কিছু কাল তো বাঁচবে। ইমিউনোথেরাপি যদি ফেলিওর হয় তাহলে তার থেকে কম সময় বাঁচবে। দ্যাট ইজ দ্য মোস্ট মানে গ্রেভেস্ট পার্ট অফ দ্য হোল কনসেপ্ট। So we started from there. Few people actually benefit from the breakthrough of cancer. Very good actually concept, very good concept. So we ask the question, why immunotherapy is failing? Because cancer stem cell concept মাত্র 20-25 বছরের বলে ক্যান্সার স্টেম সেলের দিকে এখনো পর্যন্ত ডিপ রুটেড কিন্তু অ্যাটেনশন দেওয়া হয় না, so far as the treatments are concerned. এখনো কিন্তু ক্যান্সার সেলকেই সবসময় ভেবে চলা হচ্ছে। রিসেন্টলি ক্যান্সার স্টেম সেল নিয়ে একটু একটু অ্যাওয়ারনেস বাড়ছে। কলকাতাতে আমাদের টাটা মেডিকেল সেন্টারে যারা ডাক্তার, আমার কোলাবোরেটার সবাই অনেকেই, তারা প্রথম দিকে তো দে ডিডন্ট মানে অ্যাকচুয়ালি বিলিভ মি যে ক্যান্সার স্টেম সেল আরে রাখো না, ওসব না। পরের দিকে দে স্টার্টেড আস্কিং মি, ডু ইউ হ্যাভ সামথিং টু সে অ্যাবাউট দিস রিগার্ডিং ক্যান্সার স্টেম সেলস। আমাদের জয়েন্ট মিটিং যখন হচ্ছে বলতো তানিয়া শুড হ্যাভ সামথিং টু সে অ্যাবাউট her opinion, what she thinks about why things are failing. So slowly আমাদের এই অ্যাওয়ারনেসটা বাড়ছে। কিন্তু আমি সেই কোশ্চেনটা এখানে আবার করলাম, ক্যান্সার স্টেম সেলরা কি কোনোভাবে রেসপন্সিবল? তো দেখো আমরা দেখতে চাই যে এই ক্যান্সার স্টেম সেলরা, আমি বলেছি যে ক্যান্সার সেল নয়, ক্যান্সার স্টেম সেলরাই কিন্তু টিউমার বানায়, বলেছি কি না? তাহলে যখন ক্যান্সার স্টেম সেল, আবার এটাও বলেছি যে তাদের সংখ্যা ভীষণ কম, কিন্তু আমাদের বডির সারা জায়গায় ইমিউন সেলের সংখ্যা কত? প্রচুর, যে কোনো টিস্যুতে প্রচুর। তাহলে এবার ইউ ক্যান ইজিলি ইমাজিন ইফ 3, 4, 5%, 5, 6 or 10, 100 cancer stem cells are in a tissue are trying to make a tumor, but there are so many immune cells at that time, তখন তো ক্যান্সার হয়ে যায়নি যে ইমিউন সাপ্রেশন হবে। তাহলে হোয়াই দে আর নট কিলিং দোজ ক্যান্সার স্টেম সেলস? ওরা তো 5টা 6টা 10টা 12টা কিল করে ফেললেই তো, আর তারা ইমিউন সেল তো তখন প্রচুর ছিল। তাহলে কি হয়? তাহলে ক্যান্সার স্টেম সেলের কি এরকম কোনো প্রপার্টি আছে যে তারা ইমিউন সেলকে এসকেপ করতে পারে, যেমন ড্রাগকে এসকেপ করতে পারে? তাহলে ইনিশিয়েটার সেল হলে তাকে তো ইমিউন সেলকে এসকেপ করতে হবে, নাহলে তো সে বানাতে পারবে না। মাদার নেচার এভাবে তৈরি করবে না যে সে গেল আর মরেই গেল, তাই না? তো সেইখান থেকে আমরা ভাবলাম মেবি ক্যান্সার স্টেম সেলস মাইট হ্যাভ এনাফ ওয়েপন টু ইভেন্ট ইমিউন অ্যাটাক টু। কেন ওরা ইনিশিয়েটার সেল। কিভাবে করতে পারে? দুভাবে কিন্তু এটা করতে পারে, এটা হাইপোথিসিস আমার। এক হচ্ছে ইমিউন রেকগনিশন। এর মানে কি? when the immune cells are coming, they have to understand that these are cancer stem cells or cancer cells. Am I right? They have to know, at least understand. তুমি জানবে না যে দিলীপ স্যারকে কেমন দেখতে, তুমি কার সাথে গিয়ে দিলীপ স্যারের কথা বলবে? ফাউন্ডার ডিরেক্টর স্যারকে চেনো না, কাকে একটা বলে দিলে স্যার আপনি যা বানিয়েছেন না কি হলো, বুঝেছো? তাহলে কি অবস্থা? আমার তো অবস্থা খারাপ, আর জীবনও ডাকবেন না। এবার হচ্ছে সেজন্য আমাকে প্রথমে চিনতে হবে যে তাদের কিরকম, মানে সেই কারণে ইমিউন সেলরা ক্যান্সার সেল বা ক্যান্সার স্টেম সেলকে চিনতে হবে প্রথমে। দে হ্যাভ টু রিকগনাইজ। তাহলে ক্যান্সার স্টেম সেলরা কি লুকিয়ে থাকতে পারে? evade this recognition so that immune cells cannot recognize, দেখতে দেখে বুঝতে পারছে না যে ওরা একটা অন্য রকমের কোনো সেল। এটা একটা হতে পারে। 2 নম্বর হচ্ছে যে as I already told you that the environment in our body, it is full of immune cells, তাই তো? প্রচুর ইমিউন সেলস। টিউমার মাইক্রোএনভায়রনমেন্টে প্রচুর ইমিউন সেল থাকে, 80% ইমিউন সেল, 20% অনলি ক্যান্সার সেল আর ক্যান্সার স্টেম সেল, টোটালিটিতে বলছি। তাহলে সেই যে ইমিউন সেলগুলো, even after formation of the tumor, why they are not, these immune cells, why they are not targeting those cancer cells or cancer stem cells for that matter? তার মানে কি ক্যান্সার সেলগুলো বা ক্যান্সার স্টেম সেলরা ইমিউন সেলগুলোকে কাজ করতে দিচ্ছে না, এক হলো তাকে চিনতে দিচ্ছে না, দুই হচ্ছে নট অ্যালাউইং টু ওয়ার্ক, হাউ, তাকে কনভার্ট করে দিচ্ছে অন্য কিছুতে, অথবা নিজে এমন একটা রূপ ধারণ করছে যাতে ওরা কিছুতেই অ্যাটাক করতে পারছে না। লেটস সি ভেরি ব্রিফলি, আই উইল শো ইউ ইমিউন রেকগনিশনের জন্য যেগুলো দরকার হয়, যে ক্যান্সার স্টেম সেল আর ক্যান্সার সেলের বাইরে কিছু কিছু নতুন ধরনের প্রোটিন, যাদের বলা হয় নিও অ্যান্টিজেন্স। You know about antigens, so the proteins. So these are neo means new. So when there is a cancer tumor mass, then the cancer cells have some new type of proteins on the surface. And when proteins are formed inside the cell through MHC class 1 proteins, they get transported to the surface. So first of all, the antigens has to form. Number 2, they have to go to the surface. So what happens, I will show you, I will not again show you all these complicated things. What happens is that cancer stem cells actually DNA mismatch repair. Why the new antigens are formed? Because in the cancer stem cells, in the cancer cells, there are DNA mismatch, there are mutations. So when if the mutations in the cells get cured, then there will be no antigen. We have seen that in cancer stem cells, actually mutations are actually DNA mismatch repair is higher in cancer stem cells. So mutations get repaired. So antigen is less. And another thing is that, as I told you about MHC molecules who carries the antigens to the surface, they are lesser in the cancer stem cells. So for this there is lesser production of neoantigens. For this, even if some are produced, they are not being able to go to the surface. So there are no neoantigens or lesser neoantigens on the surface, and then the immune cells cannot recognize them. Clear? And then number 2, whether the cancer stem cell make a immunosuppressive environment. See here, as I already discussed, that there are many immune cells in the tumor microenvironment. There are T cells, you know about T cells, they kill cancer cells. But we have seen that, again not in details, that we have seen that actually cancer stem cells actually convert these T cells to T regulatory cells. T regulatory cells are in favor of the tumor. So when there is cancer stem cell, they make the T cell, CAR T cell jodi inject koro, we have seen in our laboratory in adaptive transfer of T cells, we have seen that all the T cells are becoming T regulatory cells. So they are pro-tumor, ওরা টিউমারকে সাপোর্ট দেয়। সো ক্যান্সার স্টেম সেলরা কি ডেঞ্জারাস ভাবে রিকগনাইজ করতে দিচ্ছে না, অ্যান্টিজেন তৈরি করছে না, তারপরে হচ্ছে এনভায়রনমেন্টকে করে দিচ্ছে সাপ্রেসিভ এনভায়রনমেন্ট হওয়ার জন্য টি সেল টি রেগুলেটরি সেল হয়ে গেছে। From CSC they shed exosome containing different chemicals that make T cells to T regulatory, this arrow should be this way, and so there is immune suppression. And again, this is the macrophages. M1 macrophage, they can kill cancer cells, but they cannot kill cancer stem cells. So another process of immunotherapy is CAR T cell. You all know about CAR T cells therapy. So CAR T cells, when that is given, CAR T cells are becoming T regulatory cells. So those are not effective anymore. Number 2, there is macrophage therapy. How? Like M1 macrophage can kill cancer cells, but they easily can be converted to M2 macrophages, which cannot kill cancer cells. So the theory of the therapy is to make M1 macrophage again. But we have seen our laboratory, these are again many experiments, we have seen that M1 macrophages even cannot kill cancer stem cells. So T cells are lesser, M1 macrophage cannot kill cancer cells. So they are actually employing multiple tactics to ensure the failure of cancer therapies. So when chemotherapy is given, then the increase of CSC, and they are evading, they are highly drug resistant, evading the therapy and increasing the number. and when immunotherapy is given, their drug resistance pumps are high and the neoantigen production is lesser. MHC, which are carrier of the antigens, that is lesser. T regulatory cells are high and M1 macrophage cannot work. So it is using multiple weapons. So usually they are supposed to kill the cancer stem cells, but when there is CSCs, you can have you seen the change in their color. So that way I try to make you understand the cancer stem cell are changing all types of immune cells to some different types of cell, same cell but properties are different. So they are now from anti-tumor, they have become now pro-tumor. So all the immunotherapies are now failing. So and the cancer stem cell, because of the stem cell property, now they are making the total tumor mass. These are normal cancer cells, and so there is failure of cancer therapy, any type of cancer therapy, and so there is again relapse. And these findings together not only provide opportunity for predicting the outcome of cancer therapies. এটা খুব ইম্পর্টেন্ট, কারণ আমি তোমাদের বললাম যে মাত্র 25% ইমিউনোথেরাপিতে সাকসেসফুল হবে, কিন্তু আমাদের কোনো রকম এখন পর্যন্ত সেভাবে টেস্ট করে দেওয়া হয় না যে কোন পেশেন্ট সাকসেসফুল হবে। তাহলে আমি এখন এখান থেকে বুঝলাম যে অন্তত যদি ক্যান্সার স্টেম সেল মার্কারগুলোকে চেক করে যে তার এই মার্কারের কতখানি আছে, বেশি থাকলে তার তো ইমিউনোথেরাপি কাজ করবে না, অথচ এত কোটি কোটি টাকা খরচা হয়ে যাবে। অন্তত একটা মার্কার পাওয়া গেল যে যেটা আমরা দেখতে পারবো চেক করে যে এর সংখ্যা যদি খুব বেশি থাকে কারুর মধ্যে, তাহলে কিন্তু তার চান্স কমবে অবশ্যই ইমিউনোথেরাপি করলে। এতখানি তাহলে অন্তত আমরা একটা মার্কার পেলাম, আর যেটা বলে দেবে যে তোমার সাকসেসফুল হবে কি হবে না, এবং একটা পসিবিলিটি পেলাম, আজকে আমি পসিবিলিটির কথা বলবো না, দ্যাট যে বাই টার্গেটিং দ্যাট ক্যান্সার স্টেম সেলস can be in future, cancer therapy can be made successful. So that much dream I can dream actually. So hurdles can be crossed to say one day that victory over cancer, to wake up in a cancer free world. That is my dream actually. They are my funding agency, this is now the major one, and so my collaborator, he was here, and these are my overseas collaborators, many others are there, but the part of the Diptendra is a major doctor with whom I collaborate, but I have many other doctor collaborators in other hospitals also. But for today's talk, what I showed, they have actually contributed this or that. I thank you all, especially the future scientists present in this room. তোমরা অনেকে সায়েন্টিস্ট হওয়ার চেষ্টা করো, এর থেকে বোধহয় নোবেল প্রফেশন আর কিছুতে হয় না, এটা আমার বিশ্বাস। একেবারে যা সম্মান একজন বিজ্ঞানী পায়, এই সম্মান পাওয়া সম্ভব না অন্য কিছুতে। আমার ইনস্টিটিউট, আমি খুব প্রাউড যে আমি বোস ইনস্টিটিউটের মতো, যেটা একটা পৃথিবীর অন্যতম শ্রেষ্ঠ ইনস্টিটিউট, সেখানে কাজ করার সুযোগ পেয়েছি, এবং থ্যাংক ইউ সো ভেরি মাচ ফর পেশেন্স হিয়ারিং। কি আর করবে বলো, খেতেও যেতে পারছো না, শুনতেই হবে বসে।
Thank you very much, madam, for such a nice and beautiful talk. আমরা ক্যান্সার স্টেম সেল সম্পর্কে অনেক কিছু জানতে পারলাম। Now this is the time for questions. So if you have any questions, students, I know that you are hungry, but if you have any questions, okay, Professor NK sir has a question.
Madam, thank you very much for your fantastic lecture.
Thank you.
I have got some silly questions.
Questions can never be silly.
Which may seem to be silly at your level. First thing is, are the cancer stem cells similar for different types of tumors, like breast tumor, uterine tumor, prostate cancer? Are the stem cells different for different kinds of tumor? First question. Second thing, when you introduce or inject stem cells into some animal body, do they follow any specificity, or they can at random invade any organ? And finally, as a scientist, do you think that cancer chemotherapy should be very carefully monitored so that the number of stem cells does not increase in the patient's body?
Yeah, very good questions actually. Not at all silly, not at all naive, very good questions. First of all, as I remember, my memory is very poor nowadays. Whether all the cancers have same type of cancer stem cells, actually this is a very good question, and all the cancers have same type in the sense, quote unquote, that their stem cell properties are all same. Markers so far as the stem cell marker like OCT4, SOX2 nano or drug resistance pumps, those are same, but surface markers are different. There are, we actually measure 13, 14 surface markers for each type of cancer stem cell, from each type of cancer, for each experiment every day my students they measure, and there are some differences in the surface markers from lung cancer to breast cancer to other cancer.
Means difference in the neoantigens or what?
I should not say those are not that way neoantigens. The properties of neoantigens that are dependent on the mutation of the gene. From there they come, but these are the real proteins formed regularly in the cancer stem cells, like CD44 is one, and OCT4, SOX2 nano are universal for all the stem cells and cancer stem cells. So all the stem cells marker are usually similar, but cancer markers are usually different for different cancer. Number one. Number two, I've already forgot.
The second thing is, if you inject stem cells into the body of an animal, do they follow any specificity or they at random invade any organ?
Yeah, this is another very good question. Nobody asked me that is, thank you very much. Whatever, when we do the experiment, like as I showed you, when usually we, it can form tumor, like I showed you, like in the mice, it was breast cancer stem cells injected, but injected on the here, on the skin. So it can form the tumor, again because of the stem cell property.
And skin also?
Yeah, skin also, and sometimes we directly push inside the mammary pad, so then it is breast cancer, and then if you inject in the tail vein, usually they go to the lung, everywhere they can go. But once it is in the organ, like they now start following the metastatic properties of their own, so when they are in the lung, they will form cancer stem cells and lung cancer cells. When they are in the breast, depending on the environment and everything is different. You had another question.
Finally, I asked, as a scientist, do you think that
chemotherapy
should
yes,
be
of course.
carefully and periodically monitored so
yeah,
that...
of course.
And one more thing, does the do the clinicians or pathological laboratories have any way to check the number of stem cells?
Yes, I understand that. Why not? Because they have markers, and we are also checking in the same way. But you have to also understand, when the cancer researchটা এত বছর ধরে চলছে, কনসেপ্ট 100 পেরিয়ে গেছে, তাতে এখন অবধি আমরা কিছু বুঝে উঠতে পারিনি। ক্যান্সার স্টেম সেল মাত্র 25 বছর, মানে একদম শিশু, রিসার্চ বলতে গেলে একদম শিশু। So এখন till today we are trying to understand and discover the properties of cancer stem cells. এখন আমরা আমাদের এনএমইকে পুরোটা বুঝতে পারিনি, সেই জন্য এখনো কোনো ল্যাবরেটরি কিন্তু মানে অসুখের পরে, ট্রিটমেন্টের পরে এখন অবধি কিন্তু চেক করে না। কিন্তু এইগুলো পুরোটা যখন এটা টোটাল কংক্রিটলি প্রুভড হবে, তখন কিন্তু এই জিনিসগুলো অবশ্যই ধরবে। No, nowadays concept has been changed. Previously I have also faced that when I first started. So I was in Cleveland clinic that time. So I was reading many things in between, our meeting, etc. And then, so I was reading things, and suddenly I started working many years ago on cancer. But that time suddenly this cancer stem cell article, one of those articles, what is this? Then I started thinking that why not? I will also work on cancer stem cell. Just that time, cancer stem cell concept, it was arising. And so I understand that, like chemotherapy to radiation therapy, then hormone therapy, different therapies, and now immunotherapy. So one after another staircase এর মতো একটার পর একটা তো তৈরি হয়, সেরকম ভাবেই ক্যান্সার স্টেম সেল। বাট আনলেস more proof comes, then a trial করতে হবে। It will take again maybe at least 5, 6 or 10 years at least it will take to একদম এই লেভেলে আসা পর্যন্ত।
Doctors advise to measure blood levels of certain
No, not till today. No. But it will start. They will surely start, because more awareness should be generated about cancer stem cells. So now it has been started. People are aware about cancer stem cells. Not general public, but doctors.
আমাদের দেশে তো কোনো
না এখন অবধি হয় না, কোথাও
Prescription
ক্যান্সার
হয়
স্টেম
না,
সেল খুব একটা
advice
হয়
হয়
না।
না।
মানে ক্যান্সার স্টেম সেল আমি অন্যান্য দেশেও বলছি, ক্যান্সার স্টেম সেল এখন ডিরেক্টলি মানে হসপিটাল বা ক্লিনিকে এখন অবধি কিন্তু এটা এক্সপেরিমেন্টাল স্টেজে আছে। এখন অল পিপল আর টেকিং থিংস, তারা বিশ্বাস করছে, কিন্তু সব কিছু ক্যান্সার স্টেম সেলের মার্কারও তো দিতে হবে, আর যেগুলো ব্লাডে আসছে। ব্লাডে না এলে কি করে, একটা মানুষ আছে তার কেমন করে রোজ দিন তার কাটাছেড়া করবে না, কিন্তু বায়োপসি করার সময় যেটা দেখতে হবে, এগুলো স্লোলি এটা তো নতুন সাবজেক্ট। হ্যাঁ, ভেরি ইন্টারেস্টিং। আর এটা হলে আমার ধারণা এটার দিকে নজর না দিলে ক্যান্সার কোনোদিনই কমবে না।
Is there any more questions, especially from students?
Thank you, madam.
If not, let's.
ওই যে ও বলবে, ওই যে জিজ্ঞেস করবো, হাত তুলেছে।
ম্যাম যেমন বললে মেটাস্ট্যাসিস হয়, মেটাস্ট্যাসিসের পরে যখন মানে অন্য অর্গানে যখন মানে মাইগ্রেট করে, তারপরে কি অন্য কোনো ক্যারেক্টারিস্টিক দেখা যায় না সেম এতেই বোঝা যায়? যে যেমন ধরুন ব্রেস্ট ক্যান্সার থেকে যখন বোনে বা ব্রেনে যখন অন্য অর্গানে যায়, আলাদা কোনো ক্যারেক্টারিস্টিক দেখা যায় না সেটাই?
Again, very good question actually, very good. দেখো, যে ক্যান্সার স্টেম সেলটা মাইগ্রেট করে যাচ্ছে, নাম্বার 1 হচ্ছে ক্যান্সার টিউমার টিস্যুতে যেহেতু প্রচুর মানে 80%-এর, আমি 80% 95%ই হচ্ছে ক্যান্সার সেল, তাই সবারই ধারণা, আমাদেরও ধারণা এটাই ছিল যে এত ক্যান্সার সেল গিয়ে অন্য অর্গানে গিয়ে সেখানে মেটাস্টাসিস করছে। এখন দেখা গেছে যে আমি এখানে আজকে দেখিয়েছি যে তোমার যারা ব্লাডের থ্রু দিয়ে ধরো যাচ্ছে, যত ইমিউন সেল আছে, ম্যাক্রোফাজ থেকে আরম্ভ করে টি সেল, কেউ তো ক্যান্সার স্টেম সেলকে কিল করতে পারছে না, বাট দে ক্যান কিল ক্যান্সার সেলস। তাহলে পরের অর্গানটায় খুব কম ক্যান্সার সেল পৌঁছায়, ক্যান্সার স্টেম সেলই মেজর পৌঁছায়। তাহলে তারপরে আমি দেখিয়েছি যে তাদের ইনভেট করার, ঢুকার প্রপার্টিটাও বেশি, ইনভেট করে ঢুকলো ভেতরে। এই পার্টিকুলার ক্যান্সার স্টেম সেলটা, এটা কিন্তু এক্সপেরিমেন্টালি প্রুভ হয়নি যে ওই ক্যান্সার স্টেম সেলটা কনভার্ট হয়ে যাচ্ছে কিনা অন্য ক্যান্সার স্টেম সেলে, কিন্তু আমি লজিক্যালি যেটা বলতে পারি, ইভেন ইফ ইট ডাজ নট গেট কনভার্টেড, এর কনভার্টেড হলো না, কিন্তু যখন ওই এনভায়রনমেন্টে গেল, এইবার ও যে সমস্ত কেমিক্যালগুলো শেড করছে, তাতে ওখানের যে এমনি নর্মাল সেল তারাও কিন্তু প্রপার্টি লুজ করছে, চেঞ্জ হচ্ছে। আর ওখানের যে স্টেম সেলগুলো, আমি তো ক্যান্সার স্টেম সেলের অরিজিনের কথা বলিনি আজকে, ক্যান্সার স্টেম সেল কিন্তু নর্মাল স্টেম সেল থেকেও হতে পারে, নর্মাল মানে এমনি সেলকে চেঞ্জ হয়ে মিউটেশন হয়ে হতে পারে, অনেকভাবে হয়। তো আমার ধারণা যে নর্মাল স্টেম সেলগুলো, দে আর গেনিং দ্য স্টেম সেল ক্যান্সার স্টেম সেল প্রপার্টি, সো নাও দে আর ফ্রম দ্যাট স্পেসিফিক অর্গান। যদি ব্রেস্টের থেকে লাংয়ে গেছে, তো ওইখানে স্টেম সেলগুলো তো লাংয়ের স্টেম সেল, তাদের অল আদার প্রপার্টিজ আর লাইক লাং, so they are becoming lung cancer stem cells. Now the cancer cells they are producing, because they are lung cancer stem cells, they are producing lung cancer cells. বুঝতে পেরেছো? তখন ওখানে তো পরিবেশটা অন্য, ওখানে পরিবেশটা লাংয়ের মতো, সেইখানের এনভায়রনমেন্টে এমনি স্টেম সেলগুলো এই ক্যান্সার স্টেম সেলটার প্রভাবে সে যদি ক্যান্সার স্টেম সেল হয়ে যায়, তো সে তো লাংয়ের ভেতরের ক্যান্সার স্টেম সেল হলো, তার প্রপার্টি লাংয়ের মতো। এবার তার থেকে ডিফারেন্সিয়েট করে যে ক্যান্সার সেল হবে, তারা লাং ক্যান্সার সেল হবে, তারা ব্রেস্ট ক্যান্সার সেল আর হবে না, যদিও অরিজিনটা গেছিল ব্রেস্ট থেকে। দ্যাট ইজ মাই কনসেপ্ট। এইটাই হচ্ছে ক্যান্সার স্টেম সেলের বেশিরভাগটাই এখনো কনসেপ্টে রয়েছে, এভরিথিং হ্যাজ টু বি প্রুভড। ভেরি গুড কোয়েশ্চেন। ডিপেন্ডিং, হাইলি ডিপেন্ডেন্ট অন মাইক্রোএনভায়রনমেন্ট, হাইলি ডিপেন্ডেন্ট। ওটা তো প্রুভড হয়ে গেছে, আমাদের ল্যাবরেটরিগুলোতেও প্রফেসর শাহ প্রচুর কাজ করেন, অ্যাকচুয়ালি উনি মানে ওয়ার্ল্ডেরই একজন ওয়ান অফ দ্য বেস্ট ক্যান্সার ইমিউনোলজিস্ট অ্যাকচুয়ালি। আপনারা শুনলে সকলে ভালো লাগবে যে আমরা করোনার সময়, যদিও কোভিড আমাদের ডিরেক্ট কাজ নয়, কিন্তু এইসব কাজ করতে করতে অনেক মানে ভাস্ট হয়ে যায়, ভিশনটা অনেক বড় হয়ে যায়, সকলেরই হয়, তোমাদেরও হবে, তো অনেক কিছু মিলও পাওয়া যায়। আমি একটা লেকচার দিই, কোনো সময় সুযোগ পেলে নিশ্চয়ই বলবো, কারণ কোভিড নয় শুধু, অনেক ইনফেকশাস ডিজিজ এখনও আসছে, একটা মানে কোথায় কমন মিটিং পয়েন্ট অফ ক্যান্সার অ্যান্ড কোভিড, এটা আমি একটা লেকচার দিই। তো হোয়াট এভার, যেটা বলছিলাম, কোভিডের সময় প্রথমে আমাদের ভ্যাকসিন যে ঠিক হয়েছিল, মনে করে দেখো যে বুস্টার ডোজ বা সেকেন্ড ডোজ অনেক তাড়াতাড়ি দেওয়া হতো আমাদের দেশেও। আর তাহলে পলিসিগুলো কে ঠিক করে? আমাদের দিদিও না, মোদিজিও না, পলিসি কিন্তু সায়েন্টিস্টরাই ঠিক করেন, এবং মেনলি আইসিএমআর থেকে ঠিক হয়, হওয়ার পরে গভর্নমেন্টকে দেওয়া হয় তাদের নেওয়ার জন্য, তাই তো? তো সেখানে যে দুজন সায়েন্টিস্টের কথায় আমাদের টোটাল দেশের এই ভ্যাকসিন যেগুলো আমাদের দেশের অ্যাভেলেবল, সেই ভ্যাকসিনের প্রসিডিওর আমাদের দেশে গ্রাহ্য হয়েছে এবং চেঞ্জ হয়েছে, যে কতদিন পরে পরে দেওয়া হবে। আগে অনেক কাছাকাছি সময় দেওয়া ছিল। কাছাকাছি দিলে শুধু যে ক্ষতি হবে পেশেন্টের তাও নয়, 2 নম্বর কথা হচ্ছে এটা সম্পূর্ণ ইনঅ্যাক্টিভ হয়ে যাবে। এই কথাটা যে দুজন সায়েন্টিস্ট বলেছিলেন, একজন প্রফেসর শ্যামল রায়, উনি আমাদের এখানকার ইমিউনোলজি ফাদার ফিগার, আর একজন কিন্তু প্রফেসর শাহ। এই দুজনের কথাই আমাদের এখানে পলিসিটাই চেঞ্জ হয়েছে। ভীষণ ভীষণ ইমিউনোলজি। তো ওরাও যে ওর ল্যাবেও যে কাজ হচ্ছে প্রফেসর শাহর ল্যাবে, এইটা যে মাইক্রো এনভায়রনমেন্ট কতখানি ইম্পর্টেন্ট, ইমিউন সেলরা কতটা ইম্পর্টেন্ট, ক্যান্সারেরও ফেট নির্ধারণ করাতে, এটাই।
Thank you very much, madam. Let's thanks her with a applause. Now this.
I extend my sincere thanks to our dignitaries and speakers for their informative presentations, which have made this session truly stimulating and fruitful. Thank you, madam, once again. Now we will have our lunch break, and after that we will return to our oral presentation session for research scholars. Thank you.
বুঝেছো? দেখা যাচ্ছে, দেখতে পাচ্ছ? চলো এসছে, এই খাড়া করে তোলো, খাড়া করে। আরেকটা তোলো। ওটা দরকার নেই, শোনো, ওইটার দরকার নেই, তুমি ওইটাকে আমি সেপারেট একটা তুলবো। উঠেছে? কোন দিক থেকে তুলছো? উঠেছে? এইখান থেকে তুলো না, এইখান থেকে তুললে দরকার নেই, এমনি তোলো। হ্যাঁ, এইখান থেকে আবার তুলছো কেন? এদিকে লেখাটা তো আসবে না। এটা কি করছো? এটা কি স্টাইল দেখাও, কি করলে? এটা তো পরিষ্কার।
Thank you everyone gathering here once again. We are about to start our oral presentation session of research scholars. I would like to request Professor Nirmal Kumar Sarkar, former professor of Presidency University and senior most professor of Department of Biology Science, Midnapore City College, to chair the session. Now over to you, sir.
So we are starting this oral presentation by research scholars. Each research scholar will be given 10 minutes for presentation, and 5 minutes will be allotted for oral interaction. But before we start, I will request the supervisors of different scholars not to ask any question in order to avoid all kinds of conflicts of interest. However, I will request our principal, sir, to ask some questions during the interactive session. কেননা হচ্ছে এটা তো বোঝা সবার উচিত। You have learned a lot in your কি বলে ওটা কি কোর্স PhD আগে যেটা পড়ানো হচ্ছে কোর্স ওয়ার্ক যে we should be rigid enough for avoiding all kinds of conflicts of interest. So our first speaker for the day will be Miss Madhumita Pal. সেটা তো এখানে লেখা নেই। Madhumita Pal absent or present? Absent. Then I invite the second speaker, Miss Madhurai Gupta. At first, you will stand up before the podium, introduce yourself in just 1 minute, and you will start your talk when I ask you to do so. Please stand over there. Podiumer samne daran ebong introduce yourself in a minute.
A very good afternoon to respected principal sir, delegate members, staff, students and research scholars. I am Madhurai Gupta. I am research scholar of Natural Science Research Center of Belda College. And my supervisors are Dr. Rakhi De Sharma and Dr. Kaushik Das. And today I will be presenting on a part of my research work. I am still at a very early stage in my research work. So thank you.
So your time starts now.
Thank you, sir. Today my topic of presentation is proximate analysis and fatty acid profile of fermented pumpkin seed flour. Now let us first understand what is fermentation. So it is a metabolic process in which bacteria, yeast, molds, all kind of microorganisms are involved in converting carbohydrates and other substrates into simple sugars, alcohol, gases, etc. It has been widely used in over centuries for food preservation, increasing shelf life, nutrient availability, etc. Fermentation is also extensively applied in the production of dairy products, cereals, vegetables, meat and seed-based foods, which is my topic, pumpkin seeds. And it plays a significant role in the development of functional foods and probiotics and health promoting properties. Now let us look at what are already established health benefits of pumpkin seeds. They are already regarded as a superfood, or they are very much in trend nowadays. First and foremost, it is a very rich source of nutrients and bioactive compounds. It has potential anti-diabetic effects and also metabolic regulations. It has antioxidant and anti-inflammatory activities which has already been observed. They are also very rich in unsaturated fatty acids and phytosterols. So what are phytosterols? They are basically plant-based sterols which look like cholesterol, but they are not. They are healthier and are associated with improved lipid profiles and potential cardioprotective effects. Now let us look at what does fermentation do to seeds. How does it improve its qualities and what is the benefits? First and foremost, it enhances the nutrient bioavailability. Although the nutrient might be present in the seed before, but the bioavailability increases after fermentation. The probiotic potential and gut health support, because fermentation involves some kind of microorganisms, they may have a very good gut-friendly, gut-safe support. Increased bioactive and functional metabolites. Fermentation generates peptides, phenolics, and vitamins with antioxidant and anti-inflammatory properties. There are evidences already established. Cardiovascular and metabolic benefits. Specific fermented foods like natto contain nattokinase and vitamin K2, which is associated with improved lipid metabolism and cardiovascular markers. Now, natto is basically a fermented product of soybean. Antioxidant and anti-inflammatory effects. Fermentation can increase antioxidant capacity and anti-inflammatory activity compared to non-fermented foods. Now, this is the background for which I selected pumpkin seeds, fermented pumpkin seeds. Basically, it belongs to the Curcubita family. These seeds are largely regarded as agricultural waste products in India. They are very widely available. All of us are very aware of pumpkins, but the seeds are mostly wasted away. So, this also correlates with our topic for the MCCCON this year, which is sustainable development. So, but they serve as a powerhouse of nutrients with interesting nutraceutical properties, which plays a key role in disease prevention and health promotion. Seed fermentation by lactic acid bacteria is a common fermentation type. It has been used widely and over a long period of time. It is supposed to be safe. This microbial activity alters the seed's nutrition composition, enhancing preservation, flavor, digestibility with a high value of probiotic substances. Fatty acid profiling, why are we doing that? Because it provides a detailed information of the valuable dietary, healthy fats and more bioavailability to the body compared to the raw components, which is the non-fermented seeds. This study explores the effect of fermentation on pumpkin seed flour in comparison to non-fermented pumpkin seed flour with a controlled condition by using Lactobacillus acidophilus, that is the ATCC 314, that is the number. Now, what was my aim? First was to carry out the proximate analysis, and the second one was to investigate the fatty acid profiling of the fermented seed flour. These were my objectives. First, to validate the total viable count of lactic acid bacteria in fermented pumpkin seeds. Second was to analyze the ash contents of non-fermented as well as fermented pumpkin seeds. Third was to estimate the moisture contents. Fourth was to explore the protein contents. Fifth, to investigate the fat contents. Sixth was to estimate the total fiber content. Seventh, to determine the carbohydrate content. Eighth was to investigate the development of fatty acids. And ninth was to estimate the antioxidant activity in fermented over non-fermented seeds. This is the methodology. First, the seeds were collected. It was cleansed. Then it was air dried in hot air oven. It was grinded, and then an inoculum of Lactobacillus acidophilus was inserted. It was then incubated for 7 days, and every day, I mean 24 hours, 48 hours, 72 hours, 96 hours, similarly till 7 days, the viable count was counted. And then lastly, it was lyophilized at -40 degrees Celsius. That is the microbial count days: 24 hours, 48 hours, 72 hours, 96 hours, 120 hours, 144 hours, and 166 hours. The other part was the proximate analysis of ash and moisture content, which was followed using the standard protocols of AOAC. Same goes with crude protein content, fat content, fiber and carbohydrate content. The fatty acid profiling was done from Fair Labs, Haryana, and antioxidant activity was measured by DPPH assay. This is the resultant discussion part. Now the microbial growth chart showed that the maximum viable count of lactic acid bacteria was found at 96 hours, that is 4 days. So when the seeds were fermented for 4 days, the maximum number of bacteria were found to be there. Now we come to the proximate analysis. The ash content showed no significant difference between fermented and non-fermented seeds. The moisture content showed a little higher in fermented seeds rather than in non-fermented seeds. The protein content was found to be higher in fermented seeds than non-fermented ones. Carbohydrate showed no significant change. The crude fat content was a little higher in non-fermented seeds than fermented ones. Fiber, total fiber content was non-significant, so there was no change. Now we come to the fatty acid content, which is the highlight of the study. These are saturated fatty acids. Stearic acid and palmitic acid, they showed no significant change, but propionic acid, which is a short chain fatty acid, was significantly increased in the fermented seeds as compared to the non-fermented ones. Now, next we come to the monounsaturated or MUFA. There was a significant increase of MUFAs in the fermented seeds as compared to the non-fermented part. Oleic acid significantly increased in fermented seeds, whereas palmitoleic acid showed no significant change. Polyunsaturated fatty acids were a little higher in non-fermented part of the seeds. Linolenic acid and linoleic acid showed no significant changes. So the good part of PUFAs were intact in both non-fermented and fermented parts. Antioxidant activity showed that fermented seeds showed a little more antioxidant activity than the non-fermented ones. So what is the conclusion from this study? The fermentation process led to an increase in short chain fatty acids, likely due to the microbial enzymatic activity that breaks down complex lipids structures into beneficial short chain fatty acids. This fermentation also led to an increase in protein availability, likely due to microbial enzyme activity again, that breaks down complex protein structures into more digestible peptides and amino acids, and availability of crude fat, fiber and carbohydrate. Furthermore, antioxidant activity was significantly elevated post-fermentation, indicating the release of phenolic compounds and formation of bioactive metabolites. The growth of Lactobacillus acidophilus revealed optimal probiotic viability at around 96 hours, that is 4 days of fermentation, affirming the feasibility of using pumpkin seed as a substrate that supports beneficial microbial proliferation. The high viable cell count also suggests that the final fermented product could deliver a functional dose of probiotics when consumed, contributing to gut microbiota modulation and potentially supporting human health. And last, utilizing pumpkin seeds flour aligns with sustainable food production goals by reducing waste and promoting circular bioeconomic practices. These are the future prospects of my work. Future work may explore the profiling of animal toxicity and other biological tests after proper scale-up processes, sensory evaluation, shelf life stability, and in vivo health benefits to further validate its commercial and therapeutic applications. I thank you, sir. Thank you, sir.
So you have to complete in time. You have already crossed 2 minutes.
Sorry.
Anyway,
Sorry.
I think you are a fresher in the research community.
Yes.
And you have to go a long way
Yes.
before you come to some conclusions.
Yes.
The thing is that
Yes, sir. That is.
That I understand.
Yes.
Throughout your talk, you talked about how you could estimate different chemical properties of those fermented pumpkin seed. But I did not hear anything about anti-hyperlipidemic effect of those seeds. তোমাকে বলে আনতে হবে, আমি তো দেখবো যা তুমি এখানে জমা হয়েছে তাই। Anyway, তাতে হচ্ছে লেখা আছে যে anti-hyperlipidemic. This is simply an analysis of chemical properties,
Yes, sir.
or rather physicochemical properties of fermented pumpkin seed.
Yes, sir.
So if you study the chemical properties of pumpkin seed, how does it give any benefit to mankind or environment, control, management, or anything else? Simply studies on the properties of pumpkin seeds. Does it signify anything?
Sir.
Does it contribute to anything?
Sir,
কতগুলো কুমড়োর বীজের ফার্মেন্টেড করলাম, তার ভেতরে কি আছে না আছে দেখলাম। তাতে যদি না করতে তাহলেই বা মানুষের কি আসতে যেত, আর বললি ওরা খুবই ইনসিগনিফিকেন্ট পার্ট অফ ইউর ডেসক্রিপশন। At the last, at the fag end of your talk, you said something about antioxidant activity, and that also you did not show how you assayed those antioxidant activities.
DPPH.
It was a very insignificant part of your entire talk. Anyway, I wish you go ahead.
Thank you, sir.
And see you in future.
Thank you, sir. Thank you.
Next person, please. Nayanika Datto. Miss Nayanika Datto. Please introduce yourself in 1 minute.
Good afternoon, respected chairperson, principal sir, all respected faculty members present here, my research scholars and all students present here. I am Nayanika Datto, research scholar of Medinipur City College, working under the supervision of Dr. Shantanu Kaur Mohapatra. And today I am here to present a part of my work entitled Bioprospecting of Isolated Mannose Binding Lectin from the Nutshell of Arachis hypogea Lin. Thank you.
Now you start it.
Okay. The topic for today is Bioprospecting of Isolated Mannose Binding Lectin from the Nutshell of Arachis hypogea Lin for the macrophage polarization and anti-Leishmanial immune response. So, macrophages exhibit dual nature by interchanging between the pro-inflammatory as well as anti-inflammatory phenotypes. So, and the M1 polarization state is denoted as the treating of the pathogens, while the M2 polarized state is the safe space or the survival of pathogens, specifically the L. donovani parasite. Leishmania is a neglected tropical disease, and based on the clinical manifests, visceral leishmaniasis is the most fatal and deadliest worldwide. The visceral leishmaniasis affects the reticuloendothelial system and is caused by 20 diverse species of the protozoan parasites. Based on the National Vector-Borne Disease Control Program, 4.76 million are at the risk of leishmaniasis solely in West Bengal. And the agent that causes visceral leishmaniasis is the L. donovani parasites, and it has a digenetic life cycle. It completes its life cycle in two different hosts, namely the phlebotomin sandfly and the mammalian host. In India, India accounts for about 18% of the global burden of visceral leishmaniasis, and the main states that are affected by leishmaniasis is Bihar, Jharkhand, Uttar Pradesh and West Bengal. The parasite that causes the visceral leishmaniasis has been reported to be involved in the cell signaling cascade. The LPG protein or the glycoprotein isolated from the leishmanial donovani is capable of interacting with TLR2, which in turn inhibits TLR26 heterodimer TLR4 as well as TLR9, leading to the inhibition of phospho-P38 MAPK, finally inhibiting the generation of NO and ROS, which creates a suitable environment for the survival of the parasite. So this way the agent is capable of involving into the cell signaling cascade. So now I move on to the discrete drugs available. They are the pentavalent antimalarial miltefosine, amphotericin B as well as the paromomycin. But due to the diverse side effects of these drugs, there is a need of alternative therapy. The alternative therapies may involve promising natural products, host-directed therapy, nanobiotechnology, drug repurposing. So presently the host-directed therapy through immunomodulation may be an alternative for treating the deadliest disease. So a particular mannose-binding lectin has been isolated from the nutshell of Arachis hypogaea and it has been reported to possess antioxidant activity, anti-inflammatory activity, cardiovascular as well as anti-leishmanial activity. So from these things we can hypothesize that the therapeutic importance of plant originated glycoprotein utilizing agricultural waste nutshell of Arachis hypogaea led to the hypothesis to explore its role in treatment of visceral leishmaniasis through M2 to M1 polarization. So the aim of our work is the potential of MBL-AHP in developing anti-leishmanial immune response through M2 to M1 repolarization and the mechanism through which it acts. So to fulfill my aim, the objectives that have been completed are to study the potential of MBL-AHP on macrophage polarization, repolarization and its anti-leishmanial effect in vitro, as well as the study of effectiveness of MBL-AHP towards anti-visceral immune activity in biopsy mice. So the overall experimental design includes MBL-HP, macrophages as well as L. donovani parasites. Upon availability of all the requirements, in vitro and in vivo experiments have been performed. In the in vitro experimentation, ability of MBL-HP on macrophage polarization and repolarization shall be checked, and parasite killing efficacy of MBL-HP through macrophage polarization shall also be examined. In case of in vivo activity, effectiveness of MBL-HP towards anti-leishmanial immune response have been checked. This is the animal clearance certificate. So now I move on to my results. The result shows that MBL-HP glycoprotein has been isolated, and by using SDS page, a single band of the glycoprotein had been obtained at 28 kilodalton, and it was the isolation process have been done by the standard protocols. For the next experimentation, this is the workflow in which peritoneal macrophages had been isolated and cultured for 48 hours and then accordingly seeded as based on this design, and initially the cells were pre-stimulated with recombinant proteins and left for incubation for 24 hours, followed by which it had been treated with MBL-AHP and RNA isolation cDNA synthesis was followed, which was then done, after which M1, M2 markers were checked through real-time PCR. So in the dose-dependent study, we had checked the mRNA expression of iNOS and arginase 1, in which we have seen that the expression of iNOS upon treatment with MBL-HP in a dose-dependent manner shows a gradual rise in the expression. But even we can see a gradual rise as well as significant level of increase at a dose of 2.5 microgram per ml when the cells were pre-stimulated with M2 recombinants. Again, in contrast, when we look at the arginase 1 activity, it can be seen that the M2 pre-stimulated cells showed gradual decrease in its expression upon treating with MBL-HP, and significant difference was observed at 2.5 microgram per ml. Looking at the effector response, the NO generation also showed similar response to that of mRNA expression of iNOS. So from this dose-dependent study, we can predict that 2.5 microgram per ml dose can be selected for further in vitro experimentation. So then we have checked the mRNA expressions of the M2 cytokines. Here I am showing the IL-10 mRNA expression, in which the expression of IL-10 decreased significantly in the M2 pre-stimulated cells as compared to its control, as well as there is a significant decrease in M1 stimulation also upon treatment with AHP at 2.5 microgram per ml concentration. Similar results were observed in case of IL-4. Sorry, IL-12 is a M1 cytokine. IL-12 is a M1 cytokine which shows exact contrast data to that of IL-10, in which the M2 pre-stimulated cells showed enhancement in the M1 cytokine upon treatment with MBL-HP as compared to its control. These are the M2 markers, which again shows that the expression of M2 decreased upon treatment with AHP and decreased with AHP in case of YM1, MSR2, CD206. M1 cytokines such as the IFN gamma, IL-6, TNF alpha showed showed significant enhancement in the level of its expression in case of M2 pre-stimulated cells. So from here we can conclude that MBL-HP could induce the M1 polarization through, as well as could repolarize M2 to M1. Now we check the effect of MBL-HP in L. donovani infected macrophages. So the cells were isolated through standard protocols and then infected in the ratio 1:10, followed by treatment with MBL-HP in a dose dependent manner 0.5 to 2.5, and then the parasite load and NO generation had been checked. So from there we can observe that in a dose dependent manner the parasite load had been shown to decrease upon treatment with MBL, and the maximum decrease in the parasite load was observed at 2.5 microgram per ml concentration. Nitrite generation also, this is the uninfected set and this is the infected set. Uninfected set was checked for the MBL treatment sequence. There also we observed that upon treatment with MBL, the nitrite generation enhanced significantly and the best result was obtained at 2.5 microgram per ml. The mRNA expressions of iNOS and arginase 1 as well as its modulation also also showed that the effector molecule response was similar. Now these are the expressions of all the TLRs in the infected macrophage, and after the treatment with MBL-HP, it showed that there was a gradual increase in the expressions of pro-inflammatory TLRs. From here we denote that 2.5 microgram per ml can be selected as a in vitro dose for infection model also. So the expressions of pro-inflammatory cytokines were also checked, in which interferon gamma L12 TNF alpha also showed a significant enhancement, while the IL-10, IL-4 and TGF beta showed a decrease in the expression upon treatment with MBL-AHP. So from here we conclude that MBL-AHP is capable of increasing the pro-inflammatory cytokine while decreasing the anti-inflammatory cytokine, leading to the killing of the parasites. Now we move on to the in vivo experimental design. We had taken 6 weeks old Balbc mice which had been infected with L. donovani infection and kept for 14 days, after which treatment was given to it. After a 12 days interval, the visceral organs such as the spleen, liver and the kidney were isolated, and splenocytes were taken for further experimentation. These are the group details, in which each group contained 6 mice. So then we check the effect of MBL-AHP on hepatic and splenic LDU as well as enogeneration and T cell proliferation. Upon seeing the hepatic LDU and splenic LDU, it can be seen that the hepatic parasite load decreased the most in upon using of AHP at a dose of 1 mg per kg body weight. Similar result was observed in case of splenic LDU. So based on our data, we can conclude that MBL-AHP at 1 mg per kg body weight was the most suitable for as an effective treatment dose against the L. donovani infected model. So then these are the Th1, Th2 cytokines. Here is the expression through the ELISA as well as the mRNA expressions of interferon gamma, IL-12 and Th2 cytokine like IL-10. So from here finally we can denote that MBL-AHP enhanced the Th1 expression by enhancing IL-12 interferon gamma and decreased the Th2 expression through IL-10. Hence, it may be a suitable candidate as a immunomodulator. So, I summarize my slides as HPMBL as an immunomodulator binds with the cell surface receptors, activates the P38 MAPK, inducing the pro-inflammatory cytokines, promoting inos-dependent nitrite generation, polarizing resting macrophages to M1. On the other hand, it deactivates the PERK1-2 MAPK, reduces the anti-inflammatory cytokine, leading to arginase 1 activity, transforming the polarization of M2 through M1, leading to parasite clearance. These are my references. Thank you. AG83, sir.
AG83 or something.
Wild type is called.
What is its normal host?
Normal host?
Normal host.
Okay, it is the sandfly, Flavotomin sandfly, in which vertebrate host is mammals, human host.
Which particular mammal? Does it infect man?
Yes, sir.
Or does it infect
humans?
Other
it can infect dogs as well as humans.
dogs?
Yeah, visceral leishmaniasis has been observed in certain dogs.
Survive in your mouse or something?
Sir, mainly it can affect humans. So overall, globally, it is namely Kala Jor, which it affects humans, through which and since Balbc mice is a Balbc mice.
Infected with this AG83.
Yes, sir.
If man or dog is the natural host, then how did it survive in murine body?
So because the murine body, it has...
It's a reservoir host or what?
It's the preliminary model for before the clinical trial begins. The preliminary model is the murine actually.
Next, whether vertebrates harbor the amastigote or promastigote?
Vertebrates, the amastigote form, sir.
How did you, for the first part of your experiment, you took macrophages.
Right.
How did you collect peritoneal macrophages?
Okay, so we in the
And one more thing, you collected such macrophages from infected animals or uninfected animals?
uninfected animals, sir.
Why did you not take infected animals?
So because then we have to take out, because we left the infected set for the in vivo experimentation, because we would require the entire organ for the, because we need the macrophages either from the spleen or any kind of visceral organs.
You took from peritoneum, not spleen, neither spleen.
We did not take any kind of organ from there, sir. We just, we injected thioglycolate into the mice about 5 days before our requirement, and then we used chloroform, and then we injected chilled PBS into it, into the peritoneal cavity, and then we collected the peritoneal macrophages. Since we had injected the thioglycolate 5 days ago, so the macrophages from the surrounding cells phagocytosed into the peritoneal cavity, and then through the chilled PBS solution we collected it, and then centrifuged it, and we got the macrophage pellets.
It did not stick to the glass surface?
No, sir.
And something, how did you isolate MBL AHP?
Okay, so we had a dust of those nutshell powder. We had the nutshell powder which is hypoglycin, and then from there we dissolved it.
It's a lectin.
Yeah. Finally, what we get in the purified form is a lectin, mannose binding lectin. It has been previously reported and proved that the glycoprotein from there, which I have been using in this model, it had been previously standardized by our group itself. So from there we got that that particular glycoprotein had been reported to be MBLHP lectin.
It forms a band at some 28 kDa or something.
এটা না সুমনদা আমার স্লাইডটা একটু ওপেন করে দাও
28 kDa region or something.
Yeah.
Please show that slide.
সুমনদা এটা নয় আচ্ছা স্যার এক মিনিট
এই যে এইখানে কোনটা হচ্ছে
এইটা স্যার 28
এটা 28 kDa তে তো
ওটা একটু নিচে
অনেক নিচে
নেমে
না
গেছে অ্যাকচুয়ালি অনেকটা নিচে নেমে গেছে
28 kDa
নো স্যার ইট হ্যাজ
12 or 13 or 14.
Yeah, it has come below 20 and 15.
Understand what I'm saying?
Yes, sir.
You were saying it formed a band at 28 kD.
But
you were
it's a W, it's coming to the 14 band kDa actually. It's a W band, double charge. So because of that, and it's a SGS page, so it is coming to the 14 kDa exactly.
Double charge 14 kDa হবে
Single band 14 है, वो जो ना double जेतु वो जो ना 28 kDa.
Total টা
हाँ सर.
সেটা একটু তুমি একটা লেকচার দিতে এসছো তো
ঠিক করে আনতে হবে
কাজটা ভালো
তোমার কম্পাউন্ডটাকে নিয়ে লোকে কোশ্চেন করে
না তোমার তো
এটা প্রি রিপোর্টেড স্যার হ্যাঁ হ্যাঁ
বুঝতে পারছো তো আমি এইখানে বসে বসে ঝিমাচ্ছি কিন্তু ঠিক জায়গা মতন ধরে ফেলছি তাই না তো তোমার তো একটা ঠিক করতে হবে আর একটা হচ্ছে কাজটা তো ভালো কিন্তু হচ্ছে অ্যাজ অ্যান ওরেটর ইউ শুড ইমপ্রুভ ইউর টকিং স্টাইল কারণ এটা ভবিষ্যতে যখনই লেকচার দেবে ইনিশিয়াল পার্ট শুড বি কনসাইজড। তুমি কি করেছো আর নিজে যেটা
তোমার এই কম্পাউন্ডার দিয়ে যখন তুমি কাজ করবে তখন একটা কোশ্চেনেবল হবে তুমি লিখেছো কি সিঙ্গেল ব্যান্ড 28 কেজি।
হ্যাঁ এটা ওই ডাবল ব্যান্ড অ্যাকচুয়ালি
সিঙ্গেল ব্যান্ড
পড়ে আসেনি লোকে তোমার লেসবেনিয়া কেউ দেখেনি।
ওই তো বলছি
মানে লেখাটা
লিখে দিচ্ছ
কম্পাউন্ডার ঠিক নেই সবার প্রথমে এটাকে
এখানে লিখে দিচ্ছ না
আগে তো লিখে
তাহলে এখন মানে আমার কোশ্চেনটা তো
না না ওটা ওই যেহেতু আমি বললাম ওটা সিঙ্গেল
লাল দিয়ে আবার দিন টিল হলেও একটা তাই না?
ঠিক আছে
যাও যাও যাও।
থ্যাংক ইউ স্যার।
Good afternoon to my respected professors, research scholars and my dear students. I am Pipika Das, working as a DST Inspire Fellow of Biodiversity and Environmental Studies Research Center under the supervision of Dr. Shaboni Pradhan, Assistant Professor in Nutrition, Department of Paramedical and Allied Health Science.
Your time starts now.
Today I am going to present a part of my research work entitled Dietary Interventions of Linoleidic and Docosha Pentaenoic acid N6-N3 ratios on adipogenesis and hepatic lipid metabolism on mouse model of diet-induced obesity. In the industrialized world, obesity is one of the epidemics associated with an increased risk of other life-threatening pathologies. Basically, obesity results from the disequilibrium of energy intake, that is food consumption, and energy expenditure, like the basal metabolic rate, that is the biochemical processes required to maintain cellular viability, physical activity, and adaptive thermogenesis. And I mainly focused the polyunsaturated fatty acid, and based on the high fat diet scenarios, polyunsaturated fatty acid exerts or limiting the incidence of the rate of increasing rate of obesity through the multiple signaling pathways. And the main goal of my research study to develop a potential bioactive compound by using polyunsaturated fatty acid. In this study, I have used linoleidic acid, which is a geometric isomer of linoleic acid, omega-6 acid, and docosha pentaenoic acid, which is another novel omega-3 fatty acid. And the objective of my research work to investigate the anti-obesity potentiality of the different ratio of linoleidic and docosha pentaenoic acid on high-fat diet-induced obese mice, and investigated the primary molecular mechanism underlying these effects. These are the concerned methodology, and this is the overall in vivo experimental design. At first, 42 male albino mice was divided into 4 groups. First 3 groups are control, which fed the normal diet group. Second one is vehicle control, which fed the normal diet with olive oil, which is used to dissolve the fatty acids. And third one is positive control group, which fed the normal diet with the standard ratio of omega-3 or omega-6 fatty acid. Fourth one is obese control group, which fed the lard-based high-fat diet, and following 12 weeks of high-fat diet, mice was again segregated into another 3 groups, which is co-administered with different ratios of omega-6 or omega-3 fatty acids. And the visual observation revealed that mice fed with the high fat diet showed the greater body weight than mice fed with the normal diet group, and the fatty acids or the different ratios significantly reduced the body weight as well as the BMI, especially in the last 2 weeks of the experiment. I also found a slightly altered food intake pattern in my study. And next I have examined the serum lipid profile, and the serum analysis depicted that there was significant reduction of upon the treatment of these two fatty acid ratio, that is 1:1 and 4:1 ratio of linoleidic and DPA, can inhibit or reduced the level of total triglyceride, total cholesterol, VLDL, LDL, and increased the level of the HDL in a ratio dependent manner, and a dysfunctional metabolism of glucose and lipid is directly linked to obesity. That is why I have also checked the fasting glucose level. The fasting glucose level also reduced in the treatment group in compared to the high fat diet group in a ratio dependent manner. As because liver is the main metabolic organ, I have also checked the liver function test by serum SGOT, SGPT level, and the fatty acid treated group significantly down-regulate or decreased the liver weight, serum GOT, GPT and ALP level, and also reduced the hepatic triglyceride and cholesterol level. And grossly, the livers of normal diet group shows the dark brown and red color of the liver, whereas the high fat diet treated group liver shows yellowish and lackluster, and the fish oil significantly recovered the dark brown and red color of the liver. And histologically, many lipid droplets and fat vacuoles were also present in the high fat diet group, which is also significantly reduced in the fatty acid treated group. And then the adipocyte quantity and adipose tissue weight were also measured in order to investigate whether the weight reducing effect of fatty acid is due to a decrease in fat mass, and these two mixture of fatty acid can significantly reduce the visceral and brown adipose tissue weight. And histological analysis demonstrated that there is significant reduction of the inner diameter of adipocyte size of the three adipose tissue. First one is visceral adipose tissue and subcutaneous and brown adipose tissue, and the most effective group was the last one, that is high fat diet treated group with linoleidic and DPA in the ratio of 4:1. And in order to investigate whether the fatty acid affect the adipogenic or lipogenic gene expression, I have also checked the several adipogenic and lipogenic markers by semi-quantitative PCR followed by agarose gel electrophoresis, and these results showed that there is significant upregulation of the adiponectin, GLUT4, CPT1 and PPAR alpha, which can induce the fatty acid beta oxidation and also upregulate the mRNA level of AMPK, which is the major regulator of the energy homeostasis. And the fatty acids also reduced the adipogenic markers like the fatty acid synthase and sterol regulatory element binding protein, which can diminish the de novo synthesis of fatty acids and the cholesterol metabolism by reducing the activity of HMG-CoA reductase, which is the rate limiting enzyme of the cholesterol metabolism, and also diminished the mRNA level of PPAR gamma, SCD1, TNF alpha, and also leptin, because the fatty acid has the capacity to reduce leptin resistance in obese mice. And the more similar result are also found in the real-time PCR. And I have also checked several protein expression of such markers, and the protein Western blot analysis demonstrated that there is also upregulation of AMPK, CPT1, PPAR alpha, adiponectin, IL-10 and GLUT4 in a ratio dependent manner, and also downregulate the protein expression of OP, SREBP1C, CBP alpha, PPAR gamma, FAS, SCD1 and TNF alpha, and more significant results are shown in the last 2 weeks of the last 2 doses of the treatment group. And this is the immunofluorescence staining of the treated group, and this result showed that the nuclear translocation of the AMPK by using counter stain DAPI and the fatty acid in the ratio of 1:1 and 4:1 can significantly upregulate the lipolytic pathway by AMPK activation and downregulate the adipogenic pathway by CEBP alpha, because it is the central regulator of the adipogenic or the adipogenesis differentiation. And this is the overall hypothetical mechanism by which the fatty acid can exert the lipid lowering activity. And based on the evidence shown in the present study, it may be concluded that the fatty acid treatment designed to 6-week study can significantly downregulate the adipocyte inflammation and upregulate the hepatic beta oxidation by regulating the hepatic lipid metabolism. And these are the references which I have followed to fulfill my study. And these are my related publication as a first author and as a co-author. And successful completion of any type of research work requires a help from number of persons. First of all, I would like to acknowledge the DST Inspire for providing me such a prestigious fellowship, and I would like to acknowledge the founder director of Midnapore City College, Professor Dr. Pradip Ghosh, and the principal and vice principal of this institute for providing their scientific guidance. And I convey my sincere gratitude to my research supervisor, Dr. Shaboni Pradhan, assistant professor in nutrition, Department of Paramedical and Allied Health Science. And last not the least, I would like to express my sincere thanks to my colleagues. Thank you. Okay, brown adipose tissue. These are
examine the cells under 10x magnification.
Yes.
And from your 10x examination, you are coming to certain inferences. Why do you not use 40x magnification for having clear views of your cells? What 10x se ki bojhe?
Na adipocyte.
আমার প্রশ্ন হচ্ছে 10x এ একটা মাইক্রোস্কোপে আমি কোনো জিনিস টিস্যুতে 10x এ যখন আমি দেখছি, it is hardly magnified, খুবই কম ম্যাগনিফিকেশন। And since you had microscope, you had 40x, why did you not use it for making your inference more clear?
Inner diameter of the adipocyte size, 40x যে কারণে in case of subcutaneous adipose tissue 40x এ তো আমি পুরো স্কিনের লেয়ারটা বোঝাতে পারবো না।
স্কিনের লেয়ার তুমি তো পাচ্ছ adipocytes
নো স্যার ইনা
টোটাল লেয়ার তোমাকে উত্তর করতে হবে। এবার জেলের ছবিটা একটু বার করো।
এটা
জেলের এই যে এটার ভেতরে তো কোনো স্কেলিং নেই, কোনো কিছু
মানে আগারোস জেল ইলেকট্রোফোরেসিস করলে আমরা যেটা লেয়ারিংটা আসে তো সেটা আমরা কন্ট্রাস্ট করে না না স্যার না কন্ট্রাস্ট করে স্যার যেহেতু অল দ্য রেজাল্ট আর
পরের ছবিটা দেখো, পরের ছবিটা পরে আরেকটা জেলার আছে, দেখলাম আরেকটা আছে এইখানে
ও মার্কার। এটা যেহেতু স্যার প্রোটিন, এটা প্রোটিন, সেই জন্য প্রোটিনের মলিকিউলার ওয়েটটা দেখিয়েছি। ডিএনএ এর এটা না প্রোটিনে না না প্রোটিনের ক্ষেত্রে আমরা মলিকুলার ওয়েটটা ইয়ে করি। হ্যাঁ টোটাল ছবি আমাদের র কপি সবসময় থাকে আমাদের কাছে। মানে এতগুলো মার্কারকে তাহলে আমি দেখাতে পারতাম না। হ্যাঁ বুঝতে পেরেছি। আচ্ছা তাহলে তোমাকে র ডেটা দেখাতে হবে। অ্যাট এ গ্ল্যান্স যখন আমি কোনো ফুল ব্লডটা তখন আমরা সাপ্লিমেন্টারি ফাইলে দিই প্রেজেন্ট। হ্যাঁ পুরো দেখাতে পারবো না, তার সাথে গ্রাফ। না না স্যার আমি
এটা একটা কম্পিটিশন হচ্ছে এবং আমি প্রথমেই বলেছি যে any supervisor should not interfere in order to not indulge in any kind of conflict of interest. তোমাকে তোমার ব্যাখ্যা দিতে হবে।
raw
টিচার
file
ব্যাখ্যা দিয়ে দিলে তাহলে টিচার
definitely
তুমি বুঝতে পারছো তো যে
হ্যাঁ
কি প্রশ্নটা
হ্যাঁ স্যার বুঝতে পেরেছি আমরা ফুল প্লটটা জেনারেলি
একটা ব্যাংকে দিয়ে দিলাম
হ্যাঁ ফুল প্লটটা আমরা জেনারেলি আরেকটা ফাইলে রাখি আর কি ইন কেস অফ একটা সিঙ্গেল পিপিটি বা একটা সিঙ্গেল ইয়েতে যখন আমি গ্রুপ করছি
তোমাদের সেটা রাখা আছে
হ্যাঁ হ্যাঁ ডেফিনেটলি
তুমি তোমার আনসার দাও
হ্যাঁ ডেফিনেটলি
যে কোনো রিসার্চার শুড নট টেক এটা যদি আগে সায়েন্স কংগ্রেস হতো তোমায় নামিয়ে দেওয়া হবে কোনো টিচার ইন্টারফেয়ার করতে পারে না এবং ছাত্র কোনো টিচারের অ্যাসিস্ট্যান্স নিতে পারে না আনসার করার সময় ইয়াং সায়েন্টিস্ট অ্যাওয়ার্ডে গেলে তোমাকে তো অ্যাওয়ার্ড দেওয়া দূরের কথা প্রথমেই নামিয়ে দেওয়া হবে বুঝতে পেরেছো আচ্ছা আর টাইম যখন দেওয়া আছে
with your permission, in our society, anywhere, the students, teachers can defend the students.
No.
It's the basic rights of the teacher. You cannot take
সেটা করতেই পারে কিন্তু হচ্ছে এটা তো একটা না এটা তো একটা কম্পিটিশন হচ্ছে এটা কম্পিটিশন আচ্ছা এইখানে তো একটা কম্পিটিশন হচ্ছে ন্যাচারালি এইখানে এটা না করাই ভালো কারণ তোমরা তো আস্তে আস্তে আরো বড় জায়গায় ছাত্রদের পাঠাবে না আরেকটা এত টাইম আছে তুমি একটু বেশি স্পিড বাড়িয়ে দিয়েছো বক্তৃতাটা আরেকটু
স্লো বললে ভালো
এগুলো একটু প্র্যাকটিসের অভাব
হ্যাঁ
আমি এই একটু প্র্যাকটিস করবি এটা
প্র্যাকটিস করেছে 8 মিনিটের জন্য আপনি 2 মিনিট বাড়িয়ে দিয়েছেন
না জেনারেলি 5-6 মিনিট থাকে তো এভরি ইয়ার যেখানে
না ওইটা না আরেকটা জিনিস এই যে আমি 3-4টে বক্তৃতা শুনলাম তোমাদের সব রকম আছে একটা লেজার টর্চ নেই যে তুমি তোমার বক্তৃতাটা
আচ্ছা আচ্ছা দেখিয়েছি আমি না আমি দেখিয়েছি
এগুলো দেখায় না
না দেখিয়েছি স্যার অ্যাডিপোসাইট ডায়ামিটার
আমি খেয়াল করি এটা একটু সরবরাহ
ওকে স্যার
চালাও
থ্যাংক ইউ
এটা আরো রয়েছে জিনিস তোমার ইউজ করো রিয়া কর 1 মিনিট ইন্ট্রোডিউস করো নিজেকে চলে যাও
Good afternoon, respected professors and all the research scholar and all my dear students present here. I am Riya Kaur, research scholar of Midnapore City College under the guidance of Dr. Shravani Pradhan, Assistant Professor in Nutrition, Department of Paramedical and Allied Health Sciences, Midnapore City College. Today I am here to present a part of my research work. Thank you, sir. So today's my topic is entitled Gondoic acid suppress adipocyte differentiation and lipogenesis related protein expression in 3T thiolon cells. As you all know that obesity is a global public health issue which is associated with several metabolic disorders. In molecular level, obesity is defined by adipogenesis. Adipogenesis is basically a process of conversion of pre-adipocyte to mature adipocyte via different activation factor like CBP alpha, PPR gamma, SRBP1C, etc. In nowadays, fish oil and fish oil derived fatty acids are gaining wide attention by the researchers for its healthy composition, where omega-3 and omega-6 fatty acids are well characterized and well established by the researchers. But however, omega-9 fatty acids have recently wide attention received due to its emerging studies. So now the question is why I choosing gondoic acid for my study. Gondoic acid is basically a 20 carbon monounsaturated plant-based fatty acid. For the very first time, we found this fatty acid in our experimental species, which is Polynemus paradiseus. It's locally named topse fish. From our GCMS study, we get this fatty acid accounted by 6.38%, which is more than the other monounsaturated fatty acids. So our main aim of our study is to establish the anti-obesity potentiality of gondoic acid to 3-3L1 cell line. 3-3L1 cell line is a well organized, well characterized and well known cell line which is used for check the effect of adipocyte differentiation. To complete my work, this is the overall methodology where I represent here. At first, we purchased the 3-3L1 cell line from NCCS Pune, then seeded the cell in DMM high glucose media with 10% fetal bovine serum, and after confluency of the cell, we use IBMX and dexamethasone for differentiation of the cell, and when the cell is converted into mature adipocyte, we cultured the cell into 6 and 9 to 6 pore oil plate and treated with several doses of gondoic acid from lower dose to higher dose. The doses was 0.25, 0.5, 0.75 and 1 micromole. And after that, we assess several parameters like cell viability assay, triglyceride accumulation, and check the pre-adipocyte differentiation by aldehyde staining, DAPI staining, etc. We check the mRNA expression of several adipogenic markers, as well as we check the protein expression by Western blot. Moving to the result and discussion part, to check the impact of gondoic acid in 3-3L1 cell line, here figure A represent the structure of gondoic acid first, and then we treated the cell with different doses of gondoic acid, where in figure B we can see the cell viability assay was not affected by the first 3 doses of gondoic acid, which was 0.25, 0.5 and 0.75. However, the last dose, which was the higher dose of gondoic acid, shows the slightly diminished of cell viability, as well as the TG accumulation layer. TG accumulation was also high in untreated control group, and the effectiveness is noted more in 0.5 and 0.75 micromole of gondoic acid dose group. Then we check the fat accumulation by aldehyde staining. Aldehyde is basically a dye which is used for differentiate for normal cell to fat cell. Here we can see in differentiate control group the fat accumulation was very high, and the mode effectiveness noted in 0.5 and somehow 0.75 micromole of gondoic acid group. Then we use a fluorescent staining, which was DAPI staining. DAPI staining is a non-cytotoxic dye. It's basically used to visualize the cell aggregation in aggregation, where you can see the cell aggregation was high in control group, which reduced in more 0.5 micromole of gondoic acid group. At the molecular level, to check the effect of gondoic acid, we check the expression of several molecular markers. Here you can see leptin, which is the more important polypeptide hormone, and the adiponectin are the anti-diabetic and anti-atherogenic hormone. These levels were decrease in control group, and after the treatment of gondoic acid is gradually increased more in 0.5 and 0.75 group. And AMPK is the most important factor which is responsible for upregulate the lipid metabolism by diminize the lipogenesis process and increase the lipolysis process. So AMPK regulation was increased in 0.5 and 0.75 micromole group, and the PPR alpha and CPT1, which is responsible for increase the fatty beta oxidation, its level was increased in gondoic acid 0.5 group of gondoic acid group, and the FAS and SRBP1C, which is responsible for activate the adipogenesis process, this level was in decreases in GA treatment group, as well as the inflammatory markers such as IL-6 level was decreased by treatment of gondoic acid, and the anti-inflammatory marker IL-10 and IL-1RA level was increased in gondoic acid treatment group. To validate our then we check the cytokine qualification by ELISA assay, where you can see the IL-1RA and IL-10 level was increased after the treatment of gondoic acid, and the TNF alpha level was decreased after the treatment of gondoic acid. More effectiveness group treated shows in 0.5 to 0.75 micromole gondoic acid group. To validate our result more accurately, we completed our Western blotting result by using several protein markers, where you can see the AMPK PPR alpha adiponectin, which is responsible for promote beta oxidation level. This level was increased in gondoic acid treated group, and the adipogenic markers level SRBP1C PPR gamma level was decreased in gondoic acid treated group. In conclusion, this is the very first study which prove that gondoic acid have a capability to inhibit adipocyte differentiation in 3T3L1 cell line by increasing the AMPK activation and reducing the expression of adipogenic markers, as well as increase the activation of PPR alpha and adiponectin, which ultimately reduce the adipocyte differentiation and reduce the obesity process. Here we use 4 different doses of gondoic acid. Among them 0.5 and 0.75 micromole dose shows more effectiveness than the other dose. These are some references. These are the list of publication of mine. These are the list of publication as co-author. I acknowledge Vidyasagar University to affiliating our research center as Biodiversity and Environmental Studies. I want to like to thank Dr. Pradeep Ghosh to provide me this nice platform to carry out my work, Dr. Shabani Pradhan for her guidance, and Dr. Sudipto Chakraborty and Dr. Kuntol Ghosh for their valuable advices, and thanks to all the research scholars and faculties. And thank you my research lab where I carried out my work, and my colleagues. Thank you.
How do you collect those pre-adipocytes? Obtain those pre-adipocytes?
Sir, we purchased this cell line from MCCS Pune, then we cultured this cell line in our lab. Then we perform our experiments.
And how did you maintain the adipocytes? I think you work both on pre-adipocytes and adipocytes.
Yes, sir. We purchased pre-adipocyte cell line first.
That was purchased, but adipocytes were collected from some animal?
No, no, sir. It's overall an in vitro study, not in vivo study.
Not in vivo, but when is your last slide? Just before this, here mature adipocytes were treated with different doses?
Yes, sir. Actually, we purchased the cell first, then we cultured the cell, then it transferred into mature adipocyte, then we treated it. Yes.
The effects on pre-adipocytes as well as the effects on mature.
Mature adipocyte, yes, sir.
What was the difference in effect in between pre-adipocytes and mature adipocytes?
Sir.
Effect of gondoic acid on pre-adipocytes and mature adipocytes.
Sir, gondoic acid can actually we check the expression of several markers which is responsible for induced mature adipocyte adipogenesis process. So we check the markers of this gene and see that after treatment of gondoic acid, this level was decreases.
Which level?
PPR gamma, SRBP1C, CBP alpha, these are the main prior gene which is responsible for induced adipogenesis process. So we check the expression of these markers.
The
levels were decreases.
And the levels are not increased for any parameter, for any of them upon?
No, sir.
Leptin level?
Leptin levels are decreases. Sir, increase the parameters like PPR alpha, which is responsible for induced beta oxidation, increase the level of adiponectin, increase the level of PPR alpha, which maintain the
You used 4 doses.
yes, 0.25, 0.5, 0.
Most effective was
0.5 to 0.75 micromole dose is.
What happened in case of 1 micromolar?
In 1 micromole dose, we can see somehow toxicity level is noted, like the expression level was.
That the effects were, it was most effective in the medium doses.
Yes, sir.
What is your idea about the decreasing the effect of highest dose?
0.5 micromole dose.
0.5 and 0.75 were effective.
Effective dose, yes.
I am asking why the effect of 1 micromole was not so effective. What is your idea?
Sir, I think it's.
When you are increasing the dose,
Yes.
initially the effects are good.
Yes.
For 0.5 micromole and
0.75.
But the effects are not so useful when you are increasing the dose up to 1 micromole.
Sir, like every drug have some adverse effect like not drug, it's fatty acid, natural fatty acid from Topse fish. Topse fish. Sir, we send the Topse fish oil for GCMS characterization. Then we listed the fatty acid, and among the all omega-9 fatty acids, we chose gondoic acid because this fatty acid is. No, sir, we sent the sample for GCMS characterization in Hyderabad. Only the characterization completed by them, but all the experiment. Yes, they sent back the list. Then we.
How did you use different doses of gondoic acid unless you procure gondoic acid? তুমি gondoic acid কি ওরা দেয়নি তোমাদের?
না পারচেজ করেছি স্যার, আমরা গন্ডোইক অ্যাসিডটা আমরা পারচেজ করেছি।
gondoic acidটা
পারচেজ করেছি।
আবার তোপসে মাছ থেকে gondoic acidটা অ্যানালাইজ করো।
হ্যাঁ, আমরা ক্যারেক্টারাইজড করেছি। হ্যাঁ, করে ওখান থেকে আইসোলেট করিনি, আমরা পারচেজ করে ইউজ করেছি। না স্যার, আমরা তো শুধু অয়েলটা পাঠাই ক্যারেক্টারাইজেশনের জন্য, ওরা একটা জিসিএমএস এর। হ্যাঁ, তার ভেতরে আমার 29টা টোটাল ফ্যাটি অ্যাসিড আছে, পুফা মুফা সব মিলে। 6.38% গন্ডোইক অ্যাসিড আছে, টোটাল 24.08% মুফা আছে। নো নো নো, জাস্ট অ্যানালাইসিস। Yes, for my.
Today I am going to present. The topic is the protective effect of nephritine isolated from Asparagus racemosa roots on cisplatin treated NRK52 E cell. Myself, Saurav Jana, is a scholar at Midnapore City College under the supervision of Dr. Suchimita Roy ma'am, Department of Nutrition. So, introduction part of my research work. Acute kidney injury and chronic kidney injury have become a worldwide public health problem and ranked with 9 leading cause of death in the world 2023 and prevalence affecting 200 million people per worldwide. Okay. Acute kidney injury are the multifactorial condition affecting 10 to 15% of all cancer patients in the intensive care unit and hospitalized, and more than 50% patients are intensive care unit. According to KDIGO guidelines, CKD gradually kidney function, both kidneys over time and increase patient morbidity and mortality risk. So here we choose the cisplatin as a drug for the drug model, for the disease model, and cisplatin is the chemotherapeutic drug, is the cis diamond dichloroplatinum 2. The cisplatin is usually treatment for the various cancers such as the lung, cervicular and testicular and bladder also. And however, major dose limiting side effect of cisplatin resulting the acute kidney injury. And cisplatin is prescribed 10 to 12% of all cancer patients due to its the slowing cancer growth. And the renal uptake and excretion of cisplatin mediated the localized transporter such as OCT2 and MAT1 is the localized transporter, and it's the consequently the cisplatin are accumulated in the proximal tubular cell resulting in the inflammation, injury and ultimately cell death. And previous study reported that those who take the drug there have many side effects. So we choose the asparagus therapeutic application of Asparagus racemosa. Okay sir, okay. Asparagus racemosa. Asparagus racemosa is a medicinal plants in Ayurveda. It is the several in Ayurveda. It is a tonic for several therapeutic applications such as the anti-cancer activities, anti-hepatoprotective activities, and also the anti-immunomodulatory activities also, and which possesses the augment the body defense system. And aim and objectives of my research work: protective effect of nephritine on cisplatin induced nephrotoxicity, attenuating through the NFKB-Q1 NRF2 signaling pathway. So to fulfill the following research work, following the methodology, first of all we analyze the HPLC and ESIMS studies, and after that the cell were purchased from the NCCS Pune, and then cell cultured were cell were cultured in the DMBM medium, F12 DMBM medium, and the 5% FBS and 1% Penicillium streptomycin, and the atmospheric research the 5% CO2 incubator for culture. And after confluency 60 to 80% confluency, the cell was stiffness, and the different group, the control group and the cisplatin nephrotoxic group, and the group 3, 4, 5 is the cisplatin with the nephritine at the 5 microgram, 10 microgram, 20 microgram, and last one is the cisplatin plus carcinogen. Carcinogen is the FDA approved drug. It's the protective effect of the nephrotoxicity. So we choose the carcinogen, and after we analyze the MTT assay and for the cell viability test, and the RT-PCR we analyze the cytotoxicity and the protein expression study were done using the Western blot, and the tunnel assay for the positive anti-apoptotic cell percentage, and the immunofluorescence stain also expression. And here the result indicate that the TLC plate were showed the the spots were visualized the UV transcellumeter, and the UV detected peak where spots were collected for further the HPLC analysis. The HPLC analysis showed the many minor peak, and the one major peak were separated, and these major peak were collected for further ESIMS study. The ESIMS study were conducted of the time of flight mass spectrophotometer. The ESIMS and mass spectrophotometer were operated using the positive ionization mode within the scan range 50 to 1900 mH, and these fractionation were separated the molecular weight basis, and the major peak were observed. The one is the atropine and another is the nephritine. And these two compounds having the many pharmacological activities. And here the nepetin is the nepetin 7O glycosidic compounds, and the group figure 3b indicate that the nepetin were the drug likeness properties 0.84. And also the cell viability assay to using the MTT at the different concentration of nepetin treatment with the NRK52E cell at the initially to determine any toxic effect. In briefly, the 10 to the power 3 cell were cultured in the 96 well plate, and after 24 hours cell were exposed at the different concentration of nepetin with the cell control well for 24 hours, and the different concentration of nepetin were significantly increased the cell viability, and we assessed the EC50 value to be 8.32 microgram per ml. And another study reported that the mRNA expression level of KIM1, IL-18, caspase-3 and NRF2 when we treated the nepetin at the different dose at 10 and 20 microgram per ml, there was significantly increased the NRF2 level and decrease the KIM1, IL-18 and caspase-3 as compared to the CP treated cytotoxic cell. And where we analyzed the ROS level were assessed using that H2DCF-DA stain, and here the ROS level was increased when we treated the cisplatin, and there was a significantly increase as compared to the control cell. Okay, and when we treated the nepetin at 10 microgram and 20 microgram, there was significantly reduced the ROS level as compared to the CP treated cytotoxic cell. And after the tunnel assay to analyze the quantify the positive apoptotic cell, and here the tunnel assay were showed the when the DNA damage, the tunnel assay were observed the high stain, and then the tunnel assay was significantly increased the CP treated cell when we treated the as compared to the control treated NRK52E cell. And after that, after we administered the nepetin 10 and 20 microgram, there was significantly reduced the apoptotic cell as compared to the CP treated cell. And the immunofluorescence stain by the using the NRF2 expression, and the here this cisplatin group where the showed the high nuclear translocation were analyzed by the DAPI stain. By the DAPI stain is the counter immunofluorescence stain, and the high translocation were significantly high translocation, so the expression is significantly low as compared to the control cell. And when we treated the nepetin, there was less amount of nuclear translocation using the DAPI stain, and the significantly high as compared to the CP treated cell. And similarly, the KIM1 level was significantly reduced when we treated the nepetin 10 mg and 20 microgram. There was significantly reduced as compared to the CP treated cell. And lastly, furthermore, we analyzed the protein expression study were done by using the DAPI, by using the Western blots, and these bands were visualized using the DAB stain. And here the NRF2 was significantly reduced when we treated the cisplatin in NRK52E cell as compared to the control cell. And when we treated the nepetin at 10 microgram, 20 microgram, there was significantly increased the NRF2 level and decrease the NFKB and KIM1 level as compared to the CP treated cell. And here we concluded that the cisplatin nepetin could able to protect the nephrotoxicity on cisplatin induced by regulating the inhibiting the NFKB signaling pathway and KIM1 and activation of the NRF2 signaling pathway. And these are the references. Cisplatin, sir, kidney damage.
What does it do? Kidney কি তুমি তো বলছো cisplatin দিয়ে ট্রিট করেছো অ্যানিমাল তাতে কি দেখছো তাই বলো।
আচ্ছা স্যার, এখানে হচ্ছে কি আমরা যখন স্যার সিসপ্লাটিন দিয়েছি তখন 20 মাইক্রোগ্রাম আমরা সিসপ্লাটিন দিয়েছি অ্যানিমেলকে দিয়েছি স্যার। এবার হচ্ছে কি যখন হচ্ছে সিসপ্লাটিন দিয়েছি মেজর ডোজ লিমিটিং সাইড এফেক্টটা সিসপ্লাটিন হচ্ছে কি স্যার অ্যাকুইট কিডনি ইঞ্জুরি করছে। এবার হচ্ছে কি স্যার সেখানে দুটো লোকালাইজ ট্রান্সপোর্টার আছে, যেটা হচ্ছে সিসপ্লাটিনকে এন্টার করতে দিচ্ছে না না ওটা নেপ্রোটক্সিক ড্রাগ স্যার অলরেডি এটা এস্টাবলিশড আছে, তাও স্যার আমরা এটাকে হ্যাঁ কিডনিতে স্যার ফার্স্ট স্যার ইনফ্লামেশন ঘটাচ্ছে, আরওএস প্রোডাকশন স্যার বাড়াচ্ছে ওই সিসপ্লাটিন স্যার অ্যাকুমুলেশন হয়ে। হ্যাঁ স্যার, এটা স্যার আমি যে কাজটা করেছি এটা স্যার সিসপ্লাটিনটাকে আমরা 30 মাইক্রোমোল স্যার ইন ভিট্রো স্টাডি করেছি, যেটা স্যার আমরা ইন ভিভো যদি করাতাম তাহলে স্যার আমরা হিস্টোপ্যাথ স্যার আমরা সেল NRK52 সেলের উপর করেছি, নর্মাল র্যাট কিডনি সেল স্যার, এপিথেলিয়াল সেল, প্রক্সিমাল টিবিউলার এপিথেলিয়াল সেল, ওটা আইসোলেট করা হয় একদম হ্যাঁ সাইটোটক্সিসিটির সাইন দেখেছি এখানে স্যার, স্টেনটা আমরা দেখেছি স্যার আরওএস প্রডিউস করছে, মানে সেলটা স্যার সাইটোটক্সি উঠেছে তার জন্য স্যার আমরা হচ্ছে MTT অ্যাসে করে স্যার বুঝতে পারছি আরওএস প্রডিউস হচ্ছে, বেড়ে গেছে MTT অ্যাসে স্যার করে। আচ্ছা স্যার, সিসপ্লাটিন স্যার এখনও স্যার ডিজিজ মডেল হিসাবে স্যার এখনও চলছে না, সিসপ্লাটিন স্যার এখন স্যার লেস অ্যামাউন্ট হচ্ছে, লেস অ্যামাউন্ট স্যার হচ্ছে নতুন, হ্যাঁ নতুন অন্য কোনো ড্রাগ আছে কিনা স্যার, এটা স্যার অনেক আগে তো হচ্ছে
আমি যখন কাজ করেছি তখন সে আজ থেকে 40 বছর এখনকার দিনে এইসব ড্রাগ খুব একটা বেশি ইউজ হয়। তোমার যদি কোনো ডাক্তার পরিচিত থাকে যার ফলে মানে তুমি মানে নেপ্রোটক্সিক অন্য আরো ড্রাগ যদি একটা সাথে দেখতে মনে হয় যেন PhD দেখতে সেটা ভালো। এখনকার দিনে যে জিনিসটা বিশেষ ব্যবহার হয় না সেই জিনিসটা শুধু দেখলে এরকম একটু ব্যাকডেটেড কাজটা আরেকটু যুগোপযোগী হয়ে এখন হচ্ছে যে সিসপ্লাটিন এটা কি নেপিট্রিন না সামথিং,
নেপিট্রিন
এইটা যে তুমি বললে আইসোলেট করেছো টরেছো অনেক কিছু HPLC আরো
ESIM স্টাডি করেছি
এইগুলো করলে কোথায়
স্যার HPLCটা স্যার আমি হ্যান্ডেল করি, HPLCটা আমি হ্যান্ডেল করি, আমি কাজ করি এখানেই করি, আর স্যার HPLC থেকে তারপর ESIM বোস ইনস্টিটিউট থেকে স্যার করেছি কলকাতায়। একদম স্যার, একদম একদম স্যার পেপারে অলওয়েজ।
আর একটা প্রশ্ন আছে
একদম স্যার,
কাজের,
হ্যাঁ স্যার
ওই নেপিট্রিনের সাথে যেন না সিসপ্লাটিনের সাথে নেপিট্রিন জুড়েছিল মানে অ্যাড করেছিল,
হ্যাঁ হ্যাঁ
ডিফারেন্ট ডোজ
কোটেটেড
5
মাইক্রোগ্রাম, হ্যাঁ 10 মাইক্রোগ্রাম 20 মাইক্রোগ্রাম। ওকে স্যার,
কিন্তু এই কোয়ারসেটিন নাকি একটা,
হ্যাঁ স্যার কারসেটিন হচ্ছে
ওইটা শুধু একটা ডোজ ইউজ করলে,
এটা স্যার একটা ডোজ ইউজ করেছি কারণ স্যার অলরেডি এস্টাবলিশ এই ড্রাগ FDA অ্যাপ্রুভ ড্রাগ স্যার অলরেডি 20 মাইক্রোগ্রাম 10 মাইক্রোগ্রাম 40 মাইক্রোগ্রাম দে আর অলরেডি ইউজড, সো দ্য
ওরা 40 আর কত ইউজ করেছে বলে তুমি 30 ইউজ করে দিলে নতুনত্ব কিছু হলো?
No sir, 20 microgram because of the nepentine 20 microgram, so the carcetin is the 20 microgram. The cost.
আমি বলছি নেপিট্রিনের দুটো ডোজ ইউজ করে, কিন্তু এই সেই স্লাইডটা দেখলে
হ্যাঁ স্যার, হ্যাঁ
ওই কোয়ারসেটিন না কি বস্তুটা সেইটা তুমি একটাই ডোজ লাগালে, হোয়াইট
এইখানে
তুমি ওই
না নেপিট্রিন
এই কিডনি ড্যামেজকে প্রিভেন্ট করতে পারে বা কিছু করতে
না স্যার এটা এখনো পর্যন্ত রিপোর্টেড প্রথম স্যার নেপিট্রিনের স্যার ডিফারেন্ট কাজ আছে ডিফারেন্ট ফার্মাকোলজিক্যাল অ্যাক্টিভিটি আছে অ্যান্টি ক্যান্সার অ্যান্টি হেপাটিক অ্যান্টি আলসার অ্যান্টি হাইপার কোলেস্টেরলেমিক আছে স্যার ফার্স্ট স্যার ফার্স্ট
আচ্ছা তা এক সেকেন্ড একটু স্লাইডটা একটু দাও চোখে দেখি আর ইউ শিওর যে তুমি প্রথম ব্যক্তি
একদম স্যার ফার্স্ট
তোমার লেখটা বলো দেখাও দেখাও স্লাইডটা দেখাও ওটা তো আমি একটু বললাম এর আগে আরো তুমি একটা দেখিয়েছো আরেকটু আগে আরো আগে এক জায়গায় তুমি এরকম একটা চার্টের মতন দেখিয়েছো
আচ্ছা স্যার এটা
এরও আগে
আরে
এই তো বলছি তুমি দেখালে তো আমি তো আর বানাই
এইটা
এটা নয় তুমি দেখাচ্ছ যে কি কি কম্বিনেশন নিয়েছো সিপি প্লাস
yes okay sir this is model yes sir
এই দেখো সিপিট্রিন এসিপি প্লাস নেপিট্রিন তিনটে ডোজ দিয়ে 5 10 20
yes sir
কিন্তু এই কোয়ারসেটিন বস্তুটা সিপি প্লাস সিঙ্গেল ডোজ দিয়ে
because sir 20 microgram nepentine or select the dose so the quercetin we select the 20 microgram the comparison the which are the better like the FDA approved drugs the compare the
যখন এক্সামিনার কথা বলবে তখন তুমি কথা বলবে কম্পারিজন যদি করলে 20 ভার্সাস 20 কম্পারিজন তাহলে 10 আর 5 টারও ভার্সাস দেখাবে না কেন
because sir already established drug the quercetin so the quercetins are already established FDA approved drugs sir
সে হোক না
so sir
সবাই কি তোমার সমানই ডোজ দিয়েছিল
না স্যার সেখানে
তুমি আমি বলছি নেপিট্রিন 20 কারসেটিন না কি উচ্চারণ
কারসেটিন কিউরেসেট
20 দিয়ে কম্পেয়ার করবে তাহলে নেপিট্রিন 10 আর কারসেটিন 10 এ কম্পারিজন করলে না কেন বা নেপিট্রিন 5 কারসেটিন 5 কম্পারিজন করলে না কেন তিনটে ডোজ যদি তুমি নিলে ওটা তাহলে এটারও তিনটে ডোজ দিয়েই তুমি কম্পেয়ার করতে পারতে এতে কি হচ্ছে তোমারই ডেটাটাই দেখা যাবে যে তুমি একটার তিনটে ডোজ নিয়েছো আর একটার একটা ডোজ নিয়েছো আমি কি বললাম বুঝতে পারছি তোমার যদি কম্পারিজনই করতে হয় তাহলে এর 5 ওর 5 কম্পেয়ার এর 10 এর 10 কম্পেয়ার করা উচিত ছিল না এবং এর 20 কম্পেয়ার করা উচিত ছিল বলে আমার মনে হচ্ছে দেখে
না স্যার ঠিক আছে
কি বললাম আমি বুঝেছো
একদম স্যার
তোমার বাঁ হাত ওনার বাঁ হাত অ্যাক্টিভিটি আমরা কম্পেয়ার করব তোমার ডান হাত ওনার ডান হাত তুমি একটা 20 আর এর 20 তার 20 কম্পেয়ার করলে ওইদিকে এর 5ও নিলে 10ও নিলে কিন্তু ওর কিছুই নিলে না বলছো যে ওইটা এফডিএ ড্রাগ ওর তো ভ্যালুলেস মানে এফডিএ ড্রাগ ওটা হোক আর একটা জঙ্গলি ড্রাগ যাই আছে আছে কিন্তু তুমি কম্পেয়ার করলে তিনটে ডোজেই কম্পেয়ার করে কিছু সিদ্ধান্ত দিলে বেটার হতো বলে আমার মনে হচ্ছে বুঝতে পারছেন কি বললাম
একদম স্যার
তোমার কাছে মাল ছিল না তাও না যদি নেপিট্রিন কিনে আর ওই কার্সেটিনটা কোথা থেকে জোগাড় করেছি
কার্সেটিনটা করলে কোথা থেকে কিনেছি স্যার আমরা হ্যাঁ সিগমা রেস থেকে স্যার কিনেছি পারচেজ করেছি হ্যাঁ
তাতে ওই একবার 20 ইউজ করার পরে সিসি শেষ
হ্যাঁ স্যার আছে আমরা
তাহলে 10 আর 5 এর সাথে কম্পেয়ার করে দিলে ভালো হতো না
আচ্ছা স্যার আমরা করব এক্সটেন্ড স্যার করব কাজ এখন বাকি আছে
ওখানে বসো তবে শোনো কিছু রাগ না করলে একটু লিটারেচারে দেখবে আমি যতদূর জানি সিসপ্ল্যাটিন এখনকার দিনে একটু কম ব্যবহার না আমি চেঞ্জ করতে বলছি আমি মডিফাই করবার স্কোপ সব সময় থাকে তুমি অ্যাটলিস্ট যে কোনো একটা করবে
Okay, sir. Good afternoon, respected chairperson, respected principal sir, respected professors present here, and all the research scholars and all of you presented here. Myself, Sojal Das Mahapatro, research scholar of Immunology Lab, Midnapore City College, working under the guidance of Dr. Shantanu Kaur Mahapatro. Today, I am going to present my research work that was entitled as Study of Eugenol and Amphotericin B encapsulated dual drug loaded liposome against experimental visceral leishmaniasis. Thank you. So, as my title suggested that I am working with the disease visceral leishmaniasis. So, I choose visceral leishmaniasis because it encounters 18% of global burden globally, and among 4 states, West Bengal has the endemic region for visceral leishmaniasis. So, visceral leishmaniasis is normally known as Kala-jar, and it was caused by the parasite called Leishmania donovani and transmitted by using the vector Phlebotomous sandfly. And a post-treatment effect of VL also found, that was termed as post-Kala-jar dermal leishmaniasis, shortly known as the PKDL. Now coming to the pathophysiological condition, Leishmania donovani has two dimorphic form, that was the extracellular motile promastigote form and intercellular amastigote form. These two forms are interchanged inside the macrophage cell. When promastigote enters into the human body, then it it enters into the macrophage cell, as macrophage acts as the reservoir for Leishmania parasite. So, Leishmania after entering into the inside the macrophage cell, it inactivates primarily the complement system of our body, by which it will suppressing the inos production and inducing the arginase expression, so which it will survive inside our body. So, to treat visceral leishmaniasis, there are some current treatment options also available, but the main major reason of that all these drugs got registered cytotoxic side effects and also the high price limited their uses to treat visceral leishmaniasis. So, to overcome it, there are combinatorial therapy for VL treatment also in clinical treatment also conducted in India, Bangladesh, in also Eastern Africa and Southern Sudan region. But this study also has some limitations like low generic barrier to the resistance, tetragenicity, gastrointestinal side effect, and short-term follow-up of this combinatorial study will limited their work. So, a better treatment option must needed to treat the visceral leishmaniasis. So, a possible treatment option might be the liposome mediated drug delivery system. So, liposome is a very spherical structure made with the cholesterol and phosphatidylcholine, and it was primarily have bilayer of phosphatidylcholine, and it was primarily discovered in 1960, and upon its discovery it being very much popular about its property like biocompatibility, stability, non-toxic nature. It can carry amphiphilic drugs inside it. So, Doxil is the first FDA approved liposomal product to treat cancer treatment. After this, FDA approved different liposome mediated drug delivery vehicle. Among these, Ambisome is very much popular to treat visceral leishmaniasis. It will enhance the efficacy of drug amphotericin B, but it will reduce it has several disadvantages like nephrotoxicity, hepatotoxicity, hypocalcemia, and risk of the relapsing the patient of immunocompromised patient. So, in our study, we are going to one step ahead to formulate dual drug encapsulated liposome to treat visceral leishmaniasis. For this, there are some previous reports. These are suggested that all this information. Then our group also established that eugenol and amphotericin B had showed the synergistic anti-leishmanial effects against experimental VL. So, from this information, my study was hypothesized that the proposed eugenol and amphotericin B dual drug loaded liposome formulation may promote the better treatment opportunity for experimental visceral leishmaniasis. So, the aim of my study is the study of dual drug loaded liposome with eugenol and amphotericin B against visceral leishmaniasis. And to fulfill my study, I have two objectives. The primary objective is to study the anti-leishmanial efficacy of DDL against L. donovani parasite and L. donovani infected macrophages. And the second one is study the in vivo anti-leishmanial efficacy of dual drug loaded liposome in preclinical biopsy mice model. My in vitro work plan is that the peritoneal macrophage isolated from biopsy mice, and it was cultured and incubated in CO2 incubator. And for conducting this study, I have I conduct anti-promasticode cell cytotoxicity assay, anti-amasticode assay in dose dependent manner, and then the mRNA expression and the cytokine release study was conducted in fixed dose study. For these experimental set, I segregated my experimental models into different groups like uninfected macrophages, infected macrophage with dual drug, and infected macrophage with empty liposome with different dose, and infected macrophage with DDL with different doses, and with the standard drug that was the ambisome with standard dose. Okay, sir. From all these group, I'll check the parasite load of by gene sustaining and release and expressions of the effector molecule, and pro and anti-inflammatory markers expression, and mRNA expressions and the release also. So now coming to the result and discussion part, primarily I prepared the DD dual drug loaded liposome by using the thin film hydration technique, where for the phosphatidylcholine and cholesterol was used in 7:2 molar ratio, and the drugs were used in 8:10 ratio. Then it was characterized by polydispersity index, zeta potential and zeta season analysis was conducted to characterize this liposome, and it will show that these expressions will show that the liposome has a homogenized structure with a diameter of 50 to 200 nanometer, and the drug entrapped and drug release study also conducted, where we find that amphotericin B drugs were entrapped more or less 82%, and eugenol drug was entrapped inside liposome, that was 90 to 94%. And the drug release assay showed that at the time interval of 60 minutes, amphotericin B release rate was 76.53% and eugenol rate was 81.54%. The spectrophotometric study also found that both the peaks was found for eugenol and amphotericin B in DDL. That was summarized that the eugenol and amphotericin B, both the drugs were encapsulated inside the liposome. So then after we are going to the check the parasite burden by using the gene sustaining method, and where we found the in dose dependent manner, where we found that the dose that DDL 2.5 microgram per ml, it has a significant reduction of the parasite burden inside the macrophage, that was nearly 88.88% in comparison with the infection control and the infected macrophage with the dual drug. So after that, we check the cell cytotoxicity assay of both dual drug loaded liposome and the empty liposome. We found that from 0.5 to 5 microgram per ml, there are no significant cytotoxic effects was found of this liposomal product. Then we check that effector molecules response and release. There we found that the nitrite release also significantly increased with the 2.5 microgram per ml dose in comparison with the dual drug and infection control, with significantly increase of the ionos 2 mRNA expression, where the arginase 1 expression was significantly reduced in with that dose 2.5 microgram per ml. So from all these dose dependent study, we conclude we selected 2.5 microgram per ml DDL dose for the further in vitro study. Now we have checked the anti and pro-inflammatory markers expression in protein level by the sandwich ELISA method, and genomic level by the quantitative PCR method. So here we found that the pro-inflammatory markers like IL-12, IF and gamma, these markers were released and expressions was severely increased, significantly increased with the dose of PDL 2.5 microgram per ml in comparison with the infection control, whereas the anti-inflammatory markers like IL-10, TGF beta expressions were significantly reduced, and the reduction fold change was more or less 6.56% for IL-10 and TGF beta that was 4.87%. From this we can conclude that the DDL with 2.5 microgram per ml can boost the activity of the drug and has an anti-leishmanial activity. So now coming to the in vivo work plan, where biopsy mice was used for our study, and infection was done through the tail vein, and 2 weeks in rest for establishing the infection. After the treatment was conducted, there were 3 doses of dual loaded liposome was treated through the tail vein, and after the sacrification of mice, the splenocyte was isolated from mice, and for experimental set I segregate my set into different groups, where empty liposome and dual loaded liposome was in dose dependent ratio. From these all these group we will check the LDU in liver and spleen, and the Th1, Th2 cytokine released by real-time PCR and the sandwich ELISA method. So in vivo study, when we check the parasite burden, we found that DDL with 1 mg per kg body weight, it will reduce the parasite burden inside the splenocyte. Like in liver, it was 93.13%, and the splenic LD was reduced by 95.55% with the dose of DDL 1 mg per kg body weight. Now coming to the effector molecules release and expression, where we found that the nitrate generation and ionos 2 expressions were significantly increased with the DDL dose of 1 mg per kg body weight, where in contrast the arginase 1 expression was reduced with that particular dose. This data was confirmed by the T cell proliferation and IL-2 release, where we found that after treatment with DDL this dose, the T cell proliferation was significantly increased, and the IL-2 release also significantly increased in comparison with the infection control. From this data, it will signify that the DDL with 1 mg per kg body weight can have the better anti-Leishmanial activity. This data was confirmed by the study of pro and anti-inflammatory cytokine release and the expression, where IL-12 So now coming to the pro and inflammatory markers, where we found that the IL-12 and IL-12 release and IFN gamma release with the expressions of IL-12 mRNA and IFN gamma mRNA were significantly increased with that dose, and the anti-inflammatory markers like IL-10 and IFN TGF beta expressions were significantly reduced with that particular dose. From this information, we found that the DDL with that particular 1 mg per kg body weight can boost the activity of the drug by inducing the host mediated therapy. So in now coming to the summary and conclusion part, where we found that the Leishmania donovani parasite were in phagocytosis into the macrophage cell, and after phagocytosis completion, it will transform into the amastigote form. It can form the infected macrophage cell, and the liposome product was intravenously injected inside the blood vessel, and it will reach the visceral organ, where it phagocytosis into the macrophage cell. Upon being phagocyted inside the macrophage cell, it will release the drug, and that will help to reduce the parasite load inside the visceral organs. And these are my references. And lastly, I would like to acknowledge the Honorable Director and Founder, my guide, for being helpful to. Thank you. No, sir. Non-infected mice. স্যার ইনফেক্টেড মাইস থেকে আমরা স্প্লিনোসাইট আইসোলেট করেছি, স্প্লিনোসাইট আইসোলেট করে দেন আমরা স্যার ওখানে চেক করেছি যে ওখানে রেসপন্স কীরকম আসছে, ওখান থেকে প্যারাসাইট লোড বা ইফেক্টর মলিকুলের রেসপন্স চেক করেছি ওখান থেকে। হ্যাঁ স্যার, হ্যাঁ। স্যার অ্যাকচুয়ালি ইনফেক্টেড অ্যানিমালের ম্যাক্রোফাজ ছিল, কিন্তু সেখানে আমাদের যে স্টাডির জন্য যেহেতু লিসমেনিয়া ডোনোভানি যে প্যারাসাইটটা ওটা মোস্টলি আমাদের ভিসেরাল অর্গানকে ইনফেক্ট করে, সেই সেন্সে আমরা স্যার ওর থেকে বেটার রেজাল্টের জন্য ভিসেরাল অর্গান থেকে আমরা ওটাকে কালেক্ট করেছি। হ্যাঁ স্যার। হ্যাঁ হ্যাঁ, একটু মডারেট স্পিড হলে বেটার।
সেটা গেল একটা জিনিস আমার জানা উচিত। আর হচ্ছে আমি একটা জিনিস জানতে চাই, এই যে জিটা পোটেনশিয়াল মাপলে আর একটা কি মেপেছো?
পলিডিসপারসিটি ইন্ডেক্স। পলিডিসপারসিটি ইন্ডেক্স।
জিটা পোটেনশিয়াল প্লাস
জিটা সেজার অ্যানালিসিস অ্যান্ড পলিডিসপারসিটি ইন্ডেক্স। হ্যাঁ,
এগুলো দিয়ে তুমি ওই প্ল্যাঙ্ক বা এমটি লাইপোসোমটা
হ্যাঁ আমরা ওর ক্যারেক্টারটা বুঝতে পারছি। হুম।
তো হচ্ছে আমি একটা জিনিস জানতে চাইছি, এই যে অ্যাম্ফোটেরিসিন বি আর একটা কি
ইউজিনল অলিয়েট,
এই দুটোর রিলিজের রেট ফ্রম লাইপোসোম কি সেম হয়?
না স্যার সেম হবে না।
এটা না তোমরা সেই বিষয়ে কিছু দেখলে না? আমি জানতে চাই যে ইউজ টু ড্রাগস ইনসাইড এনক্যাপসুলেটেড
হ্যাঁ ইনটু লাইপোসোম।
লাইপোসোম।
ইয়েস স্যার।
So whether those two drugs were released in the same rate or not?
সেম রেটে আমরা চেক করিনি, কিন্তু সেম টাইম ফ্রেমে ওটার রিলিজটা আমরা দেখেছিলাম যে 60 মিনিট আফটার 60 মিনিট যার রেট মোটামুটি 76% থেকে 80%-এর কাছাকাছি ছিল দুটো ড্রাগেরই। হ্যাঁ স্যার ওটা চেক আমরা হ্যাঁ হ্যাঁ স্যার চেক করেছি আমরা, ওর পরে ওই স্পেকট্রোফটোমেট্রিক
দুটো কিভাবে হাউ ডু দে অ্যাক্ট অন মিসম্যাচ? মানে দুটো কি সেম কাজ করে?
দুটো অ্যাকচুয়ালি স্যার লাইপোফিলিক ড্রাগ, এগুলো লাইপোফিলিক ড্রাগ দুজনেই, তো এরা খুব ইজিলি সেলের ভেতরে ইনকর্পোরেট হতে পারে। এমনি সোললি যদি আমরা একে ইনজেক্ট করি তো নর্মালি সেলের ভেতরে খুব ভালোভাবে ইনকর্পোরেট হতে পারে, কিন্তু প্রবলেম হয়ে গেছে যেটা হচ্ছে সেটা এই ড্রাগ রেজিস্ট্যান্সের বা হচ্ছে টক্সিক সাইড এফেক্ট। সিমিলার মোরলেস স্যার সিমিলার মোড অফ অ্যাকশান দুজনেরই। এবার স্যার ওদের সিনার্জিস্টিক ইফেক্টও আমরা স্টাডি করেছি, বা আমাদের প্রিভিয়াস স্টাডিতে আছে সিনার্জিস্টিক এফেক্ট রয়েছে দুটো ড্রাগের টুগেদারলি। তো সেই সেন্সে স্যার ওরা মানে ওইভাবে দেখতে গেলে ড্রাগ রিলিজ হওয়ার পর ওরা প্রায় একই মেকানিজমেই ওরা লিসমেনিয়াকে কিল করে। হ্যাঁ একটা নিলে হতো, হ্যাঁ হ্যাঁ অবভিয়াসলি স্যার ওটা হতো, যদি আমরা সিঙ্গেল ড্রাগ নিয়েও কাজ করতাম তাহলেও হতো, কিন্তু অ্যাকচুয়ালি সিঙ্গেল ড্রাগ লোডের লাইপোসোম অ্যাম্বিজোম অলরেডি মার্কেটাইজড হয়ে আছে, অ্যান্ড অ্যাম্বিজোম বলে একটা প্রোডাক্ট মার্কেটাইজড হয়ে আছে যেখানে লাইপোসোম উইথ অ্যাম্ফোটেরিসিন বি, অনলি অ্যাম্ফোটেরিসিন বি হ্যাঁ সেখানে রয়েছে, না ওটা ছিল না। তো আমরা হ্যাঁ হ্যাঁ আমরা স্যার হ্যাঁ 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 না স্যার না, অ্যাম্বিজোমকে আমরা অ্যাজ এ স্ট্যান্ডার্ড হিসেবে রেখেছি এখানে, আমরা আলাদাভাবে লাইপোজোম প্রিপেয়ার করছি যেখানে অ্যাম্ফোটেরিসিন বি অ্যান্ড ইউজিনল অলিয়েট দুটোকেই আমরা ইনকর্পোরেট করা। হ্যাঁ স্যার নিজেরাই বানাই, এখানেই বানিয়েছি। না স্যার পিএইচডি হ্যাঁ স্যার হ্যাঁ স্যার হ্যাঁ সিঙ্গেল ড্রাগে স্যার অ্যাকচুয়ালি যেটা সিঙ্গেল ড্রাগের প্রবলেম সেটা হচ্ছে যে অ্যাম্ফোটেরিসিন বি এর ক্ষেত্রে স্যার প্রচণ্ড সাইড এফেক্ট রয়েছে, আর অ্যাম্বিজোম সব থেকে প্রবলেমেটিক যেটা হচ্ছে যে ভিসরাল লিসমেনিয়াসিসের ক্ষেত্রে ও পেশেন্টকে মানে ট্রিটমেন্ট প্রথম ট্রিটমেন্টের হওয়ার পরে পেশেন্ট ইজ কিউরড, বাট আফটার সাম ডেজ তাদের কিন্তু রিল্যাপস করার প্রচণ্ড পরিমাণে চান্স, মানে গ্রেটার দেন 95% চান্স হয়ে যায় ওখানে। তো ড্রাগ স্যার মারবে, ড্রাগ নর্মালি স্যার অ্যাম্ফোটেরিসিন বি কিন্তু প্রাইমারিলি স্যার ফাঙ্গিজোন হিসেবে কাজ করে, মানে ফাঙ্গাল ইনফেকশনকে কমায়। দেন আফটার দ্যাট ওটা ইন্ট্রোডিউস হয়েছিল ভিসরাল লিসমেনিয়াসিসের ট্রিটমেন্টের জন্য। তো ওর স্যার সোললি ড্রাগেরও কিছু সাইটোটক্সিক সাইড এফেক্ট রয়েছে। হ্যাঁ স্যার স্যার রিল্যাপস হয় তাদের ক্ষেত্রেই যাদের মানে ইমিউন সিস্টেম প্রথম থেকেই উইক, প্রথম ব্যাপার। আর দ্বিতীয় নম্বর কথা হচ্ছে হ্যাঁ হ্যাঁ মোস্ট এফেক্টিভ ড্রাগ। হ্যাঁ এক্স্যাক্ট অ্যান্সার তা জানি না, কিন্তু মানে আমার যেটুকু মনে হচ্ছে যে এটা তাদের ক্ষেত্রেই হয় যাদের ক্ষেত্রে ইমিউন সাপ্রেশন প্রথম থেকে হয়ে গেছে। হ্যাঁ স্যার হ্যাঁ রিল্যাপস হয় স্যার হ্যাঁ
100% তো আর ছাত্র জানবে না।
না না এটা বলে দেবো।
আচ্ছা সেটা পরে শুনে নেবে।
ঠিক আছে।
তো একটা লাইন আর একটা এমনি জিজ্ঞেস করেন তোমার এই ইন্ট্রনটা কোথায় গেল? মানে ধরো শান্তনু
কর্মপাত্র
পর মহাপাত্র
স্বজন দাস
ওইটাই জিজ্ঞেস করেছি যে ইন্ট্রনটা বদলালো কি করে?
আমার স্যার আলাদা আলাদা কি পর আর মহাপাত্র আলাদা।
ও পর আর মহাপাত্র আলাদা।
ওরা হচ্ছে একসাথে।
ঘর আর মহাপাত্র একসাথে।
হ্যাঁ ওর দাস দাস আর
না ইন্ট্রনটা বদলালো আলাদা আলাদা।
হ্যাঁ আলাদা স্যার।
আরেকজন আছে।
থ্যাংক ইউ স্যার, থ্যাংক ইউ।
Good afternoon all, my respected teachers, my students and research scholars. I am here to present my work. My research title is Health Status and Nutritional Status Assessment of Seafish Consumer who lives in Ramnagar 2 block, Paschim Medinipur. Sorry, Purva Medinipur. Sorry. I am Snigdha Shahu, research scholar of Belda College. My title, Health Status and Nutritional Status Assessment of Seafish Consuming Population of Ramnagar 2 block, Purva Medinipur, West Bengal, India. Then the people who live in the coastal area and consume low cost and easily available seafish daily over a long period and help to reduce the prevalence rate of different types of metabolic disorders. Tapra fish oil in the mice, they exhibit that the tapra fish oil as well as applied is to evaluate anti-obesity potentiality. When the fish oil was applied to the high-fat diet-induced obese mice, it shows significant reduction of body weight, body mass index, serum lipid profile compared to the high-fat diet-induced obese mice. Research gap. The identified research gaps points towards the need for ongoing exploration to refine our understanding, ultimate screening, the course towards evidence-based dietary recommendation and sustainable practices for the benefit of global health. Aims of my studies. The aim of the research was to study the health status, nutritional status, socio-economic status of the selected communities in the coastal areas of Ramnagar 2 block, Purva Medinipur, West Bengal. And objectives to measure anthropometric parameters, height, weight, body mass index, waist hip ratio, skin fold thickness in the different age group in selected sample population in the study area of coastal region, to measure physiological parameters like blood pressure, respiratory rate, pulse rate in different age groups in selected sample population in the study area of coastal region, and to determine the health status with reference to hemoglobin, lipid profile, creatinine level in the different age groups in selected sample population in the study area of coastal region, to compare the analyze the assessed parameters among different age groups and reference data through statistical software. This is experimental design. I mainly experimental groups 20 to 59 years, adult male and adult female, and inclusion 200 sample size, and this is anthropometric parameters are physiological parameters, biochemicals and socio-economic status by Kupusomic socio-economic scale. This is materials and methods. This is study area Ramnagar 2 blocks, and this is basically community based study and applied golden cluster sampling methods. And Ramnagar 2 block divided into 4 gram panchayat, and all participants signs informed consent before data collection of the data by directly interacting with local people regarding the types of fish consumed, for 6 days in a week. চলছে না এটা? And this is justification for choose the study area. Nutritive value: sea fish are rich source of protein, omega-3 fatty acids, vitamins, minerals essential for overall health and well-being. Assessing the consumption pattern and nutritional intake can help identify deficiencies or excess in the diet, local dietary pattern, public health implications and availability. And this is selection of sample population and sample size by these methods. And a total of 200 study participants will be selected randomly. Blood sample were collected from 100 participants for biochemical tests due to budget constraint, but anthropometric, physiological parameters, clinical assessment were conducted for all. Anthropometric parameters are height, weight, BMI, waist tip ratio, body fat percentage, and these are biochemical parameters and physiological parameters. Blood pressure by oscillatory methods, respiratory rate by manual counting methods, pulse rate by standard palpatory methods, and hemoglobin by cyanohemoglobin methods, lipid profile, colorimetric methods, and creatinine level by semi-auto analyzer using standard kit. And this is result and discussion. 20 to 24 years female, their height 161.2 and this plus minus 0.62. This is significant different from when is compared with the reference value. Weight is also significant different and BMI significant different, waist tip ratio also and body fat percentage also. And 20-24 years male, their height 176.9 plus minus 0.47 and their waist tip ratio is significant different, their body fat percentage is significant different. And 25 to 29 years males, their body fat percentage is significant different, and 25 to 29 years male, their BMI and body fat percentage is and waist tip ratio also significant different. And 30-39 years females, so that weight significant difference, body fat percentage and waist tip ratio and BMI, so significant different. 30 to 39 years male, their height and BMI, waist tip ratio and body fat percentage so significant different, and 40 to 49 years female, their weight, BMI, waist tip ratio and body fat percentage so significant different, and 40 to 49 years male, their height, weight, BMI, waist tip ratio, body fat percentage so significant different. And 50 to 59 years female, their height and waist tip ratio, body fat percentage so significant difference, and 50 to 59 years male, their height, weight, BMI, waist tip ratio, and 50 to 59 years female, their height and BMI and waist tip ratio and body fat percentage so significant difference. And this is biochemical parameters. 20 to 24 years female, their cholesterol level and their LDL level, VLDL level, creatinine level and hemoglobin level so significant different. And 20 to 24 years male, their cholesterol level, triglyceride, HDL, LDL, VLDL, creatinine and hemoglobin level so significant different. And 25 to 29 years female, their cholesterol level, their triglyceride level, HDL level, LDL, VLDL and hemoglobin level so significant different. And 25 to 29 years males, their cholesterol level, triglyceride, HDL, LDL, VLDL, creatinine so significant different. And 30 to 39 years female, their cholesterol level, triglyceride, HDL, LDL, VLDL, creatinine, hemoglobin so significant different. 30 to 39 years male, their cholesterol level, their HDL level, LDL level and VLDL and creatinine level also. So significant different. And 40 to 49 years female, their cholesterol, triglycerides, HDL and VLDL level, hemoglobin level, and 40 to 49 years male, their cholesterol, triglyceride and LDL and creatinine level and hemoglobin level so significant different. And 50 to 59 years female, their cholesterol level and triglyceride, HDL, LDL, VLDL, creatinine and hemoglobin level, and 50 to 59 years male, their cholesterol level, triglyceride, HDL, LDL, VLDL and hemoglobin levels were significant different. And this is physiological parameters. Their blood pressure, 20 to 24 years female, their blood pressure and diastolic, systolic, diastolic and their pulse rate, so significant different. 20 to 24 years male, their respiratory rate and pulse rate. 25 to 29 years females, their diastolic pressures and pulse rate. And this is socioeconomic status of this sample. Socioeconomic status using the major finding and see. The mean of anthropometric characteristic was normal according to the socioeconomic status. The mean value of biochemical indices was normal according to age, sex, physiological activity and socioeconomic condition. Most of the people like to eat fish, sea fish and they consume it daily. The main species they eat are ruli, vetki, sardine, lote, and most of them depend on the sea fish business for their livelihood. The mean value of physiological indices was normal range, and socioeconomic status score was the best indicator for underweight, occupation of the head of the family, for standing and undernutrition and overnutrition by BMI. And its conclusion, sea fish is nutrient dense fish and rich in protein, omega-3 fatty acids, omega-6 fatty acids, minerals and vitamins. In the future, an increase in the lifestyle related disease, the majority of which are a result of dietary habit, is expected in both developed and developing countries. There is evidence that increased consumption of sea fish and bioactive components could have a positive impact on the health of the people around the world. Thus, the role of sea fish in the maintenance and enhancement of the health may grow stronger and give the problem of lifestyle related disease and local food environment. Regular consumption of sea fish shows beneficial impact on several health indicators. Sea fish consumption is associated with improved cardiovascular disease, metabolic, hematological overall health, nutritional health, including sea fish as part of balanced diet, provide protective benefits and support healthy physiological functions. And all of these are references, and this is an ethical certificate. And I sincerely acknowledge Dr. Madhuchanda Iduwari for her valuable guidance, and I express my gratitude to Dr. Rupa Dasgupta for her helpful suggestion and cooperation. Deep gratitude is extended to Dr. Dilip Kumar Nandi for his expert advice, motivation and kind support, which greatly contribute to the successful completions and presentation of this work. Thank you. I have PhD scholar.
Registration করেছো? Registered. So, আমার বক্তব্য যে total number of people কতজন দেবে?
1200. Total all Ramnagar 2 block total people 1 lakh.
তুমি কতজনের উপরে?
আমি এখানে স্টাডি করছি, টোটাল হচ্ছে 200.
তাহলে তুমি অনেকগুলো ভাগে ভাগ করেছো।
24 হ্যাঁ 25 29 ইয়ার্স। ওকে স্যার, হ্যাঁ সরি স্যার, ঠিক আছে ঠিক আছে ঠিক আছে ঠিক আছে।
25 to 29 এরকম বিভিন্ন করেছো। তো আমার কথা হচ্ছে তাহলে কোন গ্রুপে কয়টা করে লোক করলো? ধরো তুমি কতগুলো ভাগ করেছো বলো?
10টা।
10টা কি মেল 10?
না, মেল এন্ড ফিমেল মিলে, মেল এন্ড ফিমেল, হ্যাঁ 5টা ফিমেলের গ্রুপ।
এক সেকেন্ড, আমি বলে 10টা গ্রুপে ভাগ করেছো 200। তার কন্ট্রোল কি?
রেফারেন্স গ্রুপ ধরে স্যার করেছি, WHO এন্ড ICMR এর যে রেফারেন্স ভ্যালু সেটার সাথে কম্পেয়ার করেছি।
এক্সামিনার কথা বলে তখন ছাত্ররা কথা বলতে নেই, এটাই তো। এই যে যে ডেটাটা দিচ্ছে তারা কোন দেশের ডেটা দিয়েছে? তুমি যে ডেটাটা নিচ্ছ কম্পারিজনের জন্য, তারা কি আদৌ ওয়েস্ট বেঙ্গলকে নিয়েছিল বা ইন্ডিয়াকে নিয়েছিল?
ইন্ডিয়া, হ্যাঁ স্যার, ইন্ডিয়ার যে ডেটাগুলো আছে সেগুলোই নিয়েছি।
ইন্ডিয়ার ডেটা নিয়েছে
age groups 20.
আরে 20 থেকে 24 বছর মেল কয়জন নিয়েছো? 20 থেকে 24 বছর ফিমেল নিয়েছো কয়জন?
20 20 জন করে নিয়েছি। 20 জন করে নিয়েছি।
সব গ্রুপে 20 জন করে জোগাড় করেছো?
না, মেল ফিমেল 10
মেল 10 জন, ফিমেল 10 জন। তাহলে একটা মানুষের মতন বিশাল স্পিসিস। আমি শান্তনুবাবু উনি এরকম 10 জনের ওজন মেপে বলতে পারি যে মানুষের গড় ওজন এত। ইজ ইট অ্যাকসেপ্টেবল স্ট্যাটিস্টিক্যালি, বায়োলজিক্যালি কোনো দিক?
না, ওটা স্যার হিউজ স্কেলে, ওটা তো স্ট্যাটিস্টিক্যাল অ্যানালাইসিস করে।
কথা হচ্ছে মানুষের মতন একটা জীবের উপরে যখন রিসার্চ করা হচ্ছে তখন এই 10টা করে মানুষ নিয়ে সেই ডেটাটাকে গ্রহণযোগ্য ধরাই যায় না। এক একটা গ্রুপে কম পক্ষে 100 জন করে দিয়েছি আমি, তাহলে এটা একটা যে অ্যাকসেপ্টেবল হয়। একটা
না, বেল্লা
একটা গ্রুপে 10 জন করে মানুষ নিলে ওটাকে ঠিক স্ট্যাটিস্টিক্যালি অ্যাকসেপ্টেবল বলা হয়। আমি 2 নম্বর কথা হচ্ছে যে মানুষের উপরে কাজ করতে গেলে ইনফর্ম কনসেন্ট নিতে হবে। ইনফর্ম কনসেন্ট এই 200 জন লোকের থেকে নিয়েছিলেন, তাদের চিঠি আছে? আচ্ছা, সেটা গুড, নিয়ে থাকলে ভালো। এথিক্যাল কমিটি আছে তোমার কলেজে ওয়ার্কিং অন হিউম্যান?
আচ্ছা,
এটা কি মানে নিজে নিজে বানানো এথিক্যাল কমিটি হলে হবে?
এটা কলেজ থেকেই প্রোভাইড করেছিল, সরকারিভাবে লয়ার, ডক্টর সবাই ছিল।
সবই মানে স্বীকৃত বডি আছে?
হ্যাঁ,
গুড, সবই ঠিক আছে। তাহলে কিন্তু হচ্ছে দুটো জিনিসের মূল দিকটাই, একটা হচ্ছে মানুষের যদি আমি ধরি, আমি একটা জিনিস তার ভেতরে আবার একটা দেখলাম। তুমি দেখো হিমোগ্লোবিনের যেটা যদি দেখি প্রায় পুরোটাই কেউ নেই, সেমি মানে আরো কম বুড়ো টাইপের লোকেরা রয়েছে। প্রায় বুড়ো 50 থেকে 59 বয়সীদের তুমি দেখছো গড় হিমোগ্লোবিনটা দেখাও 16.21, যেটা ইজ অলমোস্ট আনলাইক। তাহলে এটা কি করে দেখাটা না একটা নিত্যয় লেখা? এই যে দেখো এটা দেখলাম কত বুড়ো বয়সে হিমোগ্লোবিনটা 50 থেকে 59 বছরে 16.21, যেখানে ছোকরা বয়সে 14.something। তারা কি এত কি খেল একটু সন্দেহ হতে পারে।
আমি তো সব কালেকশন করে মানে
আমার বক্তব্য ইজ ভেরি সিম্পল। যদি সাধারণভাবে কি হয় 59 বছরের লোকের হিমোগ্লোবিন গড় হয়ে গেল 16.21, সেইখানে ছোকরাদের হয়ে গেল 14.something, এটা একটু অ্যাবনর্মাল থাকে। তারপর রেফারেন্স হচ্ছে ধরো ওয়ার্ল্ড হেলথ অর্গানাইজেশন রেফারেন্স করবো, সেইখান থেকে এটা জাম্প করে এটা একটু আনলাইকলি ঠেকছে। আর তারপরেও যদি সেটা যাই হলো হলো, এখন কথা হচ্ছে মানুষের মতন একটা জীব নিয়ে যখন কাজ হচ্ছে, মানুষের ক্যারেক্টারস আর হাইলি ভ্যারিয়েবল। ওই শুধু ওই মেরিন মাছ কি খেয়েছে বলে তার হিমোগ্লোবিন বেড়ে গেল, তার হেলথ বেড়ে গেল, বা তার নিউট্রিশনাল স্ট্যাটাস বেড়ে গেল, এটা খুব একটা বিশ্বাসযোগ্য না। মানুষ খুব ডাইভার্স।
কোন গ্রুপের সঙ্গে কোনটা ধরুন 59 ইয়ার্স, তো ওর হিমোগ্লোবিন লেভেল 16.21 প্লাস মাইনাস 0.49।
কম্পারিজনও এইটা একটা পেপার করতে গেলে স্ট্যাটিস্টিক্যালি টি টেস্ট করে দেখাতে হবে। ওরকম 16.15
এগুলো তো স্যার টি টেস্ট করেছি,
তার সঙ্গে কম্পেয়ার করে সিগনিফিকেন্ট লেভেল
এটা রেফারেন্সের সাথে কম্পেয়ার করেছি।
রেফারেন্সের তো সিঙ্গেল ভ্যালু,
এটা তো টি টেস্ট করা যাবে না।
আর হচ্ছে গিয়ে আমার এক্সপেরিমেন্টাল মডেলে হচ্ছে কি
ওটা স্যার মিনটা নিয়েছি, এটার মিনটা নিয়েছি 10 জনের যে মিনটা নিলাম, মিন ভ্যালুটা, আর ওটার ভ্যালুটা নিলাম।
আর যেটা টেস্ট করতেই তো পারবে না, ওইটাই কত স্যাম্পল, নাম্বার অফ স্যাম্পলটা ওর যে ডেটা দিচ্ছ, আমার টি টেস্ট করতে গেলে n1 n2 দুটোই লাগবে। তোমার এখানে নয় 10টা মানুষ কোন কয়জনের উপরে কাজ টেস্ট করবে, কি বললাম আমি বুঝতে পেরেছি? আমি এইখানে 10 জন নিলাম আর ওইখানে 12 জন নিলাম, টি টেস্ট হবে। এইখানে 10 জন নিলাম আর ওইখানে খালি একটা ফিগার কয়জন তার নাম নেই।
এটা আপনার যে 16.21 বেরিয়েছে না,
হ্যাঁ
এটা 100 এর উপরে যদি স্যাম্পল সাইজ আছে তাহলে ওটা কমে কম স্যাম্পলের জন্য ওটা
হয়তো দুইজন তিনজনের
আর এই সিঙ্গেল
তারপরেও এটা তো বললাম যে 59 ইয়ার্সে 16.21 হয়ে গেল, আর যেখানে 20-25 বছরে 14 সামথিং, এটা একটু ডিফিকাল্ট মান। তারপরেও কথা হচ্ছে ওই মেরিন, সবচেয়ে বড় কথা হচ্ছে ওই কি মেরিন মাছ খেয়েছো তাতে তোমার সব কিছু ভালো হয়ে গেল। সিদ্ধান্ত মানুষের ক্ষেত্রে আসে না। মেরিন ফিশ খেয়েছে, আরো নানান রকম কত রকম পুষ্টিকর খাবার খাচ্ছে, তাই না? সানডে আর মানডে খাওয়া হয়, আন্ডে আরো কত রকম খাচ্ছে।
বেসিক্যালি স্যার ওরা লো সোশিও ইকোনমিক কন্ডিশন যাদের, তাদের উপর যাদের সোশিও ইকোনমিক কন্ডিশনটা লো এবং যারা বেসিক্যালি সমুদ্রের নিকটবর্তী এলাকায় বসবাস করেন তাদের উপরেই কাজটা করেছি। তারা বেসিক্যালি সি ফিশটাই খান। হ্যাঁ সে না বেসিক্যালি ওরা 7 ডেজের মধ্যে 6 ডেজ সি ফিশ।
আর দুজনে সেম ডায়েট হতে হবে, মানে হিউম্যানে কি হচ্ছে কন্ট্রোলারই নিজে কন্ট্রোল না নিয়ে কোনোদিনও স্টাডি হয় না। আমি ওয়ার্ল্ড হেলথ অর্গানাইজেশনের ডেটা দিয়ে নিজেকে কন্ট্রোল নিতে হবে এবং এজ ম্যাচ, সেক্স ম্যাচ,
স্যার এজটা তো
যত রকম ম্যাচিং করানো যায় এইভাবে কন্ট্রোলটা নিতে হবে।
হ্যাঁ স্যার ওখানে আসলে এজ সেক্স ওগুলো দেখেই করেছি। WHO বা ICMR এর যে ভ্যালুগুলো আছে ওটাকে রেফারেন্স ভ্যালু ধরেই একটা, হ্যাঁ সিঙ্গেল ভ্যালু আছে না, ওটাকে স্যার মিন করেই করছিলাম।
মানে আপনি যেখানে কাজ করবেন এরা যেমন এরা তাদের কন্ট্রোল নেওয়ার দরকার নাই, বইয়ে লেখা আছে বলে লিখে নিতে পারেন না, কন্ট্রোল নিজেকে নিতে হবে। এটা খুব ডিফিকাল্ট টু এস্টাবলিশ, খুব ডিফিকাল্ট টু না, জিনিসটাকে পুরো খোলমলছে বদলাতে হবে। হ্যাঁ পুরো খোলমলছে বদলাতে হবে, বুঝেছেন না? এর পুরো খোলমলছে বদলাতে হবে, এইভাবে পিএইচডি দাঁড়াবে না, এইভাবে পিএইচডি দাঁড়াবে সব জিনিসটা সাইন্টিফিক হতে হবে। আমি ওই এরিয়ার সেম এজ গ্রুপ, সেম ধরুন স্কুল ছাত্র যদি 10 জন নিলেন, 10 জন
না না আমি বুঝতে পারছি কথাটা আপনার। হ্যাঁ 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 হ্যাঁ
না বার করা খুব কঠিন। এরা এই 10 জন সি ফিশ খায় রেগুলার, আর এরা আবার মধ্যে মধ্যে খায়, ও তো কন্ট্রোল রাখে না, একদম খায় না।
দেখেছে যে এভরিডে সি ফিশ কনজাম্পশন
যারা নিয়মিত খায় আর যারা এরা খুব বলা খুব কঠিন।
না স্যার ওই সমুদ্রের সি কোস্ট এরিয়া লোকেরা সি ফিশ
আর যারা খায় না মাছ সবাই খায়, এরপর গুড, এর কন্ট্রোলটা কি হবে? কিছু নেই তো, যারা মাছ খায় না আপনাকে 10 দিন না খাই রাখতে হবে, যারা মাছ খায় না এরকম লোক নিতে হবে। মাছ খায় সবাই খায়, সবাই কেউ খায় না, তাহলে তো তারা প্রায় সব লোকই সি ফিশ খায়।
না আপনি অবাকার স্যার, আপনি কন্ট্রোলটা রাখতে পারেন তো তার কি করে?
অন্য পাশে যে কোনো গ্রাম
যেখানে
একটু দূরে,
এমনি নরমাল পপুলেশন
একটু দূরে।
বুঝলেন?
হ্যাঁ ঠিক আছে ঠিক আছে। হ্যাঁ হ্যাঁ একটা ব্লক কমপ্লিট হয়েছে, 5টা ব্লক। আচ্ছা ঠিক আছে।
আর ওই যে বললাম স্যাম্পলটা যদি আপনি 10টা গ্রুপ করেন, এত 10টা গ্রুপে করার দরকার নেই, 4-5টা গ্রুপ নিলেও নাম্বার অফ পার্সনসটা বাড়াতে হবে। মানুষের উপরে এই 5টা লোকের ডেটা দেখে কখনো কোনো সিদ্ধান্ত গ্রহণযোগ্য হবে না। 100 করে প্রত্যেক গ্রুপে নিলে মানুষের জন্য এই 5-7টা মানে 10টা স্যাম্পল ইজ নট গুড এনাফ।
এটা এক্সপেরিমেন্টাল অ্যানিমেল হয়, কেন হয়? সেখানে একটা কন্ডিশনে অ্যানিমেলটা গ্রো করে একই রকম
ল্যাবরেটরিতে পালন করছে, এরা একই খাবার খাওয়াচ্ছে, সবকিছু ইউনিফর্ম করছে, কন্ট্রোলার ঠিক আছে, এক্সেস দিচ্ছে।
হ্যাঁ হ্যাঁ ঠিক আছে হ্যাঁ হ্যাঁ
Number of sample is too small, আর মানুষে কখনোই সিদ্ধান্ত করা যায় না যে আমি শুধু সি ফিশকে আমার ক্ষেত্রে ভালো লাগে
স্যার এটা না এখানে স্যার আমি আরেকটা কথা বলছি যে এটাতে আমি আমার যে রিসার্চ ওয়ার্কটা আছে ওটা হচ্ছে অ্যাডোলেসেন্স গ্রুপটাও আছে অ্যাডাল্টও আছে আবার জেরিয়াট্রিক পিরিয়ডটাও আছে তো এখানে আমি একটা স্টেজকে এখানে রিপ্রেজেন্ট করেছি আমার কাজের পুরো কাজটা এখানে নেই তো ওটা
এনাদের সাথে একটু কথাবার্তা বললে বুঝতে পারবেন যে কি করা উচিত
স্যার অনেক লাভের লোকজন আছে তারা করে ফেলবে স্যার আছে রূপা ম্যাডাম আছে সবাই করে ফেলবে আর কি আছে
একটু ঠিকঠাক করতে হবে নাম্বার অফ পিপল
হ্যাঁ টেকনিশিয়ান ও ক্লোজ
তো বটেই
ঘোষণা করতে হবে তো আপনাকে
যারা ঘোষণা করে দিতে হবে
হ্যাঁ
আচ্ছা
আমরা যেতে পারছি না
ঠিক আছে 1 সেকেন্ড বাবা এসআইআর শেষ হলো হিয়ারিং শেষ আইন সহ বলবেন এমনি এসআইআর হিয়ারিং শেষ
থ্যাংক ইউ ফর হাজিরা